Oh iya, oh minta uang Pak.
Halo, Assalamualaikum Pak Yuki. Pak.
Assalamualaikum Bu.
Waalaikumsalam. Masih di kampus Pak hari kedua atau sudah berpindah lokasi atau bagaimana?
Saya masih di kampus Bu. Oh, masih di kampus Bu.
Yang lainnya gimana Pak? Tetap seperti kemarin?
Ini udah ada Dokter Mira, Dokter Eka udah datang.
Oh, udah ada ya, tapi belum.
Iya, lagi join.
Oke, siap. Saya cek belum ada. Yang YouTube teh dijadiin co-host nggak ya Pak? Lupa saya.
Nggak usah kalau yang itu biarin.
Oh, nggak usah Di.
Oke, Bu.
Nggak usah dijadiin co-host. Pak. Nanti monitor ya Pak, siapa yang sudah datang biar kita admit dan jadi co-host ya. Sembari sarapan ya Pak.
Siap Bu.
Sembari sarapan.
Kemarin lupa ganti dulu virtual background. Nah.
Oh iya, nah.
Banyak kan sih mahasiswa dari ini lagi.
Tapi kayaknya kemarin nggak semua ya Pak mahasiswanya ya?
Kalau kemarin kan emang di suratnya kita dibagi dua, ada yang join di Zoom, ada yang di YouTube. Tapi semalam sudah saya share lagi, buat semuanya hari ini full di Zoom dulu lah. Kalau udah mentok baru gitu ke YouTube, diarahin ke YouTube.
Ya benar Pak.
Jadi sekarang diarahin dulu ke Zoom semuanya.
Benar.
Support from rector, lecturers, staff, and student activities.
Kebetulan kan kalau kemarin emang masih, kalau yang koas kan masih ada di bagian, lagi ada kegiatan. Terus yang tingkat 4 kebetulan lagi ada sosialisasi ini pesantren. Dokter Mira. Nah, Dokter Mira udah, Eka.
Bu Abe sudah datang belum ya Pak?
Assalamualaikum.
Belum.
Waalaikumsalam Bu Mira. Ibu punten saya sambil sarapan ya, jadi
Mana itu ya?
Eka tuh.
Yang dari Prodi teh diwakilkan oleh siapa ya Pak ya?
Nah, saya nggak tahu namanya. Yang dari Prodi, bentar saya konfirmasi dulu ya.
Namanya.
Halo, assalamualaikum Bu.
Waalaikumsalam Bu Mira. Ibu udah sarapan Bu?
Belum, udah sih. Cuma masih lapar.
Sambil sarapan Bu. Ini video dulu.
Tadi di mobil aja sarapan.
Aduh.
Ini Pak, ini bagus kan ya? Mukanya terlihat halus tidak?
Bagus Bu, udah gelis Bu, udah cantik.
Udah cantik begitu ya. Tadi soalnya makan saya. Ada sisa-sisa selai nganter ya. Hah? Oh, gimana nundukin lagi Bapak? Pas nanti patah. Kalau patah gimana, ganti dong. Ini juga Pak, saya pendek. Ini kali ya minta apa namanya?
Bandung City is the largest metropolitan city in West Java province.
Minta bantal.
Also known as education city, which has a scientific treasure.
Monitornya nggak bisa diinin Bu, di bawah.
Iya, dari kemarin tuh monitornya tinggi memang di meja Bu, nggak bisa diginiin. Dia udah pakem Bu, nanti patah. Ya,
based
jadi
on the ideas
begini ya.
of several
Saya harus
Muslim
tegak,
thinkers.
tapi kalau tegak
As a higher
aja
education,
sih ya.
which has an
Tinggian
Islamic
memang.
academic cultures,
Pak Yuki lagi
as its
mencari sesuatu yang
educational
bisa ditarik
strategy,
menurut saya.
philosophically, Universitas Islam
Bu
Bandung
Santi nanti untuk
offers a
moderator
comprehensive
gitu
education
ya.
concept
Aku
based on
Siap,
the
aman,
Islamic
Bu.
principles.
boleh aku ini
Since
nggak
its
di share
establishment,
apa namanya
Unisba has
profilnya?
launched 10
Oh, boleh.
faculties
Sekarang
and
gitu.
bachelor
Boleh.
degrees.
Boleh ya?
As
Mohon.
Oke, coba.
As follows.
Biar aku bisa melancarkan
Syariah,
ngomong gitu. Isi belepotan.
dakwah,
Iki. Oke, sip, mantap. Bu, yang plenary ya? Yang
Ya, yang plenary Tatsuya Iso sama Dr. Badrul.
Ya.
Ini udah datang belum beliau ya? Sudah datang belum ya beliau ya?
Oh, nanti ini yang baca Bu Mira.
Iya.
Oh ya. Ini anaknya gimana ya?
Gimana? Gimana, Mak?
Dikirim di chat Zoom.
Enggak usah gini aja. Jadi Bu Mera baca di screen. Maksudnya gimana, Ki?
Mau di copy gitu ininya.
Baca di screen aja kan, Bu ya?
Oh, ibunya lagi nyari sarapan lagi. Enggak kan?
Coba yang satu lagi, ya.
Aku udah jadi co-host ya, Mbak Santi ya?
Udah.
Iya.
Boleh di-share by WA nggak sih ke aku Mbak Santi ini? Atau
Kalau ke chat Zoom gimana, Bu?
Ya,
Ini
boleh sih,
ujungnya
nggak
masih
apa-apa.
sama?
Di chat juga nggak apa-apa.
Di screen capture gitu ya?
Ya, boleh.
Assalamualaikum, Bu Eka. Masih
Ya, aku masih
masih
mute.
menggema. Oh iya. Ibu
Tapi
udah
kurang ini kayaknya ya. Bentar dulu.
udah sarapan, Bu Eka?
Iya, minggu. Terus kumaha coba.
Ah, Yusuf udah ada.
Ya, Spira. Kamu pendampingannya pun. Ya, salah kayak
Aku juga bilang ke yang IKM kan, di IKM ditanyainnya siapa aja yang belum ngurus jam. Siapa cenah yang belum ngurus jam.
Santi.
Itu Mbak Santi, Pak Yusufnya tolong di-approve.
Oh iya, siap. Tolong dijadiin
Mohon. Ini
sudah ya. Sudah masuk. Saya masih sarapan dikit.
Saya mau belum sarapan.
Waduh.
Teh Santi berarti ini masih sama, mau di-announce event introduction-nya lagi, diulang lagi berarti ya?
Ya, mohon Pak. Pak, suaranya agak
Mira. Sebentar, dimatiin dulu.
Bu Mira sudah saya kirim ya, Bu ya?
Iya.
Pak, masih nggak? Masih ya?
Cek. Udah enggak?
Ya. Harus
ada sedikit bocor, tapi nggak apa-apa.
Assalamualaikum.
Pak Yusuf.
Gimana?
Oh iya, bocor Pak suaranya.
Menggema di sini suara. Ini lagi pada dibuka semua kebetulan.
Oh iya.
Belum pada ditutup soalnya.
Siap. Tadi Bapak mau nanya apa Pak? Oh disebut ya tetap Pak diulang penjelasan tentang eventnya ya, karena kan 7 hari.
Punten Dokter Mia. Di kohosin dulu.
Cek.
Assalamualaikum Bu Mia.
Waalaikumsalam.
Gimana Bu? Sehat?
Alhamdulillah. Saya langsung Zoom tadi malam Bu. Ada rapat yang lain sampai jam 9:30.
Masya Allah.
Aduh, kata saya teh.
Lanjut, field drop Bu judulnya.
Sekarang mah apa jam 4 kan ya Bu ya?
Jam 4 lebih lah. Tapi kalau misalnya karena kan keynote speaker-nya yang dari Denmark kan baru mulai jam 3 ya Bu?
Iya. Oh iya ya, paling 4:30 mungkin.
Iya, 4:30 kayaknya.
Pak Yuki, hari ini gimana biar ada ininya, soalnya baru. Harus buka email ya.
Cek 1 2 3.
Di Facebook aman, Di?
Aman, Bu.
Oke, sip. Bu Abe, monitor sudah masuk ke Zoom belum ya?
Belum ya?
Belum ada, Bu.
Belum ada, Bu.
Udah saya telepon ya.
Boleh. Sudah sampai, Bu, di atau di rumah beliau?
Bilangnya mau ke sini tapi nggak tahu.
Oh gitu, karena kan beliau welcome remark ya?
Ntar saya telepon aja.
Oh iya, siap-siap. Sama yang dari Prodia, udah ada namanya belum ya, Bu ya?
Sebentar.
Bisa mau disebut gitu, atau nanti
iya,
kalau...
ada sekedap, Bu.
Oh iya, siap-siap.
Ibu merah menyala. Saya langsung kata suami saya, nah. Bu, saya udah WA ya, Bu, yang dari Prodia namanya.
Oke, siap.
Nah, sama kemarin teh, Bu, berapa orang bebas aja kata saya mau masuk mah ya, Bu ya.
Iya, bebas aja, Bu, masuk aja.
5 orang juga boleh, 6 orang.
Iya.
Dikasih nama Prodia aja ya, belakangnya biar gampang saya gitu. Harus ngomongin, bukan?
Gimana, Bu?
Dikasih rename-nya pakai belakangnya Prodia biar gampang gitu.
Oh, boleh. Boleh, mangga. Maksudnya diinfo ke beliaunya ya?
Ya.
Oh, boleh, mangga. Oke. Berarti nanti
Di sini Pak, biji dari supaya saya nggak heboh.
ini tuh yang dari Prodia ya, benar ya?
Ya. Itu mah yang mau itunya, yang mau apa? Mau nanti presentasi yang 15 menit itu, Bu.
Oh, dokter anak ya?
Ya.
Dokter anak nanti. Oh, Pak Yusuf? Pak?
Ya, gimana Teh?
Saya udah kirim namanya, Pak. Untuk yang representatif dari Prodia ya.
Oh, oke.
Nanti mungkin bisa disebut ya kayak kemarin ya. Represented by gitu ya. Please welcome dokter Ana.
Bisa pakai Bluetooth?
Ina BJ apa Teh?
Ya,
Ina BJ apa? Ina BJ?
Apa Pak?
Publikasi Ina BJ apa ya?
Internasional jurnal apa gitu kayaknya Pak.
Oh, itu singkatan ya?
Ina BJ itu singkatan Pak, itu
dari Prodia kan?
Prodia.
Oh, jurnalnya
Prodia ya?
Oke, sebentar.
Berarti as the publication managing editor gitu Pak. Publication managing editor of Ina BJ. Jurnal itu Teh, Bu?
Ini coba dicari singkatan.
Ina BJ.
Oh.
Belum masuk ya.
The Indonesian Biomedic Journal.
Oh, The Indonesian Biomedic Journal.
Biomedical Journal.
Yes.
Lagi. Nyala kan?
Oke. Halo, monitor Iki. Ki di WA ya, Ki?
Ya, Mbak.
Ayah tuh
Di WA udah gimana? Oke.
Ada.
Di WA apa? Oh,
saya mah nggak suka pakai itu.
Aku WA.
Jangan mau kalian di sini aja.
bisa Mbak, bisa.
Bisa ya? Mantap. Oh ya kan juga share screen ya?
Bentar.
Lagi Bu Yani. Aduh, Ibu batuk.
Alah, menit lagi Mbak.
Oh, mikrofonnya.
Batuk-batuk.
Jadi ini sebelah kiri.
Mbak Santi, kalau yang
Iya.
Prodia nanti presentasi sendiri kan?
Ya, presentasi sendiri. Nanti
Oh,
aja
ya.
berarti ya. Sip,
Cukup.
aman ya?
Ya,
Bu Mia,
deg-degan sih.
Ya Bu.
Bu Mia nanti dipanggil untuk token appreciation ke dokter anaknya ya. Nanti saya
Baiklah.
panggil ya. Oke.
Ini sebelah kiri ya?
Ya, sebelah kiri.
Sama Pak.
Udah 1 menit
Iya.
lewat.
Ayo di-admit semua ya Bapak Ibu ya.
Oh, boleh.
Recording in progress.
Bandung City is the largest metropolitan city in West Java Province, which is also known as an education city, which has a scientific treasure and has many universities. One of these is Universitas Islam Bandung, which has established since 1958, based on the ideas of several Muslim figures. As a higher education, which has an Islamic academic cultures, as its
Ladies and gentlemen, may I have your attention, please? The opening ceremony of the second day of the BICMHS is about to begin. For this reason, we would like to inform for all online participants to activate the camera on their respective Zoom application and for participants in the meeting hall to get ready.
Check. And before we start the program, we would like to inform you about the general rules during the conference. Please ensure your computer sound or volume control is at an appropriate level. You may not hear the conference if it's disabled or turned down. Please use a headset or earbud if available to reduce external noises. Identify your Zoom account with your actual name. Stay seated and stay present. Do this appropriately. Be aware of your surroundings. Do not interrupt other speakers and also do not make distracting sounds or movements.
Fill the attendance and feedback form as can be seen in the screen, and also the committee will type it in the Zoom chat box. And last but not least, please do respect, because the key to successful virtual conference is to remember that you are in a meeting. So give your full attention to the participant as you would if you were in the same room. Don't be distracted by other tasks and try not to eat or drink so that you can be prepared to be actively involved in the session.
It is an honor to me to inform you that among us here we have His Excellency, the Dean of Medical Faculty Universitas Islam Bandung, Indonesia, Profesor Dr. Nanan Sekarwana, Dr. SPAK MRS.
Her Excellency, the Conference Chair of BICMHS 2021, Mia Pusmiati, Dr. MPD, Ph.D.
His Excellency, the Head of Scientific Publication Unit, Universitas Islam Bandung Indonesia, Mr. Dadi Ahmadi, Assoc MECOM.
Her Excellency, Co-Conference Chair of BICMHS and the Chief of International Scientific Publication Section, Universitas Islam Bandung, Dr. Hendrati Dwi Mulyaningsi, SAMM.
Her Excellency, the Founder and Publication Director of Research Synergy Foundation, Mrs. Ani Wahyu Rahmawati, SPSI MSM.
Her Excellency, a speaker from Republic Polytechnic Singapore, Ms. Aliza Ramli.
His Excellency, a speaker from King Fahd University, Saudi Arabia, Professor Dr. Omar Hasan Kasule.
Her Excellency, a speaker from University of Copenhagen, Denmark, Associate Professor Eva Wulff, Ph.D. MSc.
His Excellency, a speaker from Gunma University of Health and Welfare, Japan, Professor Tatsuya Iso, MD, Ph.D.
His Excellency, a speaker from University of Science Malaysia, Associate Professor Dr. Badrul Hisham bin Yahaya, Ph.D.
Excellency, distinguished guests, delegates, ladies and gentlemen, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. First of all, let's thank to Allah the Almighty who has been giving us mercy and blessing until we can attend this international conference without any obstacles in this happy, pleasant time virtually. Secondly, salawat and salam to our Prophet Muhammad sallallahu alaihi wasallam, who has guided us from the darkness to the light. Once again, good morning, ladies and gentlemen. It has been a great honor for us on behalf of the Bandung International Conference on Medical and Health Sciences, or BICMHS 2021 Organizing Committee. My name is Ahmad Yusuf from Universitas Islam Bandung Indonesia, and my partner
Santi Ramawati from Research Synergy Foundation.
To be sitting here as a Master of Ceremony in this international conference, and welcome to each and every one of you for being here with us today.
So before we are going to the main program, let me read out the structure of our agenda or the conference program of the second day of the BICMHS 2021. So after this welcoming, we will have a Prodia presentations, and then continue to the welcome remark of the second day of the BICMHS, continue to the plenary speaker session, the online presentation sessions, and also last but not least, we have the closing ceremony, including the awarding session for the best presentation, the best paper, and also the session chair recognition. So please ensure that you are stay until the end of today's conference.
And without any further ado, let's open our agenda by reciting Basmalah. Bismillahirrahmanirrahim. Excellency, distinguished guests, delegates, and gentlemen, ladies and gentlemen, good morning and welcome to Bandung International Conference on Medical and Health Sciences, BICMHS 2021, which is coordinated by Medical Faculty of Universitas Islam Bandung Indonesia, or FKUnisba, Research Synergy Foundation, RSF, and UPT Publikasi Ilmiah Unisba, and also supported by Scholar Vein Research Synergy Institute, Reviewer Track, RSF Press, F1000 Research, and Taylor and Francis Group. The Bandung International Conference on Medical and Health Sciences, in conjunction with the 17th anniversary or Milad of the Medical Faculty of Universitas Islam Bandung Indonesia, the conference theme is Strengthen Medical Research from Basic to the Clinic Towards International Recognition.
This international conference takes place over 2 days, starting yesterday, Saturday 25th September, until today, Sunday 26 September 2021. So the BICMHS is an international conference devoted to the various spectrum of medical and health science for oral presentation in this academic virtual.
The general objective of the conference is to provide a platform and stimulate discussions on issues surrounding biochemistry, genetics, molecular biology, herbal medicine, public health, pharmacology, toxicology, academic health system, etc. And moreover, the scientific forum is also provides great networking and knowledge sharing environment.
The BICMHS is also supported by session chairs from several well-known and recognized higher education institutions in Indonesia, Philippines, and Thailand. We welcome all the strategic partners and members from the RSF Global Research Ecosystem Network to the BICMHS. We believe that this collaboration can bring mutual benefits to all stakeholders involved.
And we also welcome all participants, scholars, and researchers from various countries in the Bandung International Conference on Medical and Health Sciences, or BICMHS 2021, such as Indonesia, Philippines, Malaysia, Denmark, Saudi Arabia, India, Morocco, Mongolia, Thailand, and Singapore. And we believe that at the BICMHS 2021, you will gain a lot of insight through the knowledge obtained from the interdisciplinary fields of study, and this will lead to fruitful future collaborations and research opportunities.
And next, regarding the material of the conference, the virtual background, the conference program, and also the abstract book can be accessed at this link, as you can see in the screen, and later on the committee also will type it in the Zoom chat box. Meanwhile, for the conference paper proceeding also can be accessed at the Research Synergy Press website, and the link also will be sent to the Zoom chat box by the committee. And then next, for the question and answer, and also the e-certificate, the question and answer can only be asked via Zoom chat room, and the session chair or the moderators will manage as per the time available. So please kindly write your questions and the name of the speakers or the presenters that should answer it. And for the e-certificate will be provided to the registered email after the session for the participant who complete and actively involved in the paper or giving the feedback in sessions. So please do not forget to fill the attendance form and also the feedback forms that the committee sent through the chat box. And for the audio and also the video, so all participants are required to mute their audio, and the host has every right to mute any participant's audio and remove who are deemed disruptive without warning.
Well, ladies and gentlemen, moving on for the next agenda, we would like to invite the representative of Prodia, Dr. Anna Meliana Emkes, as the managing editor of publication of INABJ, or the Indonesian Biomedical Journal, to give the presentation and information. Please welcome Dr. Anna Meliana Emkes.
Okay, so first of all, I apologize that Dr. Anna cannot be here today. So I am Nurani Mustika, will be in charge of the presentation in her place. May I share my PowerPoint right now? Okay. Yep. So, all right. Thank you. Good morning, sir and madam. First of all, thank you for having me in this wonderful conference. Is my PowerPoint visible already?
Yes.
Okay. Now let me introduce myself. My name is Nurani Mustika from Prodia Clinical Laboratory. For the past few years, I am in charge of journal published by Prodia, namely Indonesian Biomedical Journal, assisting Dr. Anna Meliana, which has been published. Our journal has been published since 2009. Our journal is dedicated to publishing original research and review articles covering all aspects of biomedical sciences. All right. In today's opportunity, I will share some information regarding how to get your article published in Scopus Index journals. I won't talk about the paper content since I believe in this conference many expert speakers from various medical field had or will deliver about it. So I'm going to discuss some technical matters regarding article publication. I know that many of us realize merely publishing our article in scientific journal is not enough. We need to publish it in a journal that has a great impact in our field. Publishing in a high-impact journal indeed is not an easy thing to do. And there are several factors that determine our rate of success. So through this little sharing, I will give you some considerations that you should keep in mind during the process of journal selection to drafting the manuscript. I hope that this can help you during to increase your chances of getting your manuscript accepted in high-impact journals. So there are 3 main points that we have to pay attention to while deciding to publish our article. The first one is to pick an appropriate journal for our paper. It can be through SkyMago and PubMed or through Sinta indexing for local journals. After deciding which journals we will submit our article to, we have to pay attention to details that are required by the journal. For example, the aim or the scope of the journal, author guidelines, the reviewing time, and publication charge or fee. The last but not least, after making sure we understand and follow all the instructions, let's submit the journal. That is why we also need to know how the journal's website works. Okay, so the first and the most important part while publishing is to pick an appropriate journal for our article. Choosing the right journal for publication can be challenge itself. Therefore, firstly, we should set our goal. Is it a Q1 Scopus Index journal, or is it a Q4 Scopus Index journal, or even a nationally accredited journal is okay too. To find about what journal is indexed in Scopus, so we can use the indexation website, SkyMago Journal and Country Rank. And for medical journal, we might also want to look up on PubMed Central. And for nationally accredited journal, you may want to check on Sinta indexing for Indonesian journal. And there's actually many other indexation portals that you might want to consider, such as Clarivate or Thomson Reuters. Now, after setting our goal, so what criteria that should be our basis in picking a journal? The first one is the scope of the study. It is important to consider whether our work fits within the scientific scope of the journal and the topics covered, and whether the journal is broad in its scope or a specialist journal read mainly by a particular community, as this will affect the way that we have to write our paper. And not only the study field, but also consider whether the journal is a country-specific or is intended for global audience. For example, we will not want to submit our article, which the study was conducted in Indonesia, to a journal that specifically publishing article about the American population. The second one of what type of the article the journal publish. Make sure that the journal publish the type of article that you desire to write. You will not want to submit a case report study to a journal that only publish review articles and vice versa. The third one is the significance of our article in its field of specialty. Even though maybe our article is good fit in the selected journal, however, the journal may not accept our article if there are too many similarities with existing articles. May it be not the novel topic or idea, or the similar study samples or animal models. So to know about this, we should check some articles that already published by the journal. The next thing we can do is to find out about the accessibility of the journal, whether the journal is published online or print only. If it is published online, we can also have to find whether it is an open access or whether it should be subscribed before accessing the articles. Because some journals require a more expensive cost of publishing a paper so that the article can be accessed openly by everyone. The last one is journal rank and reputation. We can check the journal rank through the indexation website, which I will give some explanation later in a bit, while reputation is often based on impact factor, which is a measure of the average citations of paper published in a journal. And while talking about journal reputation, we should also have to check whether the journal is a predatory journal or not. To know whether a journal is a predatory journal or not, you may want to check it on https://predatoryjournal.com, and you will find a list of some possibly predatory journal. Okay, so here let's take a look on how to search for a journal through the SkyMago journal and country rank to find out which journal is indexed in Scopus and what rank they are currently in. If you already have a targeted journal and you only want to know its rank, you can type the journal name or its ISSN here to find detailed information about the journal. However, if you haven't decided which journal to submit your article to, you can type the study field here, or you may want to go to the journal ranking options. And if you type a study field in a search button, you will find the list of some related journals. For example, if you search for stem cells, they will show you some journal which has stem cell on their title. And if you click on the title, it will show you the details information about the journal. All you need is to scroll down and to find what rank the journal belongs in. And for example, this cell stem cell journal is ranked within Q1 of Scopus. And if you use to choose the journal ranking option, all you need is to type the study field, the subject areas, the publisher locations, the types of articles. In this case, we might have to choose journals and what years the rank is based on. So SkyMago will sort it out for you based on the Q ranking, so it will be easier for us to notice what rank these journals are in. Next one, I will share how to search journal in Sinta indexing for some nationally accredited journal from Indonesia. Maybe many of you already know about this portal, right? So it works in a similar way to SkyMago, but it is only for journal list that is published by Indonesian publisher or affiliations. So we search the study field or the journal name in the search button to find some indexed journals. For example, if we type medicine, Sinta will show us a list of affiliation or publisher that lists journal names which contain the word medicine on their title. And the next, you may click on the title to find out more detailed information about the targeted journal. And if you want to know about the journal rank based on the impact score, or the Sinta ranking, we may go to the option sources and then click journals to see it. The portal will show us a list of all the journals from the highest rank to the lowest. However, this option is not study field specific, so I suggest you to use the previous way to find the most suitable journal. Now, let's move on to the second point that we have to pay attention to before submitting our article. So after finally deciding the targeted journal, please pay attention to detailed information that is required by the journal. That might include the author guidelines, the reviewing process and time, also the publication charge. So I will give an example using the journal I am currently managed, the Indonesian Biomedical Journal. Usually in the homepage of every journal will be mentioned a brief explanation about the journal, like the scope of the journal, what affiliation the journal related with, who the publisher is, or even maybe some information regarding the copyright and the access of each article published by the journal. Usually, more information will be provided in the About the Journal menu. In the author guidelines, you will find a specific criteria regarding the writing format for the submitted manuscript. They also usually provide the information based on the type of articles. Like here, you can find that Indonesian Biomedical Journal only publish original research article and review article. So the requirements are also different for each type of articles. Usually a good journal will also provide a template of each type of article. This will help the possible author to format their paper according to the journal guidelines without have to go back and forth to the journal website. Next, you may also want to check the peer reviewing process of your targeted journal, like whether the process is single blind peer review or double blind peer review, how many reviewers will review your manuscript, and how many days or weeks should it take for a round of review. By knowing this, you can estimate the time you need to submit your paper. For example, if you are submitting your paper to Indonesian Biomedical Journal and you need your paper accepted by May because your upcoming doctoral promotion will be in May, so you might want to submit your article at least in January or February, knowing that it generally takes around 2 to 3 months for the reviewing process. Knowing about the publication cost or fee for publication is also important. So you will know beforehand whether it is in your budget or not. Every journal usually put the information related to the publication charge in their website as well. So you make sure you read this information too. Like I mentioned previously, that knowing the significance of our article in its field of specialty is important. That is why before submitting your paper, please do check the targeted journal published articles. You can go to their archive sections and check some of their latest publications to make sure that no article published is similar to what you are currently writing. Now we move to the last part, which is submit your article. After making sure you understand and follow all the instruction, let's go to the journal website to register yourself and upload your submission. Let's take an example on our Indonesian Biomedical Journal. Before uploading the paper, author need to register themselves as an author. Then fill all the required information. Once we are able to log in the user home, the option New Submission will appear. All the author needs to do is to upload all the required file, including the main manuscript, the supplemental files, if there's any, the cover letter, or the ethical clearance copy, and fill all the necessary data related to the manuscript, such as the title of the manuscript, the abstract, references, and author's information. So to conclude all the information I have mentioned before, please read the manuscript submission guidelines carefully. Make sure that your submission is clear, follows the instruction, and is as applicable as possible to the context of the journal. Also, make every effort to improve the quality of the manuscript before submission to avoid getting rejected in the initial check process. If you're sure all is settled, let's submit your article through the journal website or send by email. Thank you. I think this is all I can share with you this morning. If you have any question, please do not hesitate to contact me on nurani.mustika@prodia.co.id or secretariat@inabj.or. Now I give the control back to our Masters of Ceremony. Thank you.
Okay, thank you. Yeah. Okay, Mr. Yusuf. Okay.
Well, thank you, Dr. Ana Meliana Emkes, for the presentation and information. So back to Ms. Santi.
Okay, so once again, on behalf of the organizing committee, we would like to invite and also give the recognitions to our speakers representing the Prodia by Ms. Nurani Mustika as the publication managing editor of the Indonesian Biomedical Journals. So we would like to invite Dr. Mia Kusmiati, Ph.D., to give a token of appreciation to Ms. Nurani Mustika. Please welcome Dr. Mia.
Yeah, thank you, Mrs. Ani and Mr. Yusuf, for me to give the token appreciation. The first, I would like to thank you to Prodia INABJ from Indonesian Biomedical Journal. Yeah.
Right.
Okay. Thank you very much for your presentation. And may we ask your slide, maybe, Dr. Mustika? Okay.
I will send it.
Yeah. Okay. Thank you, Dr. Nurani, for your presentation.
Thank you.
Okay, once again, thank you so much, Dr. Nurani and also Dr. Mia. So moving on for the next agenda, we would like to invite Dr. Arif Budi Yulianti, DRA, MSc, as the conference coordinator of the BICMHS 2021 to give the welcome remark of the second day of the BICMHS. Before that, I would like to read her profiles. Dr. Arif Budi Yulianti is a senior lecturer in the Faculty of Medicine, Universitas Islam Bandung, or UNISBA. She is currently holding the position of Head of Medical Biology Department. Her research study is neurodegeneration, especially in alpha-synuclein protein, on sporadic Parkinson disease, malnutrition across generation, herbal medicine in drug isolations, and currently active as a reviewer at UNISBA and LL Dicti, as well as a member of UNISBA Health Research Ethics. The positions currently is in the Quality Assurance of the Faculty of Medicine UNISBA in the Evaluation and Monitoring Division. Please welcome Dr. Arif Budi.
Thank you. Thank you, Bu Santi. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Huwaladzi arsala rasulahu bil huda wa dinil haq yudhhiruhu ala dini kulli wa kafa billahi syahida. Asyhadu an la ilaha illallah wa asyhadu anna Muhammadar Rasulullah. The Honorable Chairman of UNISBA Foundation, Professor Dr. Kiai Haji Miftah Farid, along with a team of Foundation, the Honorable Rector of UNISBA, Professor Dr. Edi Setiadi SHMH, the first Vice Rector of the Academic Affairs, Profesor Insinyur Haris Nukman, MT, Ph.D, IPM, The second Vice Rector Finance and Human Resources, Professor Dr. Atih Rohaiti Daria. The third Vice Rector of Student Affairs, Dr. Andes Asep Ramdan Hidayat, MPD. The Honorable Dean of Faculty of Medicine UNISBA, Professor Dr. Nanan Sekarwana, Pediatrician, March, Vice Dean of Academic Fair, Vice Dean of Finance and Human Resources, and Vice Dean of Student and Cooperation Affairs. All of distinguished speakers, Ms. Aliza Ramli from Republic Polytechnic Singapore, Professor Omar Hasan Kasule from King Fahd University Saudi Arabia, and also Prof. Nanan Sekarwana as keynote speaker. Yesterday, we saw an interesting presentation for them. Professor Tatsuya from Gunma University, Japan, Associated Professor Badrul Hisham Yahaya from USM Malaysia, Associated from Professor Wulff Helge from Copenhagen, Denmark. Today, we will see an interesting present from them. We are pleased to welcome you to Bandung International Conference on Medical and Health Sciences 2021, and on the second day. The conference is being conducted by the Medical Faculty of UNISBA in collaboration with the Research Synergy Foundation in conjunction with 17th anniversary of our faculty. Or we can say Milad ke-17 Fakultas Kedokteran UNISBA. Today, I am as coordinator of this conference. Would you like to thank for all the support and good cooperation from all parties on this conference can run well. Thank you to Rector of UNISBA who has supported this event, the Dean Faculty of Medicine and all of his staff who have supported this event, Research Synergy Foundation for their good cooperation, UPT Publication of UNISBA for support this conference, all the committee of the conference who have worked very well, and all parties that we cannot mention one by one, who has support this conference. May Allah subhanahu wa ta'ala repay all the kindness that has been given. I hope this conference will become a bridge for international cooperation, especially in research development. In future, joint research among countries is our goal in our for advance to Faculty of Medicine UNISBA. We apologize of this conference has not been well organized. We also apologize for this word or action that are not pleasing. Thank you. Hopefully in the next year, we organize international conference in the other event. And we look forward to the participant to join us again. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Arif Budi Yulianti, for the welcoming remarks in the second day of the BICMHS. Next, we would like to invite Dr. Mia Kusmiati, Ph.D., to give a token of appreciations to Dr. Arif Budi Yulianti as the conference coordinator of the BICMHS 2021. Please welcome Dr. Mia.
Okay. Thank you, Bu Arif Budi Yulianti, Dr. MSc, for your opening speech or opening welcome remark to today's conference.
Thank you, Dr. Mia. Mia inside my.
Okay, thank you once again, Dr. Arif Budi Yulianti and also Dr. Mia Kusmiati. Okay, moving on to Pak Yusuf.
Well, ladies and gentlemen, distinguished participants and attendants of BICMHS 2021, let us move to the next session, the first plenary speaker session of the second day of BICMHS 2021. Is Professor Tatsuya Iso, MD Ph.D. from Gunma University of Health and Welfare, Japan, and Associate Professor Dr. Badrul Hisham bin Yahaya, Ph.D. from University Sains, Malaysia. Are you there, Professor Tatsuya? Have you joined with us?
Yes, yes.
Okay, and
I'm here.
Professor Badrul? Yeah. Okay. So this plenary speaker session will be moderated by Ibu Mira Sariputri, Dr. PhD, and Mas Rizky A.A. Syamsunarno, Dr. MKes PhD. And before the session begin, I would like to read the moderator profile. Mas Rizky is an associate professor in Department of Biomedical Sciences, Faculty of Medicine, Universitas Padjadjaran, Indonesia. He received his medical degree and Magister of Health from Faculty of Medicine, Universitas Padjadjaran in 2017 and 2010, respectively. He graduates his PhD in 2014 at Gunma University Graduate School of Medicine under supervision of Professor Masahiko Kurabayashi and Professor Tatsuya Iso, followed by postdoctoral until 2015 at the same laboratory. He has more than 70 national and international publications, including 50 publications in Scopus Indexed Journal. His study has been presented in various national and international conferences, such as American Heart Association scientific sessions in United States in 2013 and 2014, and European Society of Cardiology Congress in 2017 in Barcelona, Spain. Several awards had been acknowledged, including Young Investigator Award from Gunma University in 2012 and 2013, Young Investigator Award from International Society for Health Research, ESHR Japan section in 2014, and Science and Technology Award from Governor of West Java in 2017. And also his expertise is in metabolism and cardiovascular field. And currently he has position as head of biomedical magister study program, Faculty of Medicine, Universitas Padjadjaran, Bandung, Indonesia. And also Ibu Mira obtained her medical degree from the Faculty of Medicine, Universitas Padjadjaran in 2006. She finished her PhD in 2015 at Gunma University Graduate School of Medicine under the supervision of Professor Hiroshi Koyama, Professor Masahiko Kurabayashi, and also Professor Tatsuya Iso. She accepted Monbukaga Kuso or Max scholarship from Japan government for her PhD study. She is now an assistant professor in the Department of Biochemistry, Nutrition, and Biomolecular Faculty and Medicine, Universitas Islam Bandung, Indonesia. Her research interest is micronutrients and metabolism. She has published many national and international publications, and also she received several awards, such as a Young Investigator Award from Gunma University in 2012 and 2013, the top oral or poster oral presenter, or the third Bandung International Biomolecular Medicine Conference in 2014, and the National Annual Meeting Indonesian Private Medical Faculty Association, or as known as AFKSI in 2017, and lecturer with the most publications award from Faculty of Medicine, UNISBA. Now the moderator, Dr. Mira Sariputri, PhD, and Dr. Mas Rizky A.A. Syamsunarno, PhD. The time is yours to lead the plenary session.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, selamat pagi, konnichiwa. Thank you for the committee who invite us as the moderator in Bandung International Conference on Medical and Health Science virtual conference. We would like to thanks to the audience who still attend and follow this meeting session. In this morning, we have a plenary speaker session that will be led by me and Dr. Rizky.
Yeah, it is a great honor for us to participate in this great virtual conference. And I'm really happy to see my teacher that already attend in this meeting. I would like to saya mengucapkan salam hormat dulu kepada Pak Dekan, Prof. Nanan, kemudian ada Prof. Hendro, ada Bu Sadia, ada Pak Samsudin. And also I would like to say hello to my previous supervisor, Professor Iso. It is a good time for me to see you in health condition. And it is also our great honor to lead our two distinguished speakers from two different countries, Japan and Malaysia. For the first speaker, we would like to introduce Professor Tatsuya Iso from Gunma University of Health and Welfare, Japan. Hello, Sensei. Konnichiwa, Sensei. How are you?
I'm fine. Thank you very much for
Are you
invitation.
ready? Yeah, are you ready for
I'm ready.
You are ready for the presentation. Okay, good, Sensei.
Okay, and for the next speaker, we have Associate Professor Badrul Hisham Yahaya from University Sains Malaysia. Assalamualaikum, Dr. Badrul.
Waalaikumsalam.
Okay, how are you, Doctor?
Alhamdulillah, sehat. How are you?
Okay, everything's fine.
Yeah, great.
Good. Okay, you will give a lecture after Prof. Iso. Is it okay?
Yeah, thank you.
Okay, maybe now you can eat breakfast, maybe nasi lemak, maybe while waiting for your speech. Okay.
Yeah.
This session will be start with Professor Isa presentations and followed by Dr. Badrul presentations. We have 2 hour time slot, and I think both of speakers have enough time to present their excellent study. First, I would like to read brief CV of Professor Iso. Professor Iso graduated from Gunma University School of Medicine at 1990. He worked as physician and a cardiologist for 7 years. During the period, he also worked in a laboratory to examine cardiac pathophysiology and published an article in American Journal of Physiology in 1997. He next worked as a postdoctoral fellow in the University of Southern California for 4.5 years under the supervision of Professor Larry Cades. He learned a lot about molecular biology skills there. After came back to Japan at 2002, he started several projects in cardiovascular medicine and metabolism under the supervision of Professor Masahiko Kurabayashi. And for the last decade, he had published his excellent research in various high reputation journals, including metabolism, cardiovascular research, and scientific reports. Most of his studies is focused on biomedical science, particularly the role of fatty acid transporter in adaptive physiology and cardiovascular disease. And Professor Iso is also familiar with Indonesian. Now you see that he wear batik. Thank you, Sensei, for wearing batik. And Professor Iso also had a great opportunities to work with 3 talented graduate students from Universitas Pajajaran. They work and published many articles, including close one in 2013 and 2014 with me, and BBRC in 2015 with Dr. Mira. Metabolism in 2017, scientific report in 2018, cardiovascular report research in 2018, and again in scientific research report in 2020 with Dr. Yogi. And And also Dr. Iso, Professor Iso had visited Indonesia and very enjoyed to eat rendang, yeah, spicy beef. So now he finally obtained current position as a professor in Gunma University of Health and Welfare, Japan. So please, Professor Iso, the time and platforms are yours.
Can you share my slides?
Yes, it's clear, Sensei. Please.
Okay, great. Selamat pagi, and thank you very much for the invitation. I'm very happy to have opportunity to give a talk at the great meeting like BICMHS. Then the title of my talk is Local and Systemic Metabolism Regulated by Muscle Type Continuous Capillary. Lessons from mice with impaired transendothelial fatty acid transport. I'm from Gunma. This is Gunma Prefecture. Gunma is the center of Japan and the north of Tokyo. The Gunma University. Now I moved to the new school, new university. But my previous university worked with the Mira-san and the Risky-kun was the Gunma University. The Gunma University is surrounded by beautiful mountain and Tone River. And we worked hard and then published the many articles with them. Then this is the agenda of today's talk. Then the first is the systemic response to fasting. Then the second is the cardiac metabolism. In order to understand the systemic response to fasting, we need to understand systemic metabolism between multiple organs. In order to understand cardiac metabolism, we need to understand cellular metabolism. Then this figure, this slide shows the systemic response to fasting. Then right after meal, the glucose level is high, and this level is gradually decreased as fasting is prolonged. But the minimum level is maintained because of the glucogenesis by kidney. The glycogen is a storage form of the glucose in liver. This level is full after the meal. Then this level is decreased and become empty after 24 hours fasting. Then plasma insulin level is also highest after the meal. Then this level is gradually decreased. In contrast to glucose level, then fatty acid, non-esterified fatty acid, or the free fatty acid, then NEFA level is increased as fasting is prolonged. And in parallel, then plasma glucagon level is increased. Then ketone body, this is derived from the fatty acid. Then ketogenesis occurs in liver. These ketone body levels are minimum after the meal. This is gradually increased and highest after the prolonged fasting. Then to maintain energy homeostasis, lipolysis is enhanced during fasting. Then released NF-FA in circulation is used for skeletal muscle and heart. Then the glucose, the utilization is minimized. In liver, then fatty acid is used for the fuel for ketogenesis and material for fuel for gluconeogenesis and the material for ketogenesis. Then the energy homeostasis is maintained by this interaction between multiple organs. And this slide shows the characteristics of glucose and fatty acid. Then glucose is more consumed after feeding, and fatty acid is more consumed during fasting and starvation. Then big difference of these molecules are the storage form. Then the glycogen storage form of glucose. This amount of the glycogen is very small. Then in the case of the human with body weight 70 kilogram, then the glycogen amount of glycogen is only 90 gram. In contrast, then if the body fat percentage is 20%, then triglyceride the content is 14 kilogram. Then the total energy is only less than the 400 kilocalorie. But the fatty acid, TG calorie, total calorie is 13,000 kilocalorie. Then this is 340-fold higher than the glycogen. Then this is very good for the storage form. Then other difference are the glucose supply is required for brain and red blood cells. Then blood glucose levels are maintained by gluconeogenesis, mainly by liver. Then fatty acid is supplied from adipose tissue for a long period. Maybe the human can survive from the energy fatty acid for several weeks. Then fatty acid is also material for the ketone body synthesis. These are the major difference between glucose and fatty acid. Then this figure shows preferable energy substrate. Brain preferred glucose, heart prefers fatty acid, and white muscle prefers glucose, and red muscle, oxidative muscle, prefers the fatty acid. The liver is a kind of the chemical factory. Then this liver can convert this the energy substrate into the other substrates. Then this liver can redistribute these the materials to peripheral organs. The adipose tissue is a storage organ to store excess amount of fatty acid and glucose. Then red blood cell, this also need the energy, and these cells don't have the mitochondria. Then they use only the glucose. Then preference energy substrates are very different between the organs. Then this slide shows overview of lipid metabolism. Then the after feeding, fatty acid is esterified as a triglyceride. Then as the fasting is prolonged, the lipolysis is enhanced and the beta oxidation is accelerated. Then acetyl-CoA is utilized for the ATP synthesis through TCA cycle and the oxidative phosphorylation. Then when the starvation is prolonged, then ketone body synthesis, ketogenesis is enhanced in liver. Then this is the triglyceride. Then triglyceride is contained in the lipoprotein in blood because it's insoluble. Then TG comprised of glycerol backbone and 3 long-chain fatty acids. Then the free fatty acid, FFA, is also called non-steroid fatty acid, FFA or fatty acid. Then the important point is that this FFA is insoluble in water. Then albumin and FABP, this is the key molecules in this talk. The albumin and FABP are necessary to solubilize FFA because they are insoluble in blood and cytosol. Then this figure shows overview of fatty acid supply system, and then the point is capillary is critical component in this system. Then in endogenous pathway, the dietary fat is converted into the chylomicron, and the chylomicron pass through the capillary and the hydrolyzed by LPL, lipoprotein lipase. Then FFA is released from the chylomicron, and FFA is used by these parenchymal cells. In endogenous pathway, the VLDL pass through the capillary and hydrolyzed by LPL, and FFA is released, and the parenchymal cell uses FFA. Then the albumin-bound fatty acid from adipose tissue is also the very important substrate for the peripheral organs. Then this albumin-bound FFA pass through capillary and transport to parenchymal cells. Then the capillary is a very important component. This is a rate limiting the component for the fatty acid transport. Then this is the classification of the capillary endothelial cells. The capillary endothelial cells is roughly declassified into 3 groups: the continuous, fenestrated, and discontinuous groups. Then capillary endothelial cells are the functional interface between the plasma and tissue fluids to exchange substrates. Then we focus on this muscle type continuous capillary. This is a focus of this talk. Then the capillary endothelial house don't have any pore. Then these cells are connected to each other very tightly with this tight junction. Then this intercellular cleft is very narrow. Then this albumin and VLDL chylomicron, the carrier proteins for the fatty acid, cannot pass through this intercellular cleft. Then the question was, can fatty acid pass through muscle type continuous capillary by passive diffusion? This answer is no. Then how the fatty acid can pass through muscle type continuous capillary? That is the first and important question for us. Then the key molecules are the fatty acid binding proteins. This family form is comprised of 9 members of FABP1 to 9. Then we focused on FABP4 and FABP5. Then FABP5, FABP is a cytosolic protein. The FABP binds with high affinity to long-chain fatty acid in the cytoplasm and facilitate the transport of fatty acid to specific compartments, like the mitochondria and ER, and also the nucleus. Then FABP4 is abundant in adipocyte and is also called adipocyte FABP, AFABP. FABP5 is abundantly expressed in the skin. Then it's called epidermal FABP, EFABP. Then importantly, both FABP4 and 5 are also abundantly expressed in capillary endothelial cells. Then this is a brief overview of functions of FABP4 and 5. Then FABP4 and 5 attract many investigators because of very dramatic metabolic phenotype observed in FABP4/5 double knockout mice. Then body weight is less compared to the wild type, and the total adiposity is less, very less. And insulin tolerance test, glucose tolerance test are very good. Then this FABP4/5 double knockout mouse shows strong protection from diet-induced obesity, insulin resistance, as well as atherosclerosis. Then the people thought FABP4/5 are bad players for the development of the metabolic syndrome. But we address this issue, this metabolic finding, this issue by the different approach, the different direction. We found the FABP4 and 5 are abundantly expressed in capillary endothelial cells in the heart. This circle is many. You can see the many circles in the heart. These are the capillary endothelial cells. We also found the strong expression of the FABP4 in the mouse heart. The FABP5 expression is a little bit weaker than the FABP4, but when FABP4 is disrupted in mice, this FABP5 expression is enhanced. This means FABP4 and FABP5 have the redundant function. Then this is a summary of the FABP4 expression in capillary ECs. Then FABP4 and 5 are abundantly expressed in muscle type continuous capillary in heart and skeletal muscle. This is important. Then heart prefers fatty acid and the red skeletal muscle prefers fatty acid. Then the question is whether FABP4 and 5 are involved in fatty acid transport through continuous capillary layer in heart and skeletal muscle. Then this is first the working hypothesis. Then we hypothesized FABP4 and 5 are involved in transendothelial transport of fatty acids. Then the fatty acids are carried as albumin-bound form or in the lipoproteins. Then the capillary continuous type muscle type continuous capillary, this take up the fatty acid, and then the fatty acid is solubilized by the FABP4 and FABP5 and released into the interstitial space. And this interstitial space fatty acid are taken up by cardiomyocyte or the skeletal muscle. We termed the mechanism as transendothelial fatty acid transport. But if this is the FABP4 and the FABP4 and the FABP5 are absent in the endothelial cells, the fatty acid uptake should be disturbed at capillary endothelial level in heart of FABP4 and 5 double knockout mice. To prove this hypothesis, we used the Y-type mice and FABP4/5 double knockout mice. Then we also used the radio label tracers, iodine labeled BMI-PP fatty acid tracer, and fluorine labeled glucose tracer FDG. Then in the heart, this is BMI fatty acid uptake is this level in wild type level. Then this level was significantly reduced in only double knockout mice, but not single knockout mice. Interestingly, the glucose uptake level is dramatically increased in the double knockout heart. And this is the BMI-PP and FDG uptake in the red skeletal muscle and the white muscle skeletal muscle. The data are very informative. In red muscle, this is oxidative muscle. This muscle uses the fatty acid as a major energy substrate. And the BMI-PP uptake was significantly reduced in double knockout mice. And the glucose uptake, FDG uptake was significantly increased in red skeletal muscle. But this change was not observed in glycolytic white skeletal muscle. Then the glucose uptake, the significant glucose uptake was also observed by FDG PET imaging here. Then these findings suggest an increase in glucose uptake is observed only in the heart and red skeletal muscle. That consume fatty acid as a major energy substrate. This means this glucose, the uptake, increased glucose uptake is mediated by the compensatory mechanism. Then this is the first summary. The capillary endothelial FABP4 and 5 facilitate transendothelial fatty acid transport in the heart and the red skeletal muscle. Then fatty acid utilization is disturbed at capillary and cellular levels. Then glucose utilization is enhanced by the compensatory mechanism in FABP4/5 double knockout mice. The metabolic change was only observed in cardiomyocyte and red skeletal muscle. Then as Dr. Ritsky said, I visited Bandung once. Then I attended the third Bandung International Molecular Medical Conference 7 years ago. Then after this meeting, Dr. Ritsky took me around the Yogyakarta, and we visited Borobudur and Prambanan, and those were very beautiful, big, and those are the very good memory too. Then this is the next question. The fatty acid utilization by peripheral tissue is reduced in FABP4/5 double knockout mice. Then the question is, what happens when demand of fatty acid increased in catabolic states? Then in the case of the prolonged fasting, cold exposure, endurance exercise, the demand for fatty acid increased. Then what happened in the double knockout mice? We performed these experiments one by one. Then this is the result. Then this glucose level was reduced as the fasting is prolonged. Then the glucose is more reduced in the double knockout mice. The insulin level was also reduced, but there was no significant difference between groups. The fatty acid was increased, and the FFA is more increased in double knockout mice. And ketone body is also the more increased in double knockout mice. And this is the most impressive data, the liver gloss appearance. Then after 24 hours, the fatty liver was observed. This is the famous phenomenon, the fasting-induced hepatosteatosis. But the hepatosteatosis was enhanced only in double knockout mice. Then triglyceride content was enhanced 24-hour fasting in double knockout mice. Then this figure shows that mechanism. Then this upper part is adaptive response fasting in wild type mice. During fasting, the lipolysis is enhanced in adipose tissue, and this released fatty acid is utilized by heart and skeletal muscle, and the glucose usage is minimized. And the NFFA influx is increased in liver and used for gluconeogenesis and ketogenesis. But in double knockout mice, the adaptive response is impaired. The lipolysis is enhanced, but the fatty acid utilization is disturbed at capillary and cellular levels. Then this heart and skeletal muscle must use the glucose instead of the fatty acid. Then this causes the hypoglycemia. Then this release of NAFA is enhanced, and the heart and skeletal muscle cannot use fatty acid. Then this causes prominent fatty liver. And then this also causes hyperketonemia. The next question is energy constant. The next question is cold exposure response. The energy consumption is enhanced under cold environment, then by the 1.48-fold increase. TG in brown adipose tissue and glycogen in skeletal muscle are major energy substrates for thermogenesis. What if FABP4/5 double knockout mice are exposed to cold environment? Then this is the result. Then we used two conditions, in the fed state and in the fasted state. The cold challenge test was terminated after 4-hour housing at 4 degrees or when body temperature reached 25 degrees. Then skin temperature was measured by this type of the thermometer. In the fed state, body temperature were maintained in the both group, but in the fasted state, the body temperature was rapidly reduced. The body temperature dropped rapidly in double knockout mice. Then this is the mechanism of the impaired thermogenesis. Then in the fed state, the fuel for the thermogenesis, glycogen in skeletal muscle and triglyceride in the blood adipose tissue are full in both wild type and double knockout mice. But after the 20-hour fast, then this fuel level was reduced. But this fuel levels was more reduced in double knockout mice. And also the glycogen level was very low. The hypoglycemia was enhanced in double knockout mice after the fasting. Then this the fuel limitation causes impaired thermogenesis in double knockout mice. This is the next question, the endurance exercise. The energy consumption is enhanced during endurance exercise. Glucose, triglyceride, and NAFF in serum and glycogen in skeletal muscle are major energy substrate for exercise. Then what if FABP45 double knockout mice are subjected to endurance exercise? Then this is the result. Then this is a protocol. Then the treadmill running started at the 10 meter per minute, and this speed was increased every 5 minutes. Then running distance was calculated. In the fed state, this running distance was not different, significantly different, not significantly different. But the running distance was significantly reduced in the fasted state. Oh, movie doesn't work. This is very interesting movie, but this doesn't work. This is movie. The mice is running, but the double knockout mouse. This metal has electric stimuli and it hurts. Then they keep running usually, but this double knockout mouse stop running. Okay. Okay, then this figure shows the mechanism of the impaired endurance exercise. Then the concept is the same as the impaired thermogenesis. The fuel for the skeletal muscle, glycogen and triglyceride, and this was significantly reduced in the double knockout mice. Glycogen and triglyceride is markedly reduced, and glucose supply is markedly reduced by hypoglycemia. Then this reduced fatty acid uptake and severe hypoglycemia and the depletion of the glycogen and triglyceride causes reduced endurance. Then this is the second summary. The peripheral uptake of fatty acid via capillary endothelial FABP4 and 5 is crucial for systemic metabolism during fasting. Then the FABP45 double knockout mouse shows impaired systemic response to fasting, impaired thermogenesis, and air exhaustion after the prolonged fasting. This is the first conclusion. Then trans-endothelial fatty acid transport involving the capillary FABP4 and 5 is indispensable mechanism to maintain energy homeostasis by redistributing fatty acid stored in adipose tissue appropriately to peripheral tissues and minimizing glucose consumption in peripheral tissues for survival under life-threatening natural environments such as prolonged fasting and cold exposure. The FABP4/5 are indispensable protein to maintain energy homeostasis in this kind of situation. So I worked with Dr. Ritsky and Dr. Mirasari, and they worked very hard, and he received the Young Investigator Award from the International Society for Heart Research, Japanese section. Dr. Mirasari and the Ph.D. from Guma University Graduate School of Medicine. Then I'd like to move on to the next topic, the cardiac metabolism or the cellular metabolism. Then I think that many people learned a lot about biochemistry and metabolic pathways, many metabolic pathways, fatty acid oxidation, glycolysis, and TCA cycle in biochemistry classes. But the important point is this pathway is not only pathway. This pathway forms metabolic network. These are functionally linked to each other. By the way, this is a KEGMAP, the Kyoto Encyclopedia of Genes and Genome. This is very good, the web information, and freely available. Then please check it out later on. Then also the important point of the metabolism is catabolism and anabolism occur simultaneously. And the TCA cycle. TCA cycle is a cross point of many metabolic pathways, or the TCA cycle is metabolic hub. Then this figure shows the metabolism of the glucose and the fatty acid. The glucose metabolism is more complex. I write this white rectangle is energy substrate, and blue is storage, and orange is a catabolic pathway, green is anabolic pathway. Then glucose, the metabolism is very complex. This is usually this glucose is the source of the ATP synthesis. This glucose is synthesized through glycolysis and the TCA cycle and oxidative phosphorylation. But also the glucose is materials for the biomaterial, the source of the biomaterials like amino acid and fatty acid, and also the nucleic acid through the pentose phosphate pathway. This is a long chain fatty acid metabolism. Then this is the oxidized through the beta oxidation and produce ATP through the TCA cycle. And this is a material for ketogenesis. And the fatty acid is also stored as a triglyceride and also used for the materials for phospholipid synthesis. This is as you know, as you see, then the metabolism is very complex, not simple pathway. Then this figure shows the energy for heart and the energy starvation hypothesis. The measure the energy substrate for heart is fatty acid. Then the rest is the glucose and lactate. And these materials are the source of the ATP synthesis. Then the ATP is synthesized through TCA cycle and oxidative phosphorylation in mitochondria. Then ATP is transported to the cytosol, and this stored as phosphocreatine, reserve energy. And this phosphocreatine converted to ATP, and this ATP is used for contractile work. Then in normal heart, the fatty acid consumption is the largest, and glucose is a little bit used. And ATP and the phosphocreatine, the synthesis is enough. In compensated hypertrophy, the fatty acid consumption is reduced. Instead, glucose consumption is increased. And ATP synthesis is maintained, but phosphocreatine, the level is reduced. In failing heart, then fatty acid glucose utilization is reduced and ATP phosphocreatine synthesis is reduced. And this is called energy starvation hypothesis. Energy depletion causes the failing heart. Then this is the data, metabolic data of the FABP 45 double knockout mice, as shown in previous slide. Then fatty acid uptake was reduced and the glucose uptake was significantly, remarkably enhanced. But cardiac function is intact. Systolic blood pressure, heart rate, and fractional shortening, they don't show any significant difference. And cardiac function is maintained by a compensatory increase in glucose uptake. Then the aim of the next study is to determine whether metabolic reprogramming in double knockout heart, limited fatty acid uptake with enhanced glucose use at baseline is beneficial or detrimental under pressure overload by transverse aortic constriction, TAC. This is TAC. This causes increased afterload of the heart. Then we used wild type and double knockout mice, and they are subjected to the TAC. We observed 8 weeks for survival rate and cardiac function by cardiography. Then we found the cardiac function was reduced in double knockout mice in 1 week after TAC. Then we performed blood analysis, histological examination, and metabolome analysis 1 week after TAC. Then this is a result. The 3 mice out of 20 three mice died in double knockout group, but this was not significantly different. The fractional shortening by echocardiography was, this is important, they reduced at 1 week after TAC, but the wild type did not change this fractional shortening, LVDD and LVDS. Then in double knockout, this was significantly reduced, significantly increased, 1 week after TAC. Then we analyzed samples 1 week after TAC. Then the heart rate was increased in both wild type and double knockout mice. And stress marker ANP, ANP and BNP mRNA expression was markedly enhanced in both mice after TAC. The fibrosis was enhanced in both mice similarly. And the fibrosis markers expression was not different between wild type and double knockout mice. Then this is glucose and fatty acid uptake. Then glucose uptake was very high at the baseline, and this was enhanced by TAC. Then glucose level was reduced by TAC in double knockout mice, probably due to this glucose uptake enhancement. Then the next question, why is the cardiac contractile function reduced by pressure overload in FABP 45 double knockout mice? Then recently, to address this kind of the problem, we usually use omics. The omics is the method to comprehensively address issues of life phenomena. When you handle genome, this is called genomics. When you handle transcript, this is called transcriptomics. The protein, proteomics. Metabolites, metabolomics. Then we employed metabolomics to address this question. Then this is a brief overview of the metabolome analysis. Then we took this CEMS, capillary electrophoresis mass spectrometry. Then the first part, we extract the water-soluble metabolites and separate roughly by capillary electrophoresis. And then this the metabolite is analyzed by mass spectrometry. Then each metabolite is detected by their mass to charge ratio. There are many advantages in this system. The major advantages are high throughput, accurate, and high sensitivity. And also, we need only a small amount of samples for this analysis. There are many advantages in this system. Then this figure shows the difference between the metabolome and fluxome analysis. By metabolome analysis, we measure the steady state levels of the metabolites. But by this fluxome analysis, we can detect the flux of metabolites. We use tracer labeled with stable isotope for this fluxome analysis. In this case, we use the carbon-13 labeled glucose. The usual carbon, the molecular weight is 12, but this carbon-13 is 1 molecule heavier than this carbon-12. Then this glucose-13 is all carbon was labeled with the carbon-13. Then this is heavier, then we can trace this heavier energy metabolites. Then, for example, then this citrate with this carbon-13 glucose, this weight is 2 molecule weight is heavier than this usual citrate. Then we can detect we can trace this tracers carbon-13. Then we can detect the pathway this carbon-13 pass through. Then one more important point. Then this is complicated, but this is very important information. Then understanding the TCA cycle is a key point to understand this result. Then as I mentioned, the TCA cycle functions as a metabolic hub. The most metabolites have the pathway fluxing into the TCA cycle. Then TCA cycle is a cross point of many metabolic pathways, or the metabolic hub. Then not only glucose, fatty acid, lactate, ketone bodies, but also the aspartate, nucleotide, BCAA, the glutamate, the many substrates go into this TCA cycle. Then TCA cycle is linked to the both catabolic and anabolic pathways. Not only catabolic pathways, but also anabolic pathways. Then nearly all ATP is produced through the TCA cycle and subsequent oxidative phosphorylation. Then lastly, the pool size. The pool size is the total amount of the metabolites in the TCA cycle. The pool size in the TCA cycle is positively associated with cardiac function, at least ex vivo beating heart. This is very important information. Then we analyze the metabolites by metabolome assay and fluxome assay in double knockout heart and with and without TAC. Then and this is the result. Then pool size in the TCA cycle was reduced significantly at baseline in double knockout mice. And this reduction was enhanced by TAC. The pool size was more reduced in TAC. This and the glycolysis, glucose uptake is enhanced in TAC, but this is not sufficient. This means total energy supply is not sufficient for the energy demand in the TAC heart. We check each metabolite, citrate, alpha ketoglutarate, and malate. Citrate, alpha ketoglutarate, and malate. Then we observe the same similar tendency in this citrate. Then the levels of each metabolite are the smallest in double knockout TAC heart. And this is very interesting, the data. Then this is fluxome assay after injection of the carbon-13 glucose. Then the glucose influx into the TCA cycle was remarkably enhanced in double knockout mice at baseline. I mean, this glucose is glucose influx into TCA cycle. But the after TAC, this glucose influx was not enhanced. Then this means that this enhanced glucose taken up by the heart did not flux into TCA cycle. Then we check the creatine phosphate energy shadow. Then phosphocreatine reserve energy was significantly reduced, and ATP tends to be reduced, but this was not significantly different. The PCR, phosphocreatine reserve energy, was markedly reduced in DKO TAC heart. Then the pool size in the TCA cycle and the ATP synthesis were reduced despite a marked increase in glucose uptake, resulting in cardiac contractile dysfunction in double knockout mice here. This result is consistent with energy starvation hypothesis. We also observed wild type TAC and DKO TAC hearts showed comparable levels of cardiac hypertrophy and fibrosis. The next question is, how are biomaterials supplied for hypertrophic response and fibrosis? Can glucose be a source of biomaterials in DKO TAC hearts? Biomaterial means amino acid, nucleic acid, and fatty acids. Then we also addressed that issue by metabolome and fluxome analysis. Then this is a malate level and alpha ketoglutarate level, and importantly, the non-essential amino acids such as glutamate, glutamine, aspartate, and asparagine are derived from the TCA cycle, alpha ketoglutarate and the malate oxaloacetate. Then, despite a reduction in malate and alpha ketoglutarate, glutamine and aspartate and asparagine are increased in DKO TAC hearts. And very interestingly, the glucose flux labeled with carbon 13 was enhanced in double knockout TAC hearts. Then de novo amino acid synthesis is promoted in DKO TAC heart despite a reduction in metabolites in the TCA cycle. Then these results suggest glucose is preferentially utilized for biomass synthesis rather than ATP production during pressure overload induced cardiac hypertrophy. This glucose taken up by heart was not used for cardiac contraction. This was used for the biomass, the synthesis. Then the next question is can energy supplementation restore cardiac function in double knockout TAC heart? Then we focus on the medium chain fatty acid. Then medium chain fatty acid are the less number of carbon. This contains 7 to 12 numbers of carbons. The long chain fatty acid contains more than 13 carbons. The biochemically, this MCFA is different from the MCFA long chain fatty acid. MCFA is different from long chain fatty acid. Dietary MCFA are not converted in chylomicron, and dietary MCFA are released into portal vein, not lymph vessel. Then importantly, the MCFA is taken up by the cells independently, FABP and CD36. And the MCFA is also consumed independently mitochondrial protein CPT1 and CPT2. This is very different characteristics of the MCFA. Then we use the MCFA-rich diet, the 6% fat from the coconut oil. Then we use this MCFA-rich diet for the next experiments. Then this is a little bit complicated, but all animals are the double knockout TAC model. Then this model divided into two groups. standard chow group and the MCFA chow group, MCFA rich diet group. Then 7 days after this diet, these mice are subjected to TAC, and 17 days and the 14 days after this TAC, then cardiac function was estimated. Then this cardiac dysfunction was improved in MCFA rich diet group. Then in this period, this is starvation. After this is a fasting. After fasting, 12 after fasting, this advantage was disappeared. Then after the standard chow diet, this cardiac function was not improved. But when this MCFA rich diet was started again, then this cardiac function was improved. Then this finding suggests the cardiac dysfunction was restored by MCFA rich diet in DKO TAC heart. Then this is summary 3. Long-chain fatty acid are the central energy substrates for ATP synthesis to maintain cardiac contractile function even under increased workload. Glucose is preferentially utilized for anabolic reactions rather than ATP synthesis during the development of the cardiac hypertrophy. The ingestion of MCFA-rich diet restored cardiac contractile dysfunction, indicating energy insufficiency causes cardiac dysfunction. And the data are published in cardiovascular research and scientific reports. Then conclusion 2. The long-chain fatty acid are central energy substrate for sufficient ATP production to maintain cardiac contractile function even under increased workload. Increased glycolytic flux is directed to synthesize biomaterials for structural remodeling rather than ATP production during the development of pressure overload-induced hypertrophy. Then the Yogi Umbalawan worked very hard, and he received the Young Investigator Award for international student from Japanese Circulation Society annual meeting. And this is the last slide. And in order to see systemic response to fasting, we need to understand systemic metabolism between multiple organs. In order to understand cardiac metabolism, we need to understand cellular metabolism. Then the failure of energy homeostasis causes functional disturbance. Thank you very much for your attention. テレマカシバニャク、ご清聴ありがとうございました。
Thank you, Professor Iso, for your nice presentations. We have 2 credit points, I think, to SKS ini ya. It's very complicated and very comprehensive study. Allow me to explain a little bit in Indonesian language. Jadi, Prof dan sejawat sekalian, studi yang dilakukan oleh Prof Iso ini adalah untuk melihat peranan dari fatty acid binding protein 4 dan 5 di pembuluh kapiler dari jantung. Jadi ada 4 tipe pembuluh kapiler, salah satunya yang continuous capillary. Nah, FABP4 dan FABP5 ini dia terekspresi kuat di continuous capillary dari pembuluh kapiler yang tipe continuous. Delesi dari atau penghilangan fungsi dari FABP4 dan FABP5 ini ternyata menimbulkan respon. Yang pertama adalah menurunnya kemampuan adaptif dalam keadaan puasa, starvasi, kemudian exercise. Dan yang berikutnya adalah pada saat diberikan atau dibuat animal model cardiovascular disease, ternyata terjadi perubahan, terjadi shifting energi yang mengakibatkan terjadinya perubahan proses metabolisme di dalam sel jantung, di dalam jantung. Nah, ini yang mengakibatkan terjadinya dekompensasi jantung dan malah memperburuk keadaan. Nah, itu barangkali that's for the brief summary in Indonesian language. Saya say thank you, arigatou gozaimasu. Berikutnya untuk sesi, for the next sessions, we would like to invite Dr. Badrul, and this session will be led will be led by Dr. Mira. So, Dr. Mira, the time is yours.
Okay, thank you very much, Dr. Rizky. After we listen the great presentation from Prof. Iso about the role of FABP Form 5 in metabolic and cardiovascular, Iso Sensei, thank you very much for your presentation. And then looking for that Gunma picture, and I'm really surprised you still keep our photos. Honestly, make us both more miss you. Okay. And then we will move forward to the next speaker, Associate Professor Badrul Hisham Yahaya. But before that, I will remind to the audience, if you have any question, please raise your hand in the discussion session after Dr. Badrul's speech finishes, or you can write in the chat box. You can write it in Indonesian and English, and we can translate it for you. Okay, so we continue. I will read Dr. Badrul curriculum vitae. First, please. Associate Professor Dr. Badrul Hisham bin Yahaya, Ph.D., currently is the Director of Animal Research and Service Center of University Sains Malaysia, Penang. Dr. Badrul obtained his BSc degree in the genetics from the University Kebangsaan Malaysia, 2002, MSc in Human Genetics from the University Sains Malaysia, 2006. Dr. Badrul pursued his PhD degree at the Roslin Institute and Royal School of Veterinary Studies, the University of Edinburgh, Scotland. In professional bodies, Dr. Badrul is an active member of various national and international societies. Dr. Badrul received numerous invitations from local and international bodies to share his current finding in stem cell and regenerative medicine in various global scientific meetings, and is involved actively in the Tissue Engineering and Regenerative Medicine Society of Malaysia and Malaysian Society for Stem Cell Research and Therapy. He is currently a visiting professor at Xianyang Medical University, Henan Province, China, and has strong collaborations with the member of academic from Universitas Unpad Bandung, Indonesia. Next. Dr. Badrul is an editor-in-chief of the Journal of Biomedical and Clinical Science, published by MD-USM, editor of for Stem Cell Biology and Regenerative Medicine, Springer, Nature, and series editor for Tissue Engineering Part A. Dr. Badrul is also an editorial board member for Biomedical Research and Therapy Journal, Vietnam, MKB, the Global Medical and Health Communication Journal, Bandung, and Stem Cell in Clinical Application, Springer, Nature. With interest in stem cell and regenerative medicine, he received research grants from various funding bodies such as the University Sains Malaysia, the Ministry of Science, Technology and Innovation Malaysia, the National Institute of Health, Ministry of Health Malaysia, the Ministry of Higher Education Malaysia, the Fundamental Research Grants, and the Prototype Research Grant Scheme. The international grant from Niposit Grant Foundation, and also funding from the Henan Province, China, to work on various aspects of pulmonary research. A number of master and PhD students have graduated and actively working under his supervision in various aspects of research related to stem cell and regenerative medicine. So Dr. Badrul has many national and international publications, including more than 60 Scopus Index publications. His research most in the cancer stem cell for lung disease field, and his team also developed some methods for stem cell therapy in lung disease. Okay. Without further ado, please, Dr. Badrul.
Assalamualaikum warahmatullahi wabarakatuh. Thank you very much, Ibu Mira. So, Bapak Rizky, I'm very pleased to see you both here, and Prof. Iso for previous speakers, and thank you very much for organizing committee for inviting me to give a talk in this session. And I hope there is something that I can share in order to get benefit from all of us. So today I will talk about the next generation cell therapy for lung disease, and the future is here. So I would like to welcome to all of you to come to ESM for initiate collaborations and strengthen our collaborations in Malaysia and Indonesia in the field of regenerative medicine, genetics, and stem cell. Before that, I would like to bring some of you in the form of issues related to regenerative medicines. And as you all know that regenerative medicine composed of tissue engineering, biomaterials, stem cell therapy. And when we call about the stem cell, there are lots of potentials, ability of the stem cell to self-renew and regenerate into any type of cell that required by the body for tissue regenerations and repair of the tissues and also the organs. When we talk about the impacts of stem cell or cell therapy into the economics, it's already been published in 2017. It says like 1.3 billion in 2017 to 44 billion in 2024 has been projected, that resulted in staggering 65% of the compound annual growth related to cell therapy, which is involved gene therapy, DNA, RNA therapeutic, and also cell therapy. When you're looking at the global reports by World Economic Forum, that you can see like 959 regenerative medicine companies worldwide that's sponsoring 1,052 active clinical trials that involve in North America, Europe, Israel, and also Asia. And in aggregate, 7.4 billion has been invested in regenerative medicines companies in 2019. So by the economic impacts, we can see that huge potential of regenerative medicine and also cell therapy in order to for economic growth and also for well-being. And when look at the impacts of the Asia aspects, the impacts of the regenerative medicines in Asia Pacific regions, in the early success, we can see like in China, for example, they have produced the first gene therapy product that was approved in 2003, followed by Oncorine for the second gene therapy products for head and neck. And also followed by South Korea, there are 3 products have been approved by the governments and also the FDA, in which to be used for development of different types of the treatment for myocardial infarction treatment and degenerative arthritis, as well as the joint tissues. And the next one is in New Zealand for, they have developed Osiris Therapeutics in 2012 to treat graft-versus-host disease. And when you look at the Japan, for example, in early years, they have developed JACE, or Japan autologous cultured epidermis to treat serious burn treatment, and also JACC, or Japan autologous cultured cartilage for osteochondritis and knee joints. And also they have developed in TEM-cell and also heart shape were approved for the following years. So when looking at the impacts of the economy, where the Korea and Thailand play an important role, in which 1,195 total regenerative medicines and advanced therapy has been developed, and throughout the Asia Pacific regions that involve Europe and Asia Pacific 361, the investment was really impactful in terms of the increments of the economic growth related to regenerative medicines. And especially in this country, they have 1,300 clinical trials. There are 3 phases, phase 3 clinical trial, in which involve for gene therapy, cell-based immuno-oncology, cell therapy, and tissue engineering. If you look at by disease in terms of how this economic growth impact on the disease, it will more related to tumor-related clinical trial, central nervous system, infectious disease, and rare genetic disorders. And the most important aspect in the economic growth and the impacts of regenerative medicine is the roles of the governments, in which the Korean and Taiwan governments has introduced the Advanced Regenerative Bio Act in 2020, in which the government aims to complete institutional framework of the safety management system for clinical trial of advanced regenerative medicines, as well as biopharmaceutical as professional review and management system for long-term evaluations. So when you look at the lung disease that I'm trying to talk to you today, the lung disease is very much important aspect that really burdened of the country, in which, if you can see the reports on the global impacts of respiratory diseases, second editions, the big five that related to respiratory disease, in which COPD, asthma, acute lower respiratory tract infections, TB, and lung cancer, which are the most common causes of severe illnesses and death worldwide. And looking at these different countries, how much this country affected by the respiratory disease, and it was really increased in terms of mortality and its associated factors, especially in the developing countries. The most aspect that related to respiratory disease is smoking and vaping, and very much associated with lung disease. And for example, in Malaysia, among cigarettes among the students, you can see like e-cigarettes uses around 20.5%, and 40% for dual smokers, either cigarettes and also the e-cigarettes. So these are also related to the students, and of course, because some of the issues when we are encouraging students and adults population to stop smoking, there is some alternative way of using e-cigarettes in the form of to quit smoking. And the Health Ministry of Malaysia stated by 2025, it will cost the government 7.5 million ringgit Malaysia to treat smoking-related disease. And WHO has reported that estimated more than 20,000 male adult Malaysians die every year from due to smoking habits. But when looking at the health status and the economic impact, you can see that the global market for e-cigarette, for example, was worth 15.7 billion in 2018, and with a projection of to double for 40 billion in 2023. So this is a very important roles of the government, how to see whether the cigarettes and e-cigarette itself can give a bad impact to the health of the populations, but at the same time, it's really a good market strategy in which the government will have more money by having, promoting e-cigarettes and etc. So the global cigarette market worth 888 billion in 2018, and also is projected to reach value 1.1 billion by year 2024. So this is how the critical aspects of the government and also other health sectors in order to balance between health impacts and also versus economic impact related to respiratory disease. There is also reports, there are some mechanistic links between COPD and lung cancer because there are common signatures between COPD, and also some signatures might also contribute to the development of the lung, especially in immune cell profile in COPD and lung cancer. They are very common, and that is very highly likely that the study has been reported for those who have developed COPD, that are likely to develop a cancer. When talking about the lung cancer, there are a huge amount of challenges that the government and also various bodies to face, in which related to genetic faults of the patients, in which the patients do not respond to the injury to the treatment, and genomic landscape of the tumor in different types of the cancer, in the different stages of the cancer. There are some involvements of different genomic landscape. And of course, the most aspect that very challenging is identifying of biomarkers for early detections of cancer. Normally, patients with cancer, they come to the hospital at the late stage, and there is no way that the cancer can be stopped and also can be eradicated because it's already spread all over the bodies. And of course, when talking about lung cancer as well, they are heterogeneous cell population in which are very difficult for us to treat for any specific cell type that are responsible for the development of lung cancer. And what what our group are really interested in is that the roles of cancer stem cell in drug resistant, in which there are also some contribution to genetic faults, where the patients are not respond very well to the treatments towards the end. There are also some issues related to relapse and also recurrent in certain type of disease. And now we have all over the world, we have a COVID pandemic and the burdens of respiratory disease. As of 25th of September 2021, has been reported that 231,000 231 million coronavirus cases, and death for 4.7 million deaths has been reported. But at the same time, we have 208 million recovered from the disease. As Malaysian alone, we have reported yesterday upon 25,000 Malaysians died because of COVID-19. And but when we're looking at the pathogenesis of the COVID itself, is a cause. We all know that the cause of the SARS-CoV-2 virus, and in which it was first identified in Wuhan, China, in 2019, and because of that, it's called COVID-19 or COVID-2019. And at least 1/3 of infected individuals do not develop noticeable symptoms. It become more difficult for us to separate between people that carry the virus and also the people that already have the symptoms. So there are different stages of the virus infections and pathogenesis involved. And towards the end, the damage of the lungs and can be controlled. And normally people will die because of the failures of the lungs and lots of comorbidities that related to the disease. So we all also know that AC2 or angiotensin converting enzyme 2, the most abundant in type 2 alveolar cells in the lung. That is because of the COVID-19 are really interested to infect the alveolar cells and also towards the end collapse, the lungs collapse. And now because of COVID-19, we have new norm due to COVID-19, and we have to use a face mask that on every day and every time when we socialize ourselves. And we need to also practice a physical distancing, and we also required to have a frequent hand washing. And of course, because of COVID-19 as well, I cannot go to Bandung, I cannot meet Dr. Rizky and Ibu Mira, and because everything needs to do online. And there is no vacations at this time point because we have been locked at our home almost 2 years now. So when talking about the possibility of use of stem cell for cell-based therapy for COVID-19, there are also very interesting papers that published in 2020, cell-based therapy to reduce mortality from COVID-19 as a systematic review. And there are studies has been conducted and has been evaluated, and only 9 studies that fulfill the requirement by the papers and has been evaluated. It was a very interesting study, and it shows that MSC therapy is a very useful for patients with ARDC, acute lung injury, with focused analysis on safety, efficacy, and related immunogenic pulmonary disease. Among 200 patients with acute lung injury were treated with intravenously or intratracheally of MSC or placebo as a control, and 99 patients died, in which 30 were active treatment group, and none of the 30 deaths were found to be related to MSC related treatments. And MSC therapy on important immunology and inflammatory process, and it also can enhance anti-inflammatory response of the lungs, as well as also has capability as antiviral activity of MSC that can stop the spreading of the virus into the lungs. And various studies have shown potential benefits that minimum risk and preservative mechanisms of MSC therapy for acute lung injury, especially for COVID-19 infected individuals. And when looking at the roles of MSC in reducing anti-inflammatory responses, we can see there are 2 differences in terms of healthy alveolus and also injured alveolus in which infected with the COVID-19. So AC2, for example, is highly expressed in various organs, but in this case, for COVID-19 infections, the most important aspect is on alveolar regions of the lung. And you can see that MSC, by having the COVID-19 infections and also infusions of MSC by intravenous infusions, and they can also have a possibility to treat COVID-19 because of the MSC itself and exerts immunomodulatory activity by secreting any inflammatory molecules that can stop the inflammatory responses, that also secret growth factors related to the healings of the airways, and towards the end the airways will become a normal and patient will have healthy lungs and also can recover from COVID-19 infections. But what NIH, the FDA says about cell-based therapy on COVID-19, based on the COVID-19 treatment NIH page, it says that COVID-19 treatment guidelines panels have recommended that against the use of mesenchymal stem cell to treat for COVID-19. So I think my personal view is that the NIH was trying to convince the populations, the people or the patients, that there is some potentials of COVID-19 treatment with MSC, but the fact that they're hoping there is no some kind of misuse of COVID-19 treatment using MSC because of lacking in terms of clinical data. So none of these products, especially products related to FDA approved of the mesenchymal stem cell by the Americans products, none of these products are approved for the treatments of COVID-19 of any other viral diseases. So the one that I've shown you from the systematic review, it shows that among 1,000 plus studies, only 9 has fulfilled the criteria by the authors. And 9 of those, although there's some shows that some potentials of MSC treatments for COVID-19, but there's some lack of evidence whether the MSC itself can play an important role in reducing the symptoms of COVID-19 infections. When talking about the stem cell of the lung, there are various types of progenitor cells residing in different regions of the lung. And because of their huge amount of cells that are responsible that residing in the trachea and also the proximal airways of and also the distal parts of the lung, which of these stem cells can be used as a target for cell therapy. Because now we are talking about the MSC, is meaning that the external source of the stem cell can be transplanted to the lung in order to help to regenerate of the lung. But can we possibly have a possibility to induce the localized stem cells of the lung that can regenerate of the airways? So because of their heterogeneous populations of the cells in the lung, they are difficult for the targeting of the specific cell type that require to regenerate of the airways. When talking about the stem cell, as you all know, that there are 2 common characteristics that play important role in stem cell, in which is self-renewal capability, in which the stem cell can maintain its pool. So at the same time, when some kind of a trigger happens into the lungs or into the tissues and organ, and this particular stem cell pool that have self-renewal, they are able to give rise to other type of cell in which developments in the differentiation stages of the stem cell. They can give rise and also regenerate of the injured tissues. Because of stem cell, mesenchymal stem cell is a unique stem cell. They play an important role in regeneration of the tissue. It must be very important characteristic that we need to understand about the mesenchymal stem cell. They are immunomodulatory properties of MSC. They have, release a lot of growth factors that involve regenerations, and also they are play an important role as antivirals or anti-inflammatory. And there are another studies conducted using adipose-derived mesenchymal stem cell that shows that by having patient injected with various stages of adipose-derived mesenchymal stem cell, they will increase the survival rates of the patient. Only few of the studies, like in this case, there are the patients that due to embolism, for example, and other causes of the injury, but mostly around 70% of the patient they are survived. And we're looking at the X-ray, chest X-ray of the patients with COVID-19. You can see that differences in terms of before and after treatment with adipose-derived mesenchymal stem cell. So another issue that related to stem cell treatment is that what is the best route of cell delivery? So in our case, there are some areas of systemic delivery or local delivery in which using intravenous or intratracheal, intraarterial delivery of the stem cell, and also there has some advantages and some limitation. And the common use for specific tissues and organ, for example, they can use a local delivery in which a topical for wounds and very transcardial for heart, for example, or direct injection to the brains and also muscle. So there are some options, advantages, and there are also some limitation that surrounded this route of delivery. So the next question, whether for respiratory disease, for example, is it possible for us to develop aerosol-based cell delivery to deliver the stem cell directly to the lung so that it can have an optimum delivery of the stem cell? At the same time, we can have the optimum regenerations of the damaged tissues. Our group have also developed aerosol-based cell therapy for treatments of lung diseases, and we have published these methods, and we have proved that using aerosol-based delivery using fibroblast cells, for example, to proof of concept, whether this kind of a technology can be used, and we have shown that using aerosolization itself, it doesn't give any impacts on airway epithelial cells as well as mesenchymal stem cell. And we can see that by using aerosolization techniques also, we can see the regenerations of the tissues following injury. For example, in this case, we have using aerosol-based epithelial cell delivery that we have shown that improve in terms of regenerations and the repairs of the airways. So the reductions in terms of inflammatory responses, fibrosis reduce, and as well as engraftment increase in terms of the engraftment of the airways, airway epithelial cells in the lungs compared to other parts of the organs. And also we have shown that when talking about the airway epithelial cells or mesenchymal stem cell as a source of stem cell to be transplanted into the patients, so whether the stem cell itself or airway epithelial itself play an important role in regeneration, but the various studies have shown that there is some secreted compound in the airway epithelial cells that play an important role in regenerations of the airways. This secreted compound is not only limited to mesenchymal stem cell or airway epithelial cell, it was actually released by any type of cells in our body. So the secreting compound, or what we call the secretomes or extracellular vesicle or microvesicle, are important ways of communication between cells and cell communication, in which they're also important for homing process of the stem cell or progenitor cell or cell that's surrounding of the injured airways. So the communication of the cells and tissues, in order for them to inhibit or not inhibit, they stimulate the migrations of the cells surrounding of the tissues, can come and do the regeneration process that involve the secreted compound released by that particular cells. So we have shown that in our study that the effects of secreting compound in airway regeneration and repair, and we have developed co-culture techniques using direct or indirect techniques, and we evaluate whether the cells can be separated using transwell, can also enhance regenerations and repair of the adjacents of the epithelium. So in this case, when you use a direct, we have published in scientific reports in 2017, when we use a direct in which the stem cell cultured directly with the damaged trachea, we can see like regenerations by the transplanted cells in which covered the injured trachea of the rabbits, and the regenerations and differentiation process into goblet cell or secretory cells. And we can see that by having direct contact between transplanted cell with the injured cell, it can back to the normal airways in this particular case. But when looking at the whether to evaluate whether indirect co-culture also play an important role, we separate between our trachea, damaged trachea with the stem cell on top of the trachea, and then we have a control only the airway epithelial cell without any stem cell or airway epithelial cells cultured together. So in this case, using these two methods, direct and indirect methods, we have proven that the secreting compounds released by airway epithelial cells play an important role to to regenerate and remodels of the airways. So by looking at this, also by having indirect co-culture, where in which you can see like here, the regenerated tissues was actually coming from the adjacence of the airway epithelium, in which they start to migrate and differentiate to become airway epithelial cells. So by having this experiment, it was very clearly shows that by having secreting compound released by the airway epithelial cells or any type of cell also can be used as a regenerative agent for regenerate and remodel of the airways. And this is the one that I just mentioned to you by using a direct co-culture techniques. So now the next question is that how MSC exerts its effect. As I mentioned earlier, you can also, the MSC can have cell-to-cell contact in which they will start communication between cell and enhance or induce migrations of the cell to come to the injured area. And also communication can also be done by paracrine factors in which the injured cell or the injured tissues will release cytokines such as VEGF, IL-10, and also other cytokines that are released by the cell. And of course, the extracellular vesicle released by other adjacents of the epithelial cells can have some effects, in regeneration of the tissues. So what we have looked at our previous studies, we have shown that the effects of mesenchymal stem cell secreting factors in regenerations and of airways. We can see like all these regeneration process happening using mesenchymal stem cell not alone, and also mesenchymal stem cell with the secretion factors. And when we do the proteomic analysis, we have found that most of the proteins released by the MSC or that contains in secreting factors involved in cell growth and migrations, immune regulation, proliferation and differentiation, and cell signaling. So by having all the proteomics analysis, it was very clearly shown that the secretum compounds released by the mesenchymal stem cell or any type of stem cell are important secreting compounds that play an important role in regenerations and repair of the tissues. So therefore, we hope that for the future, exosome can be a future cell-free therapy because it can also, as I mentioned earlier, that MSC release extracellular vesicle, they can call as an exosome or microvesicle that contains a lot of cytokine and growth factor that play an important components for tissue regeneration and repair. So these vesicles are involved in cell-to-cell communication, cell signaling, and altering cell or tissue metabolism, in which later on can influence tissue response to injury, that also can important role in infections and disease as well. When carefully study on extracellular vesicle as a novel communicators in lung disease, we can see like they have different types of exosome, microvesicles, and apoptotic bodies. So these are components that released by not only specifically by mesenchymal stem cell or any stem cell, but was actually released by any type of cell as a form of communication between cells. And if you look at the different types of lung disease, for example, lung cancer, there's some release of various types of extracellular vesicle that can be a biomarker or can be also as a target area for future targeted therapy. For example, in lung cancer, what you can see here is that you can use mesenchymal stem cell. There are lots of microRNA cargo, and you can, if you look at the COPD, for example, as a model system, there are also cell types that involve epithelial cells and also endothelial cells that have been affected by the COPD, and vesicle type is involved extracellular vesicle, microparticles, and as well as a different type of microRNA cargo. These are also can be used as a biomolecular marker for detections of COPD as well as any other types of respiratory disease. When looking at the extracellular vesicle as a form of the new regenerative agent, it is actually a small membrane vesicle of multivesicular bodies. The size ranges from 40 to 1,000 nanometer and can be sedimented by centrifugation at 100,000 G. So when looking at the potentials of extracellular vesicle, recently we have published in stem cell research and therapy, where we can use extracellular vesicle released by mesenchymal stem cell or human umbilical cord mesenchymal stem cell has shown to be a very good potential for cell-free therapy for animal model with COPD. And there are some characterizations of human mesenchymal stem cell, like having expression of CD63 and also using SEM to show the effects of the mesenchymal stem cell or extracellular vesicle. You can see here this treatment mesenchymal stem cell release EV and also mesenchymal stem cell is a very much associated as compared to self-healing as well as the injury and also the naive one. So meaning that having MSC release EVs or human umbilical cord MSC itself, they can release inflammatory response in the animal model system, as well as in peribronchial and perivascular inflammation, especially the impacts of the EV in remodelings of the airways. Extracellular vesicle for tissue repair and regenerations, when looking at the applications of the extracellular vesicle for future cell-free therapy, so you can see like it can be used for tissue regenerations. It can also be drug delivery techniques. You can also use extracellular vesicle for drug delivery, and also because of it, it's involved in various type of the organs. And it's mediate tissue repair by promoting cell proliferation, differentiation, and angiogenesis by inhibiting apoptosis and inflammatory response. Whatever potentials of MSC that you can see in various publication or also in one that we have published that I've shown you here, that potential also can be gathered from extracellular vesicle. Because as I mentioned earlier, mesenchymal stem cell is not a sole agent for regeneration, but because of mesenchymal stem cell release the extracellular vesicle like exosome or microvesicle, these was actually the main components of the effect mesenchymal stem cell and extracellular vesicle and remodeling of the airways. And as well as I also would like to highlight here, as I mentioned earlier, EVs and particles protein can serve as reliable biomarkers for cancer detection and determining cancer type. So because of lung cancer, if you can get the fluids by the airways or bronchial bulb, bronchial alveolar lavage, for example, that also can be obtained from the patient with COPD for lung cancer, for example. We can get and profile the bulb fluid. It can be used as a determinations of biomarkers from the patient. For prostate cancer, for example, probably because of the urine released by the individuals, that can be extracted as a biomarker from the urines of the patients or urines of the individuals. You can make a comparison. That also can be a biomarker for early detections of prostate cancer in the case of prostate cancer. And for breast cancer, there are some probably that chemicals or any form of fluid released by the breast also can be used as a biomarker for early detections of breast cancer or whatever cancers by using extracellular vesicle that released by these particular tissues and also organs. The challenges in cell therapy is that the source of the stem cell, whether we can use, we have shown that we have proven, Egi Kadia is our student. He was actually the graduate from Pajajaran University. And she's now in Australia doing a postdoc. She was my PhD student. We have shown that the progenital cell also can be used as a form of cell therapy. In our model system, it shows that by having airway epithelial cell transplanted to the animal, it can release the inflammatory response, and that also can use in regenerations. And we have also shown that mesenchymal stem cell, for example, from adipose and human umbilical cord stem cell, can also be used in tissue regeneration. In our collaboration with Xinjiang Medical University, China, we have shown that a menstrual blood stem cell, in China, because of they lack in terms of umbilical cord, because the birth rates in China is a very minimum as compared to the other parts of the world, because they need to find another source of stem cell, because of the woman release the menstrual blood every month, around 7 days per month, for example. Rather than wasting that blood, the Chinese scientists have shown that by collecting menstrual blood stem cell, by extracting the stem cell from the menstrual blood, also can play an important role as a source of stem cell for the treatment for regeneration. We have shown that menstrual blood-derived mesenchymal stem cell play an important role in acute lung injury, reduce inflammations, and as well as in the Parkinson's model system in collaboration with China. So because of the stemness capability, self-renewal and differentiation capability, whether the adequate number of stem cell, what would happen if we propagate the stem cell in vitro, what would happen to the long-term effects of the stem cell itself. The number of cells to be delivered to the specific locations of the organ might be another issue, and also the safety and effectiveness in which a lack of long-term studies, and some of the effects also, for example, whether the stem cell can be used to treat the cancer. We have shown that in our lab by having mesenchymal stem cell co-cultured with cancer cell or indirectly co-cultured with cancer stem cell. This mesenchymal stem cell itself enhance the cancer, the carcinogenesis of the lung cancer. So whether the question now, whether the mesenchymal stem cell or any type of stem cell can be used to treat any type of cancers. But they have shown also the variation between one lab to another in terms of the effectiveness or the efficacy of the stem cell treatments. And of course, when we wanted to translate the preclinical setting towards clinical application, we require the expensive lab setting and maintenance, for example, like GLP for preclinical testing and GMP lab for clinical application, in which we need to isolate and propagate our stem cell in the lab before we can transplant to the patients. And now, another aspect of our groups now, when we are trying to understand from the animal model system, the type of stem cell that we have studied in our animal model system, we have shown that we have proven that mesenchymal stem cell, AV epithelial cell can regenerate of the lung tissues or remodels of the lung tissues. And we also shown that in our study that secreting compound like extracellular vesicle or microvesicle or exosome play an important role as well. Have proven that in direct or direct culture, where these microvesicles or extracellular vesicle play important roles. But now when we're trying to understand from that aspects of the lung tissue, and we have shown that, I have also shown to the recent publication in which we have proven that mesenchymal stem cell, human umbilical cord mesenchymal stem cell, derived extracellular vesicle can regenerate of the airways following cigarette smoke or COPD model system. But the fact that what we don't know how much the cigarette smoke itself trigger or impact on the lung of the stem cell of the lungs, where when people, because what we have done that when we give smoking to the animals, we start seeing that the regeneration or inflammatory responses of the lungs. But in normal situation, when inflammatory response, that also the animals, the lung itself can go through the self-healing system, in which the localized stem cell can play an important role, release the mesenchymal stem cell, or release whatever compound that can reduce inflammatory response. Meaning that without mesenchymal stem cell treatment to the lung also can regenerate themselves. But what would happen if we have a prolonged exposure of these secrets to the lung? And what happened to the stem cell? Why the stem cell cannot regenerate and self-renew so that it can heal from the COPD or heal from the cancer stem cell? So this is why we wanted to now looking at different angles on how the stem cells of the lung play an important role when there is some exaggerations or trigger by the smoking cigarettes itself. So we have developed what we call as a lung organon, as a model to study lung stem cell. So in which we isolate the stem cell or the tissues from the lung, we culture, and then we study in terms of morphogenesis and the histology of the lungs. And our interest more towards to see the effect, how much the cigarette smoke inhalations was actually effects on the stem cell of the lung. Our hypothesis is that when we give a cigarette smoke to the animals, the cigarette smoke itself can defect the lung stem cell of the airways. And towards the end, because the stem cell defects and they cannot regenerate of the lung, that is why the COPD developed in these particular animals or in a human being. By looking at this aspect, we are now trying to 12 weeks inhalations as a model that we have developed now. So to see whether that was the first hypothesis, meaning that cigarette smoke itself will defect the lung stem cell. So whether by defective lung stem cell, whether it can repopulate themselves and whether it can develop regenerations, remodelers of the lungs, or they can develop a cancellation. Why organoid? Because organoid is a miniaturized and simplified versions of the organ.
I'm sorry, Badrul,
and self-organized three-dimensional tissue cultures, and provide unique opportunities for the study of human disease. And it has been used to study infectious disease, genetic disorders, cancers through genetic engineering. And what we have done is that we studied on generations of airway organoid. We harvest the trachea, lung tissue from mice, and we mechanically and chemically dissociate tissue into single cell, and we transfer and we culture to become the organoid. And later we use organoid in matrigel in order to see the morphology of the airways. So these are the morphology of the trachea and lung organoid that we cultured in our labs. So you can see the early developments of the lungs. It was a very like a ball shape, and later it will differentiate to become like lumen regions, meaning that these have become the whole the space already developed in which there is the structures of the airway surrounding of the lumens. And later, at around D10 to D14, it now become like different shapes of the like a flower shape of the lungs, which is already developed in vitro. And you can see like establishment of our organoid model D3, D5 in all the stages of the development of the organoid at D14, and it will already develop into a very unique structure of the airways, different lobes and different regions of the airways. So this is a very interesting aspect to use the lung organoid as our model system, and you can see it's a very beautiful structure of the lumens and already been differentiated into the structure of the airways and the structures of the lobes and the segmental parts of the airways already developed in our model system. So when we culture and we do the histology analysis, you can see very clearly the pseudostratified of the airways, the different parts, different stages of the lungs. And there is also the very interesting, if you can see on the right side, a very clear lumens and basal cell, also differentiated airways. Then we can confirm that with our staining. You can see like, a very huge aspect of the basal cell, which is stained in trachea, for example. The most common stem cell that have in regenerations of the airways is a basal cell. So all the green color is actually stained with a basal cell. And you can see here the pseudostratified of the airway, then and already have differentiated into like a ciliated cell, like in which the positive for alpha tubulin, in which the hairy cells or ciliated cells already form at the D14s or D10 to D14s of the trachea in the lumen in the culture. So now I would like to bring as a final part, it's a good model system using lung organoid, and it's promised for model for organ development, drug discovery, and disease modeling. And before I end my presentation today, I would like to bring some very interesting aspects of regenerative medicine already been mentioned in the Al-Quran. I'm not really good in translating of the Quran, but I would like to highlight here Surah An-Nisa, ayat 56, for example. You can see very clearly that Allah mentioned when we are in the hell, for example, our skin will be burned, and after that we shall change them from fresh tissue, meaning that when our skin already been burned completely because of Allah trying to give you the painful itself by putting us in the hellfire, but later the Allah clearly mentioned the regeneration process of the tissue will be happen again in the hellfire, in which a continuous regenerations happen is infinity, in which the next question that I would like to highlight here, does heat regenerate the skin tissue? Meaning that if we, the skin is a burn, and also with whether the hot conditions, is also the hot condition, the heat itself also can regenerate the tissue so that it can has a continuous kind of regeneration aspect. So are there any heating-based technique or laser can be used to regenerate tissue following burn injury? So can heat energy convert progenitor cells into pluripotent stem cells? As I mentioned earlier, pluripotent stem cells play an important role in the regeneration conversion. It's already been proven by Yamanaka, in which by converting transcription factors, it can convert progenitor cell into become pluripotent stem cell like cells, in which also can heat also can play important role to regenerate or reprogram transcription factors that later control pluripotent activity of progenitor cell, in which in this case to regenerate the skin tissue. So with this, I would like to end my presentation for today. I hope that the last slide will open up a new areas of study by having the heating process for regenerations of the tissue, or whether the heating itself can transform a pluripotent stem cell, a progenitor cell to become pluripotent stem cell for regenerations of the skin tissue. With that note, thank you very much for your attention. I would like to highlight the appreciation to my groups and University of Science Malaysia, as well as all the organizing committees and my friends and my family in Unpad for inviting me, and also the Ibu Mira University, that organizing this conference, that inviting me to give a talk today. Thank you very much. Assalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you for your presentation, Dr. Badrul. For the audience, please give a virtual applause to Dr. Badrul. Okay, so now we will open a discussion session. We have one participant here who want to raise hand. Professor Nuzirwan Achang, are you here? Do you want to ask the question? Okay. Professor Achang? Okay, if not, I will...
Thank you very much.
Thank you, okay, please.
Thank you very much, moderator.
Yes.
And thanks for Associate Prof. Badrul for your very interesting topic. But my quick comment is, as right now, the study medicine about the stem cell was rapidly growing, especially for oncology, for cardiology, but the last for neurologic, they use for cosmetic. Now, because the function of stem cell is very like immunomodulator, proliferation, regeneration, and sometimes for induction of cell proliferation. My question is, in Malaysia, do the stem cell production controlled by kerajaan, by government? Because so many using of stem cell right now. But sometimes I am afraid it's misuse for the patient. I think it's all my question is simple question.
Yeah, thank you, Bapak.
Because in Indonesia, I worried. Sorry.
Yeah, thank you, Bapak, for the questions. Yes, in Malaysia, there is a Minister of Health, also NPRA, National Pharmaceutical Agency, is controlling the use of stem cell for the treatment with patients. In 2015, for example, the government, Minister of Health, has already instructed all the industry key players. For example, we have 13 to 15 tissue bank industry that collecting umbilical cord and also do banking, stem cell banking, and they make money of that. So some of the private hospital, for example, they try to use this stem cell for the treatment of the patients. But in 2015, governments already instructed you need to come back to the preclinical aspects of the study, in which you need to get the GLP lab facilities, to follow all OECD GLP guidelines, and whether there are some effects, preclinical effects of the stem cell. And this company needs to come back to the lab-based analysis. And by 2021, this year, for example, for those companies that already been given 5 years, to evaluate, to come back to the government with all the preclinical data, then they will be evaluated by the NPRA. That NPRA is another agency under Ministry of Health that control the use of stem cell or any stem cell product for the therapy, where we know that lots of illegal kind of use, but unfortunately, some since 2015 it's already been highly regulated by the government.
The second question, in our conference we have a motto, from basic to clinical. My question is, do you research about the using of stem cell for treatment resistant tuberculosis? Thank you.
Okay, so far we don't have any experiment that running on the tuberculosis, because of in IPPT, for example, in our institution, there are some studies on cardiac stem cell, eye stem cell, and also lung stem cell, but none so far that working on tuberculosis.
Okay, thank you.
Thank you,
Moderator.
Okay, thank you very much, Professor Nurjirwan Acem. Please, Dr. Rizky, continue.
Thank you, Dr. Mira. We have a lot of questions addressed for both of speakers. I would like to choose one question from Dr. Yuniarti to Professor Iso. Why ingestion of MCFA-rich diet is more effective to restore cardiac dysfunction than protein-rich diet? Or may I add the questions? Is it possible? Is it a prospective to give a high-fat diet to the patient with a cardiac dysfunction?
No. Then the important point is this medium chain fatty acid is only effective for the heart with reduced fatty acid uptake. Then only in the double knockout or CD36 knockout mice. Then we also tried this treatment for the wild type mice heart. Then that didn't work. Then the medium chain fatty acid can be used only for the heart with reduced fatty acid uptake. Then if the fatty acid uptake is enough, then medium chain fatty acid cannot be used for heart energy.
Thank you, Sensei. So I think I hope both of the speakers can address the question that rise in column chat box. All of the questions are very interesting, but our time is we have a limitation in time. So I would like to bring I give back this session to Dr. Mira.
Okay, we want to speak about the conclusion, but I think this time is very up. Do you want to say a little bit about the conclusion, Dr. Rizky? What is a sensei?
Okay, for the wrap up to Iso Sensei speak for today is FABP45 is the chaperone of fatty acids, and on his study, Professor Iso proved that FABP45 are expressed in cardiac endothelium and play a role to facilitate fatty acid from vascular to extracellular matrix of heart. And the lesion of FABP45 have been proved to cause disturbance in adaptive process, such as fasting and cardiac disease. Furthermore, the study FABP4 and 5 in adaptive physiology and cardiovascular disease in animal model show us the importance of energy balance for homeostasis. And disruptions of metabolic component, in this case FABP4 and 5, cause energy shifting from fatty acid to glucose and further cause imbalance of energy process in cells.
Okay, And then for Dr. Badrul conclusion, cancer stem cells are responsible for tumor maintenance and spreading. Diseases are resistant to chemotherapy. That's one reason why stem cell is one of the promising therapy for lung disease, including lung cancer. And also there is the role of CSC in the type of human mesenchymal stem cell in SARS-CoV-2 infection and their possible therapeutic application in COVID-19. There are several routes of cell delivery. Dr. Badrul and the team have developed aerosol-based cell therapy, which effective for lung disease. And there are still some challenge in cell therapy. Okay, thank you very much. There are so many questions in the chat box. The speaker can't answer all the questions. We really appreciate maybe Professor Iso and Dr. Badrul can answer all the questions in the chat box. You can write your answer in the chat box for every questions. And then we choose, okay, we choose two participants who have the most interesting question. And for Dr. Prof. Tatsuya Iso, we choose Dr. Sadia Ahmad. I'm sorry, we can't, yeah. Dr. Sadia Ahmad, and for Dr. Badrul, we choose Professor Nuzirwan Acang. Please, for the participant who got the question, who have received the rewards, please contact the committee, or committee will contact you. Thank you very much. We close this session, and please give a very big applause again to our very greatest speakers. It's a very interactive discussion, very interactive presentation. Thank you very much. We give back to the MC, Mr. Yusuf, silakan.
Well, thank you. Check. Well, ladies and gentlemen, thank you so much. Such a good discussion today. And thank you so much to Dr. Mira Sariputri, Ph.D., and also Dr. Mas Rizky, Ph.D. as the moderator, and Professor Tatsuya Iso, M.D., Ph.D. from Gunma University of Health and Welfare, Japan, and also Associate Professor Dr. Badrul Hisham bin Yahya, Ph.D. from University Sains, Malaysia, as the plenary speaker. And we learned a lot from the topic in the discussion, and thank you very much.
Okay, as the recognition and also the appreciation from the BICMHS committee, we would like to give a token of the appreciations to our plenary speakers and also the moderators. So in this occasion, please welcome the second Vice Dean of Medical Faculty Universitas Islam Bandung, Dr. Lisa Adia Garina, SPA MSc Med, to give a token of appreciation to Professor Tatsuya Iso, MD, PhD from Gunma University of Health and Welfare, Japan, as the plenary speakers.
Thank you for MC. To the honorable speakers, Prof. Tatsuya and Associate Professor Dr. Badrul Hisham, on behalf of the Medical Faculty of UNISBA, we thank you very much for the interesting presentation and useful information. Hopefully, we will see you again in the next collaboration. Once again, thank you very much. Big applause for the speakers.
Thank you so much, Dr. Lisa and also Prof. Tatsuya. Okay, and then the next.
And also, as the recognition and appreciation from the BICMHS 2021 committee, we would like to give a token of appreciation to the plenary speaker. Please welcome the second Vice Dean of Medical Faculty Universitas Islam Bandung, Indonesia, Dr. Lisa Adia Garina, SPA MSc, to give a token of appreciation to Associate Professor Dr. Badrul Hisham bin Yahya, PhD from University Sains, Malaysia, as the plenary speaker.
Okay, thank you very much again for Associate Professor Dr. Badrul Hisham. On behalf of the Medical Faculty of Unisba, we thank you very much for the great presentation. And hopefully we will see you again in the next collaboration. Thank you.
Terima kasih.
Thank you so much, Dr. Lisa, and also Associate Professor Dr. Badrul. Okay, and then for the next recognition and also the appreciation, it goes to the moderator. So once again, we would like to invite Dr. Lisa Adia Garina to give a token of appreciations to Dr. Mirasari Putri and also Dr. Mas Rizky Syamsunarno as the moderator of the first plenary speaker session in the second day of the BICMHS 2021.
Okay, thank you for the couple, Dr. Mirasari Ph.D. and Dr. Mas Rizky Ph.D. for contribution as moderators in the event.
Thank you very much.
Once again, hatur nuhun.
Thank you.
Big applause for them.
Thank you, Dr. Lisa.
Welcome.
Well, ladies and gentlemen, before we continue to the next agenda of the BICMHS 2021, let us have an e-group virtual conference photo session. Please turn on your Zoom video or camera in your device, and I will start to count and take the e-group photo together. Okay, please make sure your camera. Yeah. For the first page, in 3, 2, 1. Okay, next. Second page. In 3, 2, 1. The third page. In 3, 2, 1. Okay, next page. In 3, 2, 1. Okay, next page. In 3, 2, 1. Next page. In 3, 2, 1. Next page. In 3, 2, 1. Okay, for the last page. In 3, 2, 1. Thank you for all the smiles and wonderful energy for today's e-group photo session. And I hope the positive vibes can be spread to all the people around us. And back to Ms. Sandy.
Thank you. So moving on to the online presentation sessions. So today we will have 2 sessions of the presentation session. Now we will have the first session that will consist of 3 presenters from a biochemistry track. And in this session, we will have Professor Heri Garna, Dr. SPAK PhD, and Dr. Kasine Kateleka as the session chair. Yeah, before Professor Heri Garna and Dr. Kasine Kateleka chairing the session, I would like to read his profile. Professor Heri Garna, Dr. SPAK PhD, is a professor of pediatrics at the Faculty of Medicine Universitas Islam Bandung and was born in Ciamis on December 21. He earned his undergraduate and specialist degree in medicine from the University of Pajajaran's Faculty of Medicine, as well as his doctorate in medical science from Vrije University Brussels, Belgium. And he is currently focused with writing academic papers, and he continues to write articles in both Indonesian and English and to attend the seminars both nationally and internationally. And he is currently active in writing books with 4 books already published in the last 5 years. And then the next session chair. Yeah, next slide, please. Yeah, the next session chair is Dr. Kasine Kateleka as the researcher of the Halal Science Center, Chulalongkorn University, Bangkok, Thailand. So Dr. Kasine is a researcher from the Halal Science Center, Chulalongkorn University. She has biochemistry background and achieved her Ph.D. in biomedical science. Her research of interest are biochemistry and halal science and bioanalytical chemistry. She is one of the editors who wrote the age number halal haram masbuh ingredient list, a directory book of halal food additive established for facilitating food manufacture to reduce the cost required by the verification of raw materials that used in halal production. She has experience as a technical committee in working with the Standard and Metrology Institute for the Islamic Country, or OIC, or SMIIC. So thank you so much, Professor Herigana and also Dr. Kasine for being with us today to chairing the sessions. Hello. Yeah, Professor Harigana and Dr. Kasine, now is the time for you to chair the sessions. Please welcome. Thank you.
Okay, thank you.
Yes, thank you very much, committees, and thank you very much, Santi.
Yes, okay.
And very good morning to Professor Dr. Henry.
Okay. Now we start.
Yeah.
Okay. Yeah.
So, before we start the sections, let me indicate the presenter, the rules of the presentations today for just 1 minute. Okay.
Yes. Okay. Only 10 minutes and 5 minutes for questions.
Yes. So for today, presenter will have 10 minutes for presentations, and there we have 5 minutes for question and answer. And the Q&A will be facilitated by the section chair. And the audience and other co-presenters can ask questions using the chat features, which will be read by moderator. For today's presentations, recordings of the presentation is allowed. And make sure that you are in the place free of noise to avoid disruptions of the presentations. And please help the presenters give the presentations that they deserve by not talking, not eating, drinking, or engaging in activities that are uncalled for during the presentations. So, shall we start with the first presenter?
Yes.
Okay, so may I invite the first presenter for the biochemistry track.
Okay.
He will be in the topic. He will be giving us presentations in the topic of neutrophil lymphocyte ratios and C-reactive proteins in non-dialysis chronic kidney disease. Please welcome Professor Dr. Nusiwan Ashans. So, Tom is for you.
Yeah, thank you.
Okay, the first chance for Professor Achang, please.
Yeah. Sorry. It's clear, my slide?
It's clear.
First of all, I would like to express my thanks for moderator. And Assalamualaikum warahmatullahi wabarakatuh. Bismillahirrahmanirrahim. Assalamualaikum Muhammad wa ala sayyidina Muhammad. I would like to present my simple research in Cibabat Hospital. The topic is neutrophil lymphocyte ratio and C-reactive protein in no-dialysis chronic kidney disease. I work with my 3 colleagues from head of unit hemodialysis, Dr. Vidya from Cibabat Hospital, Susilewati, clinical pathologist from Cibabat Hospital, and Budiman is statistician from FKUnisba. As you know, chronic disease is a condition in the glomerular filtration rate for less than 60 millimeter per minute and per 1.373 cubic square meter. It found for more than 3 months. Now, we know that CKD is one of the main problem right now because of both of the health and economic field. You know, hemodialysis take a lot of money to support it. About the epidemiology of chronic kidney disease, estimates of global prevalence of CKD range from 11 to 13%, with fairly high mortality rate, about 9%, almost 10 to 20 from higher than general population. The report of Indonesian Renal Registry in 2017, the number of CKD patients was 3,800 and increased 6 times compared with 2027. The main factor death of CKD patients are infection. That's why it is a figure from US. You see the increase of year by year the incidence of CKD. Now about staging, because we research the stage 5 of CKD. Stage 1, GFR more than 19 and stage 5 less than 15. Oh, sorry. We know that inflammatory in CKD patient is a lot of report of Fazeri, report Transakaki, report on American Society of Kidney Diseases, so many report of infection in CKD. That's why we try to detect so early of the infection. The presence of chronic inflammation process can be detected early, examining various inflammatory markers such as C-reactive protein and interleukin in CKD. But the traditional inflammatory marker has been widely studied predictably. But that I said that. But a simple and cheaper inflammatory marker are still needed. That's why we try to research about simple and cheaper of inflammatory marker is neutrophil to lymphocyte ratio. It's very simple. We count the number of neutrophil and lymphocyte count, and we get the result. No need a special technique or no special. No, this is a base of the theory. Neutrophil is a cell for inflammatory reaction. If you get inflammatory, neutrophil will be increased and lymphocyte will decrease. That's why neutrophil lymphocyte ratio will increase. C-reactive protein. Protein is product in aqueous liver cell, produce amount, and CRP test measure amount CRP of the blood, detect inflammatory due to acute or chronic condition. Sorry, I repeat it because the time only 10 minutes. Several studies show that neutrophil lymphocyte ratio, that is marker of systemic infection. High and neutrophil lymphocyte ratio was reported to be associated with cardiovascular and in infection. Therefore, the aim of the present study was to determine whether lymphocyte ratio and CRP are associated with renal outcome in CKD patient. Material method. Method insists of Dr. Budiman. This study is observational analytic study placed in Cibabat Public Hospital, Cimahi, in period 4 months. But actually, I'm also very sorry. In July and August, the pandemic of COVID increased. The patient of COVID increased, but our college taker Hemorrhagia get COVID-19, and she should be private in mobilization. Private. Then we stop about 3 to 3 weeks. That's why the result is only 20 cases. Inclusion criteria, CKD state 5, of course, willing a participant on exclusion, sepsis, and so on. The result, in the baseline data, research subject, we got only 12 cases, women more than men, and for age equivalent. This is baseline data in table 1. Yeah. We said that the increase of CRP, increase of neutrophil lymphocyte ratio. We get a 12%. About distribution of age, our patient is more than 60 years. That's why in geriatric disease, the normal of neutrophil lymphocyte ratio slightly increase. Neutrophil lymphocyte ratio level, state 5, about 11, and CRP 31.95. The normal of lymphocyte CRP is less than 5.2. About the sex, yeah, female more than male. The correlation between neutrophil lymphocyte ratio and C-reactive protein, yeah, both of the marker is increased, but not satisfactorily significant. Correlation between you see in the table 5.4 discussion. You know that the mean of all patients CKD side 4 was 45 years. The same in another study. Distribution of the age the same of the other study. Neutrophil lymphocyte ratio will increase because normal only 3.31. And the same is another study in Europe. No, NRR, neutrophil lymphocyte ratio inflammation correlated with FKC2 score according to Zohorek study. The same with Bruno, increase of neutrophil lymphocyte ratio. C-reactive protein, the same. I not take a detail because we have limited time. And we have research limitation. One, the study could not rule out of phase B inflammation induction by pro-inflammatory cytokine by comorbid chronic disease like hypertension, like diabetes, like obesity, and so on. The study does not consider of treatment of the patient before. Conclusion: the mean lymphocyte nasal ratio was significant increase, and also C-reactive protein. Both of inflammatory marker increase, even though it's not significant. Suggestion: further research need to be patient do with more number of cases and may need it on the factor play role of the current of chronic inflammation. That's all. I think we have only 10 minutes to present. Thank you for audience. Thank you. Back to Prof. Harry. That's all my presentation.
Thank you very much, Dr. Nusit. If any audience have questions, you can type in the chat box. Then I will read your questions to our presenters. Yeah.
From the chat.
So may I ask, may I ask some questions, small questions? I wonder the sample size you use in this study. I'm not sure if we increase the number of the samples, the sample size, then we may have seen some significant or some trend of the relationship between NLR and CD values. So I'm not sure. Can you suggest in terms of the sample size for us? Maybe you have some ideas or some suggestions.
Thank you for your question. According to my statistician, we should take minimal 21 cases, but in present study we get only 12 cases. As I told you before, because in July and August, so increase of pandemic of COVID, and my patient or patient chronic renal failure to do hemodialysis can get it because it should be private isolation, and the doctor work there, get a COVID also. He stopped the research 3 weeks, and according to statistician, I should get 9 case anymore to get a conclusion. Without 21 cases, we can get my conclusion with statistician. Thank you.
Okay.
And the distribution is not good because of the standard deviation is so high.
I want to ask Professor Achang, can you divide the chronic kidney disease for all? Do you divide it into stadium?
Yeah.
The chronic kidney disease is all chronic or divided into the stadium?
Yeah.
Stage of the stage.
In this study, we should use chronic kidney disease in stage 5. That is indication for hemodialysis. not state 1 to 3, because it's not seriously ill. So many studies do it in state 5 of chronic kidney disease.
Okay.
Maybe in the future, we check all of the states and we compare the result.
Yeah. You wait until the number of cases increase.
Yeah.
Yeah. Okay.
Yeah.
It's no.
I hope in October my should stop it. I get a 25 patient.
It's no question anymore. So
thank you very much, Professor Nusiswan Asang, for your wonderful presentations today and very informative information for us today. So thank you very much.
Okay.
Thank you. Assalamualaikum.
Thank you.
Waalaikumsalam warahmatullahi wabarakatuh. So now we move to the second presenter. The next presentations will be on the topic of correlation between use of contraceptions and exclusive breastfeeding in Indonesia in 2017. Please welcome Dr. Fabrianti Prasmono Putri. Yeah, the time is for you, so you can share your research now.
Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi
wabarakatuh.
Good morning. I'm Febrianti Prasmono Putri from Universitas Negeri Malang. Today, I will present our research about correlation between use of the contraception and exclusive breastfeeding in Indonesia in 2017 Indonesia Demography Health Survey analysis data. Exclusive breastfeeding is recommended for infants up to 6 months of age without any other food. One of the factors that cause pneumonia, diarrhea, and high stunting in Indonesia is non-exclusive breastfeeding. Only 32.6% of the 13 million babies in the world are exclusively breastfed. World Health Organization target in 2030 is that 80% of infants worldwide are exclusively breastfed. Based on data from the World Breastfeeding Trend Initiative in 2012, Indonesia is ranked 49th out of 51 countries in the exclusive breastfeeding category. Exclusive breastfeeding in Indonesia in 2015 amount to 55.7%, in 2016 amount to 54%, in 2017 amount to 61.30%, in 2018 amount to 68.74%, in 2019 amount to 67.74%. This is still below Indonesia's strategic plan target of 69%. Breastfeeding mothers should pay attention to the use of contraceptive because improper use can reduce milk supply. So the baby does not get enough breast milk. The study used data obtained from the result of the Indonesia Health Demography Survey in 2017. The study also examines contraception without separating by type of contraception. Literature review. There are two factors can affect exclusive breastfeeding. The first factor is mother factors consisting knowledge, psychology, education, contraceptive use, and breast anatomy. And the second factor is environmental factors, consists of husband support and health officer support. The research design of this research is use descriptive analytic with a cross-sectional approach. The population of this research is women of childbearing age who were married and had their last child aged 0 to 6 months. The samples of this research is 1,891 respondents. The sampling technique is used total sampling, and this research is secondary data from 2017 Indonesia Demography Health Survey data. And data analysis that used in this research is a chi-square statistical test. And the first is univariate analysis. That first is age. Based on the table, shows that the most respondents were aged 25 until 29 years, namely 528 people, or 27.9%. The second is province, and based on the table, shows that the most of the respondents live in the province of West Java, namely 173 people, or 9.1%. The third is less education. Based on the table, shows that the most of the respondents have the less education of secondary school, which is 1,057 people, or 55.9%. And the next is baby age. Based on the table, shows that the most of the respondent babies were 4 months old, i.e. 320 babies, or 16.9%. The next is contraception types. Based on the table, shows that the type of contraception used by respondent is hormonal contraception. 74% of the 1,003 respondents who use contraception. And the next is contraception methods. Based on the table, shows that the most respondents use contraception, namely 605 people, or 60% of the 1,003 respondents. The next is contraception use. Based on the table, shows that many respondents use contraception, namely 1,003 people, or 53%. Meanwhile, respondents who did not use contraception were 888 people, or 47%. The factors that can affect the use of contraception are age, number of children, or parity, religion, and public knowledge. The next is exclusive breastfeeding. Based on the table, shows that the most respondents who did not give exclusive breastfeeding to their babies were 1,021 people, or 54%. Meanwhile, a total of 870 people, or 46% of respondents gave exclusive breastfeeding to their babies. Several factors can affect exclusive breastfeeding are including mother knowledge, mother psychology, mother education, use of contraception, mother breast anatomy, husband support, and support from health workers. The last is bivariate analysis. The table based on the table shows that as many as 605 people, or 60.3% women of childbearing age, used contraception and did not give exclusive breastfeeding to their babies. The results of the chi-square analysis can be seen that the p-value of the research results is 0.000. Thus, it can be interpreted that there is a correlation between contraceptive use and exclusive breastfeeding in Indonesia. This research is in line with other research conducted with theory, namely there is a correlation between the use of contraception and the smooth production of breast milk. Contraceptives that can cause slow breastfeeding are contraceptives that contain estrogen and progestin, such as pills, injection, and implant. which do not contain estrogen and progestin like the IUD or traditional methods. The conclusion is based on the study results, it can be concluded that there is a correlation between contraceptive use and exclusive breastfeeding in Indonesia in 2017. Thank you.
Thank you very much. We have a question from the chat box. So the question is, since it was almost 4 years ago, for the data, and the situation now is changed, do you think the findings still relevant to the current situations with the COVID-19 methods and others?
Thank you for the question. I think I will talk about the reason. The reason is Indonesia Demography Health Survey is the last data is in 2017. It took once in every 5 years. So the last data is in 2017. And is this relate about the current situation? I think is relate. That's all.
Yes, this one question from Santi Rahmawati. Yeah, this the presentation, but is the reason of taking the secondary data in year 2017. Since therefore now is changed until now. Yeah. Do you think the findings still relevant to the current situation with the COVID-19?
Thank you. I think it's relevance.
Because this for in 1917, the data, and now it's very changed. Because COVID-19, for example, this is
maybe.
The relevance is still relevant. Maybe.
Maybe.
Okay.
Yes. Our finding is relevant. So, are there any factor that you think it should be considered during the COVID-19? If you can design in order to get more information, do you think are there any factors that may be involved during the COVID-19 about the relationship between the contraceptives and exclusive breastfeeding?
Yeah. Other factors I think is husband support. I think husband support for use contraceptive and exclusive breastfeeding itself.
Yeah, you mean environmental factor maybe more effects during this time, right?
Yes.
One more question from Dr. Lisa. How to control confining factor in your research?
In my research, because it's a secondary data, so there are factors that cannot be researched in this research because the other factor doesn't exist in Indonesia Demographic Health Survey.
That is because it is only from the data, yes, not retrospective or prospective study. Okay, yeah. Okay.
Yes.
Other questions? There is no other questions. Dr. Kasini, because it is not question anymore.
Okay, so
or you can you
we move
have
to
more questions.
No, I have no questions. Thank you very much for the presentations.
Thank you.
So we move to the last presenter. The last presenter will be talking on the topic of factors affecting nutritional status among students in pandemic period, sleep durations, physical activities, and knowledge of balanced nutrition. Please welcome Meta Molida Damayanti.
Okay. Thank you very much, Prof. Helge and Dr. Kasini for introducing me. Let me share my presentation. But sorry, it's raining here. Hopefully my voice is clear and the internet connection is stable.
It's good. Nice voice.
Okay. Okay. It's so great to be here and honored to be here. To begin with, I would like to thanks to the organizing committee of Bandung International Conference on Medical and Health Sciences for allowing me to be a part of this event. My name is Meta Molida Damayanti, and in this great opportunity, I would like to give a short presentation about my research with my friend Yuniarti on factors affecting nutritional status among students in pandemic period, sleep duration, physical activity, and knowledge of balanced nutrition. Well, ladies and gentlemen, why am I interested in this research? The pandemic of COVID-19 that has been spreading worldwide give a massive impact on all aspects of our life, including nutritional needs fulfillment, especially in children. Children in the period of optimal growth are forced to adapt to this depressed condition. The government emits the policy of social distancing to prevent disease transmission. But on the other hand, it significantly limited our activity and strongly affect the sleep patterns and ultimately on nutritional status. Malnutrition is currently an important issue among children in Indonesia and even globally, and especially in developing countries. This phenomenon, for sure, needs serious attention and correct handling. How is the condition of nutritional status and limited access to health service in social distancing period? As we know, many factors affecting the nutritional status. So why did I choose the sleep duration, physical activity, and knowledge of balanced nutrition to assess in relationship with nutritional status? First, lockdown is applied to cut off the transmission of the virus, but the impact is it changes the physical activity quite significantly. During social restriction, activities we do outside is very limited. We spend most of our time at home, the drastic decrement of physical activity. Physical activity and regular exercise provide many health benefits. And the second, Changes the physical activity is usually accompanied by poor sleep pattern. Sleep is very important for the sake of children's health and happiness. Sleep characteristic of children such as sleep duration, time, quality, and variability were becoming more linked to health problems. This condition is quite worrying, especially among children because it will affect their general health. And the third, nutrients we eat are needed for tissue growth and regeneration, producing the energy required for metabolic processes. Nutritional knowledge covers all processes towards a good balance between body needs and nutritional intake at the right time and in the right proportion at the level of cells, tissue, organs, and the whole of body. Knowledge of balanced nutrition is needed to meet essential nutrition needs fulfillment. Simple screening can be conducted to see someone's nutritional status by calculating body mass index. That's the reason why in this research I intend to analyze the relationship between nutritional status with sleep duration, physical activity, and knowledge of balanced nutrition among a group of students in pandemic period. Here are the material and methods I have used in my research. This study is an analytic observational study with a cross-sectional design that was conducted at the Manarul Huda Islamic Boarding School in early March 2021. Subject all the students who meet the criteria. The inclusion criteria were students who were registered at the Islamic boarding school in 2021, 9 until 14 years old, didn't leave the dormitory during the pandemic, and were in the good health. While the exclusion criteria were students who were not present at the time of data collection. For the data collection, BMI calculation were used to assess nutritional status, and the independent variable in this study was sleep duration, which was obtained from the question of how much sleep is needed in one day, and physical activity obtained from the duration of exercise in 1 week, and knowledge of balanced nutrition through a validated questionnaire. And a statistical analysis was performed using SP is 23.0 version. The relationship between categorical variable was assessed by means of the chi-square independence test. Approval to conduct research was obtained from the Research Health Ethics Committee from Faculty of Medicine, Universitas Islam Bandung. After conducting such procedure, I have found out that sleep duration of less than 4 hours mostly have less nutrition or underweight category, as many as 7 people, 61.1%, and students who have sleep duration more than 8 hours mostly have normal nutrition, it's 75%. And based on table 2, students who have physical activity, and in this case exercise, more than 5 days in 1 week have less nutrition or underweight category as 80%, and students who have no physical activity or exercise most of the body in 1 week have more nutrition as many as 80%. In table 3 shown that the numbers of students who have more nutrition or overweight category with not good knowledge of malnutrition are 23.8%, more than students who are overweight with good knowledge, as many 5.6%. In this study, we involved a group of 39 students from Islamic boarding schools. Many factors affect nutritional status. Significant interaction was apparent for relationship between nutritional status with sleep duration, physical activity, and level of knowledge about balanced nutrition. In study states that body mass index with underweight category is closely related to less sleep duration. Sleep is crucial to one's overall health and well-being. Sleep that is both adequate and of high quality protects one's physical and mental health while also improving one's quality of life. This finding similar with Matriciani et al. In 2019 that state sleep in children such as duration, quality and variability are becoming more linked to a variety of health problems. An association was strongest between duration of physical activity and BMI. Respondents who didn't exercise had a higher body mass index than those who exercised. Physical activity with the right frequency increases the body's ability to properly care for vital organs, good metabolism, and body homeostasis. Respondents with a high level of knowledge to balance nutrition needs have a normal to underweight BMI. The level of knowledge, attitude, and behavior of balanced nutrition greater influences habit in fulfilling. Limitations are recognized due to the pandemic period. There is a risk of transmitting COVID-19 in primary data collection in form of direct doing face-to-face research for a long time. Further research can add other variables such as diet pattern, medical history, or family history of nutritional status. Let me conclude this presentation. My research resume is nutritional status determines the quality of life. Sleep duration, exercise, and knowledge of balanced nutrition are factors that can determine a person's nutritional status. Further analysis of other factors that affect nutritional status is needed in comprehensive manner. Well, this brings me to the end of my presentation. Thank you very much for paying good attention on my research presentation. I would be grateful if any kind of feedback. Alhamdulillahirrahmanirrahim. Thank you.
Okay, it is a nice presentation. Maybe from in chat, maybe the question from Vivek Setiawati. How do we explain why there were more children who were overweight who have less than 4 hours sleep per day?
Okay. Thank you very much for the question. I will try answer the questions. Maybe I will, please let me say it another way about sleep in children. It's not clear yet, but as I know, similar to adult, one's theory that on short sleep, increasing hunger and the hormone cannot control the appetite. Sleep is regulated by our circadian rhythm or body clock located in the brain. The body clock responds to light cues, ramping up production of the hormone melatonin at night and switching it off when it sends light. The body experiences in cycles throughout the night. Maybe, as I know, sleep divided many stages. Light sleep and deep sleep. In light sleep is transition from being awake to falling asleep. Heart rate and breathing begin to slow, body temperature falls, and muscle may twitch. Then a deep sleep is sometimes referred to as delta sleep because from the short delta brainwaves that are released during this stage, and our cell produce the most growth hormone to service bone and muscle, allowing the body to repair itself. And in this stage, begin to dreams. The body creates a chemical that renders it temporarily paralyzed, so that we don't act our out of our dream, expect ngalindur maybe, yeah. In this stage, the brain is extremely active and our eyes, although closed, dark back and forth, is if we were awake. So if the children not in good in sleep, maybe it's something wrong, or this maybe change with the useful of sleep, like the growth of growth hormone, and also the many functional metabolism in sleep. Okay, thank you. I hope my answer.
Okay. There's questions from Santi Rahmawati. The question is, based on your observation, how can we know that the children have good sleep in terms of duration of quality?
Okay, in this kind of pandemic, the data collection by personal data form and by questionnaire, and before the research begin, we, the team researcher, explained the benefits and goal and other requirement and all about the research, especially about sleep. Sleep duration is take with the questions of how many long do you sleep one day, and there is a multiplication optional. Maybe my answer is clear enough for you, Miss Santi.
That's interesting. I think one more question from Henny Mustafa, yeah, Mufliha. Is there any suggested maximum frequency of physical activity as those with mild physical activity were underweight in your study?
Yes, actually, I have similar curiosity about that, but I will try answer your questions. In this research states that over exercise is not good. Instead, the under exercise. So I think based on my optional answer in perform data collection, I think the best exercise is about 2 until 3 times a week. Maybe it's based on my research. Thank you, Dr. Henny. I hope my answer gave better understanding about my research. Thank you, Prof.
Only maybe one more, the last questions from Yuke Andriane. Is this condition really affected by the pandemic? How do you ensure that the conditions will be different from the non-pandemic period?
Okay, thank you very much, Dr. Yuke. Your question is comparing about before and during pandemic. It's actually my research is my paper is one of the result of my main project research. And before I do the research, it's a pre-research conducted maybe a year before, and the result states that there is changes pattern of sleep before and during pandemic. And it's a kind of pandemic. There is limitation interaction, social and physical activity. And physical activity sometimes very relevant with sleep pattern. Maybe it's my answer, Prof.
Yes.
Maybe
yes. The time is enough. It's more time more. Maybe you can answer in the
yes,
chat.
we have
Yes. Maybe.
Okay. Again, I thank you all for having me. Hopefully, in the near future, we could collaborate in other research. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. So the next section will be give certification to our presenter. So Professor Dr. Henry, maybe you can say some appreciations. Maybe you can say something to our presenter before giving a certificate for them.
Yes, I think it's very interesting session, and thank you for all participants. And I hope we will again meet another event. Thank you for sharing for Dr. Karini. This is the first time we meet. Yes, okay.
This is my first time at a session here as well.
Okay. Yeah.
Thank you very much, our committee as well, for inviting me to be part of this very grateful seminar. So, may I call the first presenter to receive the certificate? Am I correct, Miss Santi? Okay.
Yeah, so we will display the display the PPT first. Okay, please.
Okay.
Yeah.
Okay. Congratulations to Professor Dr. Nusirwan Assange. His round of applause podiums. And the second presenter, Miss Febrianti Prasmonoputi. And the last presenter, Miss Nita Molida Bamayanti. Congratulations to all, and thank you very much for very good, very interesting presentation today.
Mr. Yusuf? Okay.
Well,
So now,
thank you, ladies and gentlemen. Thank you so much once again, Professor Heri Garna and Dr. Kasini Kateleka for the great sharing in this session. And for the next, congratulations to all presenters who are already presenting their papers. All of you already done a really good job. And finally, thank you to all audiences and participants who already participated and actively involved in giving the questions, comment, and feedback, and also even suggestion to the presenters. Your active participation and contribution in the scientific forum of the BICMHS 2021 is highly appreciated. Well, ladies and gentlemen, to memorize and cherish the moment, let's have a group photo together.
Okay. Before we take group photos, we would like to inform some informations regarding, because Dr. Kasini Kateleka is also already being asked today. So I would like to inform also on behalf of Halal Science Center Chulalongkorn University, Thailand, and also the Research Synergy Foundation, that we will invite all of the participants to join and also to participate in the our upcoming conference with the theme of the halal studies, but it is interdisciplinary studies. So Dr. Kasini, do you want to say something, and maybe the admin can share a screen for the ISATEC for a while? So because this is also a good opportunity that we can connect between the Unisba and also the Chulalongkorn University and also the other audience. Okay, please, Dr. Kasini.
Thank you very much, everyone. For our upcoming event, on behalf of the Halal Science Center Chulalongkorn University in Thailand, we will have the Thailand Halal Assembly, and we also conduct the International Halal Science and Technology Conference. This is our 8th in Thailand on December 14 to 15. And we call for the papers from all participants in fields in various field of study. You can see in the picture here, and you can submit your works, your thesis, your research in order to join our events. And we also conduct as a virtual conference as well, and we hope that we can see all of you in our conference. It will be on 14 and 15 December, but the time for submit the papers, full paper, will be available now until 14th November. I hope to see you all in our conference as well. Thank you very much for time for sharing the our conference.
Okay, thank you so much.
You're most welcome, Dr. Kasine.
Thank you so much. And don't forget that this conference is fully supported by the Halal Science Center Chulalongkorn University. So it is free of registration, both as the presenter who submit the paper and also, of course, the attendee. So, okay, back to Mr. Yusuf. And once again, thank you, Dr. Kasine, for being with us today.
Okay, thank you so much. So we're back into memorize and cherish the moment. Let's have a group photo together. Please turn on your Zoom video on your camera device. I will start to count and take the E-group photo together. For the first page, in 3, okay, please smile. 3, 2, 1. Okay. Okay, next page. In 3, 2, 1. Okay, next page. In 3, 2, 1. Okay, the next page. In 3, 2, 1. Okay, for the last page. In 3, 2, 1. Okay, well, thank you, ladies and gentlemen, and back into Miss Santi.
Okay, thank you so much, Mr. Yusuf. And once again, thank you again for our beloved session chair, Dr. Kasine, and also Professor Helge. Okay, allow me to read some information regarding the post-conference process or the post-conference regarding the publication of the BICMHS 2021. So once again, to all the presenters or the author who already submitted their conference paper, so please pay attention to the timeline and also the process that you need to do after the conference end. So if you want to be included in a given journal recommendations for published in the international journal, so please kindly send your full paper, your extended one, because your conference or your short paper already published in conference proceeding. So you need to make some paraphrase for your submitted conference paper, and then at least 80% to avoid the self-plagiarisms. So because there will be another second round of scientific review process, yeah, including the plagiarism check and also the content review check. And then after the conference finish, the ISSN and also the DOI numbers will be assigned soon to your article. So your article will be indexed in a Google Scholar and can be cited by the other scholars around the globe. And in addition, in this opportunity also, please, I want to inform you that you can upload your full paper in your ScholarVein account under the menu of upload, or can send through the email at info@bicmhs.com. Okay, And next, for the sharing sessions, before we take a break, so we would like to invite one representative in this room to share the experience in participating at the conference, because we believe that by sharing, we can encourage other scholars or others, students, academicians, and also practitioners to join the upcoming scientific forum or the conference. So in this opportunity, it is an honor for us to invite Professor Nuzirwan Achang to share the experience of today, the BICMHS 2021. To Professor Nuzirwan Achang. Hello, Prof. Okay. Is Professor Nuzirwan Achang is okay? Maybe he is not around. Okay. So opportunity, I would like to give this opportunity to share the experience. To share the experience to all of us today. So I would like to invite Dr. Meta Maulida Damayanti.
Okay, thank you very much, Ms. Santi. I will try to give feedback or testimonials about this conference. I think this conference is good and should be held regularly as a forum to share information about research. And especially for Research Synergy Foundation, it's my second time, because my first time was when I took part a series of coaching articles for F1000. For me, who just learning and just trying to publish international articles, it's my first time, Ms. Santi. The guidelines provided by the team were very clear, systematic, and open. Hopefully, this kind of program will continue. And I thank the Research Synergy Foundation for that. And I seem to be getting addicted to trying to publish my article. And I don't really expect the results to be immediately accepted, but I still enjoy the process. Thank you, Ms. Santi.
Thank you so much, Dr. Meta. Hopefully that we will hear a good news, yeah, from your article that already submitted. Okay, once again, thank you. And don't forget to submit your extended if you want to get another recommendation for the journal publications. Once again, thank you, Dr. Meta, for representing these online presentation sessions for the first sessions. Okay, so don't forget to fill the feedback form, okay, to fill the feedback form. And later on also the committee will send through the chat box, yeah, we'll send through the chat box. Okay, back to Mr. Yusuf.
Okay, thank you, Ms. Santi. And please remember to all audiences that at 4 p.m. we will have the closing ceremony. At the closing ceremony, there will be an announcement for the best presentation category, and also best paper category, and acknowledgement for session chairs. So please ensure that all the participants stay until the end of today's conference session. And now we will have a short break until 1:10 p.m. And once again, thank you so much for your kind attention, and see you soon again at 1 p.m. in the same room and the same link. Thank you very much.
Thank you.
Oh, so.
Yuki ada bolpen tuh.
Nggak bawa. Di tas ada nggak?
Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, distinguished participants and attendees, welcome back again to BICMHS Day 2, today 2021. So moving on to the online presentation session, now we will have the second session that will consist of 6 presenters from medicine, medical education, and nutrition track. So in this session, we will have a Dr. Henny Muflihah, Dr. MKS, Ph.D., and also Associate Lecturer Marcela Vera as the session chair. So before they chairing the session, we would like to read their profile. Henny Muflihah, Dr. MKS, Ph.D., graduated from the Faculty of Medicine, Universitas Gadjah Mada, and had her master degree in Pharmacology from Faculty of Medicine, Universitas Padjadjaran. She had her PhD from Sydney Medical School, University of Sydney, Australia, sponsored by the Australia Award Scholarship. She worked at the Tuberculosis Research Program at the Centenary Institute Australia starting 2013 until 2017, and received some awards during her work from Keystone Symposia Future Science of Fun, USA, the Federation of Immunological Society of Asia, Oceania, Singapore, Australasian Society for Immunology, Australia, and Sydney Southeast Asia Centre, Australia. She has interest in the field infectious disease, particularly tuberculosis and COVID-19 disease. Her research about the immunology of tuberculosis vaccine have been published in 2 reputable international journals, Vaccine and Mucosal Immunology. Henny is a lecturer at the Department of Pharmacology, the Faculty of Medicine, Universitas Islam Bandung, and now being the head of the research laboratory. Welcome, Henny, Dr. Henny.
Welcome. How are you?
Alhamdulillah, good. Okay, Ibu, we will start to read the second profile of the session chair first. Okay?
Sure.
Okay.
Well, ladies and gentlemen, we also have associate lecturer Marcela Vera from Arellano University, Philippines. She earned a Master of Arts in History from Philippine Normal University, Taft Avenue, Manila. She met the completed Master of Arts in Education in Social Science in the same school, and also she is a candidate for Doctor of Educational Management from Polytechnic University of the Philippines, Sta Mesa, Manila. She is a member of the Asia Pacific Consortium of Researchers and Educators, as known as APCORE, and also she is an associate lecturer in the College of Arts and Sciences at Arellano University, and a research advisor in expanded tertiary education, equivalency, and accreditation program department in the same school. She is a practical research teacher in Mariano Marcos Mem High School, Depth Education Manila. She is a module writer at AMACAP, Quezon City, and Arellano University, and book writer in CNE Publishing Inc., Quezon City, Philippines. Okay, Dr. Henny Muflihah, PhD and Associate Lecturer Marcela Vera, thank you for being here with us today. From now onwards, we'll pass the session to be led by both of you. And now the time is yours to chair the session. Please welcome Dr. Henny Muflihah and also Associate Lecturer Marcela Vera.
Thank you, Pak Yusuf and Ms. Santi. So thank you for the introduction for me and Dr. Sheila. So, ladies and gentlemen, we are coming to the last session for oral presentation today. And I think during the BICMHS conference, Marcella, Farah, and I would chair this session. Marcella is going to chair the first 3 presentations, and I'm going to be the timekeeper at the time. Then we will swap the task. I'm going to chair the last 3 presenters, and then Dr. Sheila will be the timekeeper. I would remind to all the presenters that the total time for each presenter is 15 minutes. Ideally, it consists of 10 minutes presentations and 5-minute discussions. The timekeeper may remind you 2 minutes before the 10 minutes of presentations end. I invite to the audience for giving feedback or questions or suggestions, either by writing on the chat box or by raise hand on the discussion sessions. Please don't worry about the language. You may have questions in Bahasa, and I'm more than happy to translate it for you. I think we already understand each other anyway. So, all right, Dr. Sheila, I would like to hand over the chair session to you.
Okay. Good afternoon. I want to present our presenters today. This afternoon, we have Miss Aliana Tercina. The paper title is The Effect of Gymnastic in the Healthy Elderly on Physical Fitness and Cognitive Function in Indonesia or Elderly Institutional of West Java Province. Okay, so that's for track medicine. And then we have Ms. Mia Kusmiati. Her paper title is The Most Influential Factor to Pass Rates of Modified OSCE Exam on Student Batch 2 during Pandemic Period. And the third, Ms. Sylvia Mustika Sari. The paper title Contested Perspective from Undergraduate Students and Teachers on Formative Assessments, a Qualitative Study. These are from track medical education. So for the next track, for nutrition, we have our Mr. Ariel Budi Irianti. The paper title is Effect of Unbalanced Nourishment on growth period to memory in mice. Then Miss Anna Sarjan Betterini. The paper title, Extra Virgin Olive Oil, Nano Emulsion, Attenuated Inflammatory Response in LPS-Induced Sepsis. And the last one, but not the least, of course, Mariska Sabrina Setcha organoleptic test of boba flour substitution of pumpkin seeds high in fiber and protein. Okay, so thank you for that. I'm sure all the presenters are present. Dr. Henny?
Yes, we should start, yes.
Okay, so now may I call on, yes, the effect of gymnastic in the healthy elderly on physical fitness and cognitive function in Indonesia Elderly Institutional of West Java Province by Miss Aliana Tercina. So, Miss Aliana Tercina, are you ready?
Yes, of course. Thank you very much.
Have your time is yours. The time is yours.
Yes. Thank you for Dr. Marcella Tivera and Dr. Henny for the opportunity given to me. I will present a research article with the title, The Effect of Gymnastic in the Healthy Elderly Please, I share my PowerPoint.
Yes.
Good afternoon, everyone. Today, I will present a research article with the title, The Effect of Gymnastics in the Healthy Elderly on Physical Fitness and Cognitive Function in Indonesia Elderly Institutional of West Java Province. The World Health Organization, WHO, estimate that between 2015 and 2030, I'm sorry, 2030, there will be an increase of 60% in the number of elderly people in Asia and Africa. According to the Indonesian Central Bureau of Statistics, the number of elderly people in Indonesia reached 20.24 million, equivalent to 8.03% of the entire population of Indonesia. Indicators of the success of health development in a country can be seen from the benchmark of progress in improving the quality of life and increasing health status, one of which is the number of elderly people from year to year. This can be overcome through the WHO active aging concept, which aims to build elderly people, who are physically, socially, and mentally healthy despite the aging process still happen. Aging can be recognized from changes in the body, both physically and psychologically. Physical changes in the elderly include the 5 senses consisting of eyes, ears, smell, taste, and skin. But not only physical changes that occur in the elderly, psychological factor will also experience significant changes. Physiological changes that can occur include paranoia, behavior disorder, depression, and economic post-power syndrome, socialize well and still consider himself to be part of the community. In this study was conducted using an observational analytic with a cross-sectional approach to determine whether there are influence of elderly exercise on physical fitness level using the 6-minute walk test. Assessment method will determine the effect of elderly exercise on cognitive function using the mini mental state method examination. The sample using a hypothesis test to proportion, sample size 40 people, 20 people treatment group, and 20 people control group. Data type, primary data. And processing use a chi-square test because both data have a categorical measurement scale. For place, the Indonesian Elderly Institute, West Java Province, for the period February until June 2017, and data analysis using SPSS for Windows version 18 with 95% confidence degree and p-value less than 0.05. Inclusion criteria for age, elderly group include healthy elderly women aged 60 and 69 years, and education level at least grade 3 elementary school graduates, and attending gymnastic at least 6 months until 1 year with a frequency of 3 until 5 times per week, or have participated in exercise for less than 1 month with a frequency of exercise less than 3 times per week for the elderly group who do not exercise regularly. This is category physical fitness, consists of excellence, good, moderate, fair, and low. And category of cognitive examination or MMSE, score normal is 24 until 30, probable cognitive impairment 70 until 23, and definitive cognitive impairment 0 until 16. This is questionnaire of mini mental scale examination. We use MMSE versi Indonesia. In the table, characteristic of the elderly based on age in the elderly group who routine and not regularly exercise between the age 60 until 65 years, have an average age that is not much different, namely 63.68 years old with regular exercise with a standard deviation of 1.64, and 63.25 years, the elderly do not exercise regularly with a standard deviation of 1094. In contrast to the elderly with an age range of 66 until 70 years, the average age of the elderly for routine exercise was 66.83 years with a standard deviation of 1.33. Based on the education level of the elderly routinely in gymnastic, the level of education equal to elementary school is 60.3%, more people when compared to the others. While in the non-routine group of gymnastic, the education level is at the junior high school level. The largest is 58.3%. The average height in both groups was 149.35 meters with a standard deviation like that. The weight characteristic of two groups were 60.32 kilogram with a standard deviation 11.15 and 59.59 kilogram with a standard deviation 11.81. Maybe I will trouble this. Okay. In table 2, overview of elderly mileage in Indonesia, elderly institution in West Java province. Based on the result of the 60-minute walk test, information is obtained that the maximum mileage is 723 meters. Oh, sorry. 657.50 with regular gymnastic and not regular gymnastic are 579.60 with a standard deviation of 109.58. And the effect of gymnastic with physical fitness level in elderly at Indonesian Elderly Institute, West Java Province. The data were analyzed using SPSS program for window, obtain a p-value of 0.025, so it can be concluded that there is a statistical significant relationship between gymnastic activity and the level of physical fitness of the elderly at the Indonesian Elderly Institute in West Java Province.
You have 1 minute more.
Okay. For discussion, this is the distribution of elderly with good physical fitness who routine participate in gymnastic. It's good 18 people and fair 2 people. This is distribution. Oh, sorry. In not regular gymnastic like this. And the effect the gymnastic with elderly cognitive function for regular gymnastic it's 20 elderly people, and not regular gymnastic normal 15 elderly people, and probable cognitive impairment were 5 elderly people. And this is for discussion. According to Fajar Sriwahyuniati, there is an effect of elderly exercise on physical fitness, the same as Rahmat and Nurce, and maybe American Medical Association, regular physical activity preference is cognitive decline, and elderly who participate in gymnastic are 60 times more likely to have normal cognitive function, is according to Lanawati. In conclusion, the result of the research on the level of physical fitness physical fitness of the elderly who are p-value is 0.025. There is a significant relationship between gymnastic activities with level of physical fitness and level of cognitive function. P-value the same as 0.024. There is significant relationship between gymnastic activity with level cognitive function. Thank you very much.
Thank you. Dr. Sheila, we have 1.5 minutes more.
Okay. So may I see the questions? Let us see if there are questions in our chat box.
There is one, Ms. Sheila.
Okay, so here. Yeah, it's yeah, thank you, Miss Alia. So your presentation is really interesting. So here the question, can you explain me, this is from Yushiriana, why there are some elderly that have a good physical fitness level, but they have no regular gymnastics?
Because this research explain this The has the education. Sorry. Hasil MMS-nya lebih bagus gitu, maaf ya. Sorry, I can speak in Indonesia. Dengan olahraga teratur, ternyata dari hasil penelitian ini hasil fungsi kognitifnya lebih bagus dibandingkan dengan para peserta yang non-regular gymnastic. Thank you.
Dr. Henny, please kindly translate.
Oh, okay. So yeah, Dr. Arya said, so the elderly people that the study found that that the regular gymnastic can have effect in good physical fitness. I think the question is maybe why there are some good physical fitness, but they don't do the regular physical activity. Maybe Dr. Alia want to explain more? So it's not what you find here. So the question is not what you find, the finding in your study. So the question is why there are elder people who have good physical fitness, but they didn't do regular gymnastics.
Because the active aging can be elderly people can jadi
you can speak by bahasa.
Yeah,
should be.
The aging process can be diperlambat gitu ya, diperlambat dengan kalau kita melakukan olahraga dengan teratur gitu.
Oh, okay. So the aging process can be postponed or can be slowing down by the regular gymnastic activity. That's the point of from Dr. Alia. I'm afraid we ran out the time, Ms. Marcella.
Okay, so thank you. Thank you so much.
Thank you, Dr. Alia.
Yes.
Thank you, Dr. Henny.
So let's go to the next presenter. So the title of the presentation, of course, is the most influential factor to pass rate of modified OSCE exam on student batch 2 during pandemic period by Ma'am or Miss Mia Kasmiati. Okay, Miss Mia Kasmiati, are you now ready?
Yes, ma'am. Inshallah, I'm ready.
Okay. So now you may have your floor or your time.
Okay. May I share my screen? Okay. Whether my slide is you can see now?
Yes, but maybe in the slideshow, please.
Okay. Wait a moment. Is it okay?
Yeah, that's very good.
Okay. First, maybe I would like to thank you to Dr. Henny Mufliha, MKes Ph.D., and also Madam Stella Rivera for giving me a chance to present our study. My study, the title is the most influential factor to pass set of modified OSCE exam on student batch 2 during pandemic period. This is my research or my study collaborate in the medical education unit team, me and my colleagues, Dr. Rika, Dr. Ibu Anissa, and Miranti Kanyadewi. Okay. As we know that the pandemic COVID situation caused increasing use of the technology in medical education. Also, the learning process of clinical skill is among the most complicated applied online. Therefore, it requires standard patients and supporting equipment to perform clinical skill learning activities. And at the same time, the significance of online learning on skill domain is influenced by many factors. For the research gap in my study, we have found it that in Indonesia and other countries in the world, however, health profession education wonders it's necessary to set a modified online OSCE. The pandemic situation affects the OSCE execution in undergraduate medical education. Thereby, it was implemented the modified OSCE exam. Yet, effectiveness of this assessment ought to be evaluated in terms of achieving the learning outcome. And until now, there has been no evaluation on the implementation that has been running. So for the background or the introduction I mentioned before, we have two objectives to our study. The primary objective, we aim to identify the most influential factors to achievement of the modified online OSCE. And the secondary objective, we aim to evaluate the students' learning behavior during online learning activities. As we know that according to one literature, stated that there are three components that influence the effectiveness online or blended learning in terms of transactional distance, cognitive, social, and teaching presences, and independent learners. The online learning is widely used in the medical education, lead to relevance one perspective under medical students. This is according to the other literature by Sandu and Wulff in 2020. And also the learning behavior towards clinical skills course of medical education reflects noticeable behavioral patterns that students show as the undertaking learning path. So furthermore, the effectiveness of online learning might be impacted by many factors such as technological aspects, user-friendly online platform or tools, class activities, and assessments. So research has identified in the other literature a lack of social and teacher presence in online course as the major factors leading to student attrition. And this is affect to student academic achievement. For the research method, I have two phases for conducting our study. The first phase is conducting by exploratory factor analysis, using or selected the participant amount of 92. And from EFA, we found out of 5 things were expanded to become 39 items, and was then yielded 8 imaging factors. And the factors or the mind questionnaire was well constructed and valid by EFA. And the second part, we have we do the through survey. We select the participant amount of 87. Through survey was conducted to 87 participants of batch 2 in academic phase of medical students by using the simple random sampling strategy. And for the analysis of the data, we have the method, or we have to this method maybe, yeah. The true simple linear regression using SPSS version 24 with p-value 0.05. And the other, we use the Amos version 26 to see learning behavior still online. And this is represent structural equation modeling regarding 8 variable latent or 8 factors. This slide show that the PET analysis of 10 factors related to the independent learners and pass rate of the OSCE of our study that has found start from teaching presence, experiential learning, student access to resources, learning platform availability, student preparedness, student reflection, and transactional distance. This is influence the independent learner, and the more advanced influence the pass rate of the OSCE. Our study finding also that the characteristic demography, something like this in the table, that in this table depicted that the amount of students who taken regular admission test amount of 79.31% was more than specific intersection with the ratio 4 proportionate 1. And our study also found that the average rate of the OSCE pass in the academic batch 2, student batch 2, amount 87.07% with the high achievement. And for the most influential factor of the pass rate of the OSCE in the some variable, we can also found that the variable of learning platform availability and transactional distance are the most influential factors to pass rate of modified OSCE online. The power of correlation on transactional distance amount 23.5%, and the rest of 76.5% is influenced by the other factors. And the amount of 8.7% component of learning platform contributed to success the passing rate of the OSCE. Why this is happen? I think the reason is this is due to the most foundation concept of success passing rate rather than teacher-learner relationship in our study on in our faculty. And this slide depicted us that diagram of structural equation modeling between the characteristic of independent learner with various factors influencing. We can see that, okay, from this figure that two factors that most influence the independent learner in terms of self-reflection, because we have the value 0.60, and the student access to research learning. Student access to resources have 0.053. And for the goodness of this parameter by Kasule per DF or mean in terms of 2.179, and for RMSEA as 0.0 and this is the rule of according to the rule of thumb, if the RMSA have value less than 0.8, this is indicated the model is fit, and the other. Okay, so our study, we are conclude that two factors to objective or to point. The first, the factors influencing pass rate of the modified OSCE. To answer the objective one, there has been a positive impact of the learning platform availability with the value 0.013 and multiple square correlation 0.087, and from transactional distance toward passing rate be modified on the OSCE with the value like this. And the other thing that the learning behavior toward clinical skill, our findings highlight that self-reflection and student access to resources have significant contribution towards independent learner characteristics. Thank you for my presentation. This is my or our presentation about the our study. Again, thank you so much to Dr. Henny and Madam Sela Tifera. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. We have 5 minutes discussion, Marcella.
Yes. Okay. Yes, Dr. Santi. So let us see the questions in our chat box. So we have.
You have one actually.
Yes. Ma'am Mia or Miss Mia, can you explain about transactional distance in your research? So that's the question.
Transactional distance in my study is depicted or indicate that the how far the distance between student and lecturer to get engagement to do the learning. I mean, because in the online learning, sometimes we can sometimes the distance between student and teacher or the lecturer sometimes is far, sometimes is near, but this is also, or this is depend on the how to engage or how to build the relation between student and teachers to do the engagement in the our learning. I think something like that, Miss Yuni. It is enough.
Okay. Transactional, of course. Maybe it's related of how you are going to maintain, the score for the objective structural clinical examination. And we would like to see the sample maybe for them to be familiar with the OSCE. Then I think it is a form of a performance-based testing. So it's not the written exam. It is the performance of a clinical candidate. So it is how you are going to see or to find of course, the learning modality in clinical students or medical students. Okay, so my question, of course, is in your study, what are the possible self-reflection do you think is very effective in this kind of learning modality?
Okay. Thank you, ma'am, for your question. I think for the self-reflection, it is the important factor to build on to get the intention or to insert or to untuk bisa masuk ke dalam proses pembelajaran. My students have the factor self-reflection, because with this self-reflection, students can assess self by self. What is the strengths? What is the weaknesses to do the learning? I think because this is the important. Yes, because depend on the student, but this factor can be reflected how student can be assessed his or her strength or weaknesses, or also maybe how to get the learning achievement by student itself. I think that is Ms. Madam Sarah.
And another one is, I just want to know what are some challenges for the resources in our online modality. What are some challenges in terms of resources?
Okay. I think the other challenges for online modality, for online learning, something like cognitive presence. And also the social presence maybe, because a bit far to social presence or social relationship between student and student, or student and the lecturer. And cognitive presence here indicates the degree to which students can create meaning through constant exchange of thoughts, questioning, answering, brainstorming, and also and so on.
Okay, so
we are not the time. We are not the time, Ashela.
Yeah, sorry. Okay, so let's go to, thank you so much, Ma'am Mia. So let's go to the next presenter. Okay, so let us call Ma'am Sylvia Mastikasari for her paper title, Contested Perspective from Undergraduate Students and Teachers on Formative Assessments, a Qualitative Studies. So are you ready now, Miss Sylvia Mastikasari?
Yes, inshallah, yes.
Okay.
Thank you.
The time is yours now.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Thank you so much for this opportunity. I would like to share my slide. Okay. Is it clear enough, my slides?
Yes. Yes, you can put on the slideshow now. Yeah.
Okay.
That's
Thank
great.
you. Thank you. Bismillahirrahmanirrahim. Let me introduce myself first. My name is Sylvia. I'm from General Ahmad Yani University, and I'm in Medical Education Department, same as Miss Mia. And today I would like to share a bit about my research. It's about contested perspective from undergraduate students and teachers about formative assessment. And this research is actually the qualitative study. I would like to start my background of research with this slide. This kind of this slide share us that the assessment right now is has to be change in paradigms. First, we know that we already assessed the student, but what we assess is actually the achievement of learning outcome. So that's what called as assessment of learning. And then after that, many research has more attentions to what student has from the assessment, or what impact of assessment in students. So that's called as assessment for learning era. And then right now, we are actually in the middle of assessment for learning paradigm to moving forward into the programmatic assessment era. So that many factors that influence this paradigm change, especially how the approach of teaching in that context. For example, we have to move from the teacher-centered to student-centered before we know how to assess students for their impact in learning. And right now in Indonesia, we are moving from the assessment for learning to the programmatic assessment era. But we know that many factors that has been influenced the implementation. So based on the paradigm change, currently right now, research in medical education, especially leading in student assessments, try to explore what is actually the relationship between learning and assessment itself. So the mechanisms of how actually assessment can drive learning is in undergraduate context of medical educations is defined as continuous feedback, and the continuous feedback in formative assessment, such as in a tutorial or even in a skills laboratory activities. So, but these implementations actually need a strong relationship, because the continuous feedback needs real or partnership relationship between undergraduate students and teacher as well. So it's actually related to the socio-cultural theory, and then also I come up with the Hofstede classifications of national cultures, and Hofstede classified Indonesia that has a high power distance and also collectivist. So this cultural aspect bring us to have underlies what actually happened in this recently point about the perspective of our context in undergraduate students and teachers about this formative assessment, how well they can aware that the formative assessment can be done as an assessment for learning concepts. So So our research questions actually about the perspective of undergraduate students and teachers about the formative assessment. Our methodologies is a qualitative inquiry with a phenomenological approach, and we use this research because we want to have a deep explorations about their perspective in formative assessment from teacher and students. And for this inquiry, we provide the guiding questions as follows: how actually students and teachers defined a formative assessment for them, and then how students and teachers experience during formative assessment, presently formative assessment right now, and then how students and teacher are actually perceived during feedback in formative assessment. Because as we know, feedback is actually the motor cycle of a formative assessment, or the concept of for assessment for learning. Our respondents in this study was actually students and teachers from undergraduate levels. And then we collected, the data was collected with semi-structural interview through focus group discussions for students and lecturers. And then we conducted 6 FGD of undergraduate students and 2 FGDs of teachers. And then to increase the degree of trustworthiness of this study, we triangulate this data using in-depth interview for 2 lectures and 4 students to have triangulate our data, and also the member checking methods. For ethical considerations, it's has reviewed by our institutional review board of ethics. And for data analysis, we use the phenomenological approach, and then the transcript for the focus group discussions and in-depth interview were coded and then categorized, and then become the themes from teachers and students. And for the final themes analysis, we contested the teachers and students' point of view about formative assessment. So our first finding is actually about the themes we found from the transcript, code, and then categorized, and ending in the 4 central themes of student perspective in formative assessment. First is they actually feel assessed in learning activity. Actually, they don't know much about the definitions of formative, but when we use the probe questions in focus group discussions, they already feel, so formative is actually the assessment in learning activity. So they feel like they are assessed in learning activity. The second one, they feel assessed when learning activity, and then their concern was the final score, not the feedback, not the relationship, but the final scores. And then we also found that the preference is in the instructive feedback. So they preferable to have the instructive feedback from teachers to students. So as we can see, one of the quotations here, I hope they just tell us what to do. They just tell us what is the right way to do that. For example, that's one of quotations. And we also found that there are some negative feedback form in our students' perspective about this, and the negative feedbacks currently accepted as a consequences of the students. That's our term. So they feel that these lectures, they a little bit confused, for example, in this quotations, but the method of teachers give them feedback is found to be a little embarrassing for them. That's our that's our first finding. And the second findings is how the teachers Okay, how the teachers perspect, and we found that the teachers having the difficulty in determining individual scores, concern about final scores also, and feedback given is actually related to students' materials, and students always need to improve themselves. That's from the teacher's point of view. And the last one we contested to found the 3 terms of our formative assessment cultures. Right now, we found that from students and lecturers' point of view, we found that actually students and teachers right now is focused on the summative perceptions of formative assessment, so that they care about final score. And the feedback, we found the theme that there is a hierarchical relationship to provide feedback, and it's also related to teacher-centered feedback. So from the students and also the teacher's point of view, they actually preferred the teacher-centered delivery feedback. So we found that the summative perceptions was actually emphasized by Chillers, that there is a strong relationship between teaching approach with assessment. So that's the rationale. And also we found that in the Indonesian context, students seems to happy with the instructive feedback, and also teachers happy with the given one direction feedback. So as a conclusions, we know that there is a long way to have a paradigm change of assessment for learning, especially in an Indonesian context. But we recommend this research as a basic point to develop another innovative formative assessment that have more positive learning impact in our students. Thank you so much for this opportunity. I hope I'm not run of time. Thank you.
Yes, we have 3 minutes. We have 3 minutes for discussion, Marcella.
Yes, thank you, Dr. Hani. Yes, Dr. or Ma'am Sari. Let us see the questions in our chat box. So I would like to add about the other.
No, it's.
So I'm sorry. So no questions. I have only one question for the assessment. We have our formative assessment and the final assessment. So what do you think is the preferred one of your institution?
The preferred one.
The preferred result of the students. Okay. I'm sorry, not the institution, but the students. They are prepared the results of the formative assessment or the final assessment.
Well, to answer your questions, actually they are not preferred summative assessment, but they actually perceive the formative one as a summative. So they only in my research, we found that students only or predominantly feel that they assess during the learning sessions, and they only care about the score.
There is one question, Marcella, in the chat.
Yes. Once they have a question, please use the chat box. So
there is question. There is question.
There is one question.
How to feel assessed?
Yeah, Marcella, do you want to read it?
How feel assessed in learning activities in your research?
Okay. Thank you for these questions. Actually, maybe this is a questions about the methodology. So I'm using the qualitative inquiry to assess how students actually feel about their feeling about being assessed in learning activity. So we use the qualitative inquiry. We use the focus group discussions, and we found the theme based on the transcript of focus group discussions.
So we ran out the time. There is another question, so but we need to move to another presenter.
Okay, yes, thank you, Ma'am Sylvia Sari. So let's go to the fourth presenter. So
Thank
it's your
you.
turn, Dr. Hani.
Yes, so maybe Ms. Sylvia could continue the discussion on the chat room because
Yes.
yeah. Okay, the next presenter is Dr. Arif Budi Yulianti to present the paper titled Effect of Unbalanced Nourishment on Growth Period to Memory in Mice. To Dr. Arif Budi Yulianti, the time is yours. Are you ready?
Yes, okay. I want to share my. Udah matiin satu, Bu. Matiin satu yang sorry.
Yeah, we can see that clearly. You can start now.
Okay.
Dr.
Thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thank you for the moderator, Dr. Hani Mufliha and Miss Marcelati Vera. The title our paper is Effect of Unbalanced Nourishment on the Growth Period to Memory in Mice. I am Arif Budi Yulianti. We have our team, Widayanti and Ike Rahmawati. We are from Faculty of Medicine. Introduction in my research. This paper is part of a research about stunting. We realize that balanced nourishment is very important, especially during growth period. So, quality of life in adult is determined by quality life of the embryo. Nutrition determines the quality of embryo life. Nourishment intake during the growth period is very necessary. Complete nourishment consists of a carbohydrate, protein, fat, vitamin, mineral, and water. In Indonesia, people have eating with high carbohydrate. In the period, development period, especially at under 5 years old, they need more protein calories that will result in growth barrier, such as stunted in the children under 13 years old. Stunting due to chronic undernutritional intake from the pregnancy until 2 years old. The stunting indicated in Indonesia reached 27.5%, or it can be said that 1 in 3 Indonesia children get stunting. Stunting is not only a small body size, but also has an impact on intelligence. In this study, mice were exposed to the nourishment condition during growth period and analyze memory with the new object recognition. The objective this study is to analyze the effect of unbalanced nourishment on growth period to memory in mice. The research methodology. We use 24 female mice strain DDY from Biopharma Bandung. 3 until 4 weeks after we wean off. The mice divide into 4 groups.
Group 1 is normal feeding, and the second group with high carbohydrate. Third group high protein, and fourth group high lipid. Every day, the mice have give 100 gram of feed, and the next day, the rest of the feed was weighed and replaced with the new feed. This done until the mice are pregnant and give birth. After pregnancy and before giving birth, the memory of the mice was measured using the NORD method and not new object recognition. That's right. Memorize and measure memory is the first to recognize mice recognize the location for 2 minutes, and then maybe you can solve. After that, one object was after that we give a new object and left the mice over 2 minutes. After that, one object was replaced with a new one and measured the time the mice recognized the new object. This is a composition of mice feed. Feed 1 is only standard feed mice feed CP551. The second feed is enriched with rice flour and cassava flour. And the third feed enriched with fish flour and Dutch albumin. And the fourth feed enriched with duck yolk and solid cow fat. We analyze the nutritional content of the mice feed, and this we look the feed 1 is standard feed, and is a high carbohydrate, and feed 2 is a high carbohydrate and 9.4% is carbohydrate, and the third feed is 5.1% of protein, and the fourth feed 0.51% of lipid. The result. Feed 1 to 4 calorie content per 100 gram were 77.6, and the second is 42.49, and the third is 148.76, and the fourth is 47.28 kilocalorie. The average consumption of the feed 1 to 4 per day per mouse was 6.5, 6.8, 7.1, and 6.2 gram, or equivalent to 5.04 kilocalorie, 2.89 kilocalorie, 10.8
6 kilocalorie, and the end is 0.96 kilocalorie. In this study, mice group with CP115 average consumption is 6.5 gram or 5.05 kilocalorie, and fit 2 average consumption 6.8 gram or 2.89 kilocalorie. The high protein feed average consumption is 7.1 or 10.56 calorie, and the feed 4 the high fat consumption 6.5 gram or 2.93 kilocalorie. The condition can be said that the amount of feed consumption is higher in this study, but the energy value is the smaller, is especially for feed 2 and 3.
Okay, it's already time.
Okay, okay. It is the new objectives recognized. I can in fit 1, 3, 2 relative decrease the mean of the mice fails to recognize the new objects, but in the fit 5 it is vice versa. The mean of the mice led to the recognize the new object. Maybe discuss conclusion is completely feeding effect memory decrease in mice. The mice exposures to the low calorie diet, so a memory decrease, although the differences were not statistically significant. This will be continue with the research in the second and the third generation with the aim to prove whether unbalanced nourishment in the first generation will pass on to the next generation. Thank you.
Okay, thank you, Dr. Arif Budi Yulianti. I think it is interesting to know how the food can change our brains. Is there any question from the audience? I haven't seen any question on the chat, so maybe I would start. So, Dr. Arif Budi Yulianti, it's very good study. I'm just wondering how you define the unbalanced nourishment, whether all the all the feed other than standards you define as unbalanced, or the standard one is balanced and the other is unbalanced, or how is it?
Okay, thank you. I combine with the standard of the feed for mice CP 551 in the feed 1, and then I enrich in feed 2 in rice flour and cassava flour. And then in the feed 3, we enrich with fish flour, and that's albumin, I'm sorry. And for feed is duck yolk and soluble cow fat. So I say it's unbalanced because it's enriched with like a high carbohydrate like rice flour or cassava flour is a very high carbohydrate. And then in feed 3, we enrich with fish flour and duck albumin.
And 4, we enrich with dark yolk, only the dark yolk, and soluble cow fat. So it is very different, like a texture or something like that. So it's very different exposure to mice, I think. Thank you.
Okay, thank you. Thank you for the answer. We have one question. Yeah, we have one question on the chat. Dr. Arif Budi, how to affect a core if we reduce our calorie consumption? So how the effects if we reduce the calorie consumptions? Would you explain? Yeah.
Okay, thank you. It is if we say in calorie value, I think in my research, FIT4 is a very low calorie value. So it's impact to the memorize. The memorize is very late to recognize a new object in fit a mouse in the fit 4. I think it's in this we can only carbohydrate, only protein, and it's very difficult. And in this time we value with calorie value. I measure with the calorie value. Thank you.
Okay, thank you so much. So we ran out the time. Yeah, we are moving to the next presenter. Yeah. We are moving to the next presenter. Thank you, Dr. Arif Budi.
Okay, I stop there.
Yeah, thank you. If there are any question, we still can continue on the chat box. So the next presenter is Ms. Anna Surgian Peterini with the title Extra Virgin Olive Oil Nano Emulsion Attenuated Inflammatory Response in LPS-Induced Sepsis. So, Ms. Anna, are you here?
Yes, I'm here.
Yeah, you could
Dr.
start.
Henry.
Yeah, please to share your screen.
Yes. Start. Is my presentation?
Yes.
Can see clearly?
Yeah, we can see that. Would you, yes, do the slideshow and you could start now.
Okay.
Yes.
Thank you very much, Dr. Henry and Mrs. Seila. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Healthy greeting also from us in Surabaya. In my respect to all colleagues in this meeting, nice to meet you. Thank you very much for this opportunity to present our research entitled Extra Virgin Olive Oil Nano-Emulsion Attenuated Inflammatory Response in LPS-Induced Sepsis. Yes. Please allow me to share a short story before I start this presentation. This study was started because our curiosity in finding a solution for enteral nutrition techniques for critical ill patients, especially case of sepsis. And then please allow me to start the introduction. Yes, as we know in critically ill, because we work in ICU, so we emphasize in sepsis case, where there is big problem when we give enteral nutrition. But on the other hand, most of literature or evidence show that giving enteral nutrition as early as possible will provide a better outcome than parenteral nutrition. We think to overcome this phenomenon, And the question arises whether the provision of certain nutrients or bioactive in nanoform maybe will be able to be a solution. Then we look for what materials are chosen to make this happen. Finally, we chose EVO, extra virgin olive oil, for various reasons. But actually, the simply reason when I wrote this research proposal, because EVO was stated clearly in Quran. Yeah, and some other reason why we choose EVO is because there is a lot of literature states that EVO is superfood, which contain antioxidant, anti-inflammatory, unsaturated fatty acid that good for our body and others. And also the other reason what prompted us to do this research is that fat is difficult to be absorbed by GIT. So the aim of this research was to develop EVO nanoemulsion as a nutrient carrier to improve its permeability while maintaining the intestinal mucous integrity in mouse model of LPS, lipopolysaccharide induced sepsis. And from the many bioactive ingredients in EVO, we hypothesize that oleic acid and hydroxytyrosol have the greatest role in the body after going through the literature research and what is popular now, computational research. Yes, and once again, before moving on to research method, we would like to emphasize again that we want to improve pharmacokinetic and pharmacodynamics of EVO for critically ill need. Now, how we did the research, let's continue the two materials and method. Yes, for the first, materials used to prepare EVO nanoemulsion were chromophore, RH40, PEG 400, and distilled water. The process and manufacture of EVO nanoemulsion using ultrasonication agitation method that has been submitted for publication. And we add EVO nanoemulsion to food given to the mice. The EVO brand we chose Lesino, that was chosen for this research because it was proven that this brand has the highest phenol content. We cite from Bayram et al. At 2012. The physical characterization of EVO nanoemulsion consisted of globule size and polydisparity index determination using particle size analyzer. And after we prepare the EVO nanoemulsion, we continue prepare the animal model. In this research, we chose mice. We chose male musculus mice, 10 until 12 weeks. And we separate into 10 groups of mice. Can we see in the screen in K1 is control group or non-sepsis model that was injected with PPS with the same volume with LPS. K2 is sepsis model with LPS injection 10 milligram per kilogram body weight intraperitoneally. And K1 and K2 must observe 1 hour to get sepsis condition. After 1 hour observation, we got clinical sepsis. These two groups were the pretest group to get the baseline data. Oh yeah, in this research we did pre-post-test control group with different sample. For post-test group, we did in K3 until K10. K3 is non-sepsis, we give AFO nanoemulsion. K4 is non-sepsis, but we did not give AFO nanoemulsion. K5 is sepsis model, we gave AFO nanoemulsion. K6 is sepsis model, we did not give AFO nanoemulsion. For K3 until K6, we sacrificed at 6 hours after injection LPS or PPS, and we collect sample to be measured the TNF-alpha, EL8, EL10, surfactant protein D, and histopathology examination in lung tissue. The 4 rest group consisted of K7 is non-sepsis, we gave AFO nanoemulsion. K8 is non-sepsis, we did not give AFO nanoemulsion. K9 is sepsis model, we give AFO nanoemulsion. K10 is sepsis model, we did not give AFO nanoemulsion. For K7 until K10, we sacrificed at 24th hour after injection LPS or PPS, and we collect sample to be measured. There were TNF-alpha, EL8, EL10, surfactant protein D, and histopathology in lung tissue. The reason why we separated into
You have 1 minute more.
Oh, 1 minute. 8 hours and 24 hours was because we tried to simulate the condition represented acute, subacute, and the beginning of chronic condition. And how we did analysis of lung biomarkers, inflammatory factors, and organ failure markers. The first step, of course, we assess sepsis mice, and then we get the blood to examine cytokines. This is the step how we did this research, how we did histological examination and data analysis did with SPSS one-way ANOVA. For the result, this slide show that the data support after LPS injection. This is one of clinical sign of sepsis. Beside clinical sign, it is significant different about the change of body temperature in mice sepsis model. And this is I just want to show that we did HE stain histopathology examination, but unfortunately for this measurement, we did not give any significant difference for K1 till K8. But K9 and K10, we have a significant difference, that is some change in histopathology examination. Yeah, it is the support data when we analyze with SPSS, K9 and K10 is significantly different. And after that, we do immunochemistry histopathology examination, and the result is the activity of NF-κB P65 was measured by determine the ratio between NF-κB P65 in the nucleus and cytoplasm. So we calculate by ourselves to get the activation of NF-κB, whether it was happen or not. And in this region, we saw that there was significant difference that in the group with sepsis mice model that we give nanoemulsion, AFO nanoemulsion, there is postponed NF-κB activity. So it is, I think it is maybe good news if we routinely consume AFO. Yeah. And this is the graph, that summary of my research. TNF alpha can decrease also, follow the decreasing of surfactant protein D in 24 hours after giving nanoemulsion EVO. And conclusion, the effect of EVO nanoemulsion through the enteral route of sepsis mice occurred 24 hours after administration. This phenomenon probably indicates that enteral absorption of EVO nanoemulsion can occur through sepsis mice. The mechanism that causes this decrease in circulating SPD level is probably through pro-inflammatory and anti-inflammatory cytokine pathways. But of course, further research needs to be continued to explore other paths, for example, genetic, the nature of infecting pathogen and environmental factor that may also affect SPD level. Thank you very much for the opportunity.
Okay, thank you. Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Anna. We already have 2 questions actually, so I'm just directly to reading the question from Dr. Yuniarti. Dr. Anna, why phenol content in EVO must be considered in your research?
Yes, thank you for the question. When I start this research, of course, there's a rising question. Actually, what is the bioactive that dominant in EVO? And I try to find in literature research that is state there is oleic acid in 78% and some small hydroxytyrosol. And I'm actually so curious about hydroxytyrosol. And before I start with this research, we did computational research to find out what is the effect of that hypothesis bioactive. Thank you.
So there is one more question, but I maybe need you to answer quickly because we run out of time. From Dr. Mirasari, after injected with LPS, how you know the mice really on the sepsis condition? Any measurements?
Yes, I'm sorry I did not state clearly in my presentation, but actually I examined with clinical sign by Gonzalez and Mariades, sorry, forget the name. They have the score to measure clinical sign of sepsis, and I support with the data of cytokine and the change of body temperature. Thank you.
Okay, thank you so much. The time is just up, and we just thank you, Dr. Anna. And we are moving to the last presenter actually. This is about it's very good, a fresh study about a drink from Dr. Maurizka Sabrina Setia, who want to present the organoleptic test of boba flour substitutions of pumpkin seed high in fiber and protein. So to Dr. Maurizka, are you here?
Yes.
Yeah, so I give the time and screen to you now, please.
Yes.
So we can see it. So you may start now, please.
Assalamualaikum warahmatullahi wabarakatuh. My name is Maurizka Sabrina Setia. I'm one of the authors on this paper, and this paper titled Organoleptic Test of Boba Flour Substitution of Pumpkin Seed high in fiber and protein. And this is what my research has been looking at. And so let's get started. In 2013, the obesity rate in Indonesia for people over the age of 18 was 14.8%. By 2018, the obesity rate had risen to 21.8%, obesity suffered in East Java province in 2013 until 2018 with the age of more than 8 years experienced a significant increase. In 2013, it was 8.4%, but by 2018, it had risen to 22.37%. Obesity is induced not just by exercise, sugar consumption, but also by a lack of fiber consumption such as vegetable and fruit. The daily fiber consumption of Manal City teenagers is 3.19 grams per day, which is much less than the recommended daily fiber consumption of 25 grams per day. Cucurbita muscota seed, also known as pumpkin seed, are good source of protein and fiber in Indonesia. According to BPS data, pumpkin production in Indonesia reached 942,928 kilograms in 2015, while it feels likely to 805,173 kilograms in 2016. Cucurbita muscota, sometimes known as pumpkin seed, are abundant fiber, protein, mineral, and antioxidant. Pumpkin seed contains 6 grams of fiber and 30.23 grams of protein per 100 grams, as well as 7.2 milligrams of zinc per 100 grams, double unsalted fatty acid, and phytosterol. Literature review: pumpkin, according to Zufahmi, 2014, in Fadila, 2018, pumpkin as Cucurbita muscota is a plant source of food with high nutritional content and fine fiber, making it easy to digest. Pumpkin seeds are high macronutrient, magnesium, phosphorus, and calcium, and low in micronutrients, calcium, magnesium, copper, and zinc. Pumpkin seed can be utilized as a good food supplement because they are high in nutrition value. Also, pumpkin is inexpensive. Pumpkin seeds are well known for their nutritional value. Furthermore, pumpkin seed can be used to treat joint inflammatory illness and prostate cancer. From research and methodology, I use a procedural model and the procedure utilized in research and development is based on opinion Brock and Gall 1984, and is then modified based on the research demand. In material and tools, tapioca flour, pumpkin seed flour, sugar, water, and food coloring were the ingredients used this research. Among the tools used were a blender, basin, Teflon, sieve, spatula, pan, knife, and memory oven. Making pumpkin seed flour, the initial step to select pumpkin seed with no skin should be utilized to avoid bitter taste. Furthermore, pumpkin seed without skin are mashed in blender for 8 until 10 minutes to make a powder, which then filtered through a sieve with a mesh size of 60 to get a homogeneous particle size and smooth texture. The pumpkin seeds are then stored at room temperature 28 until 30 degrees Celsius. For formula boba, ingredients pumpkin seed flour. In formula control 0, formula 1 4.5 gram, formula 2 6 gram, formula 3 13.5 gram. Tapioca flour in formula control 45 gram, formula 1 40.5 gram, formula 2 39 gram, and formula 3 31.5 gram. In palm sugar, water, and food coloring, the formulation is same. Organoleptic test assessment. The organoleptic test was performed using a hedonic scale or a scale to evaluate the level of preference consisting of 5 scales, very much like 5, very like 4, like 3, somewhat like 2, and dislike 1. Finding and discussion. In Table 1, the highest parameter assessment finding were discovered in the formula control, namely 4.33. The formula control and formula 3 formulation had the highest aroma rating, 4.33. The highest texture assessment was into the formula control and formula 3, namely
1 minute more.
Okay.
1 minute more.
Okay. Namely 4.4, and the panelists with a score of 4.33. Based on the analysis fragment test, all parameters, the results show that no difference in the panelist acceptance. In the table 2, untrained panelists without milk, the color parameter of the formulation control had the highest average score 3.33. The aroma parameter was ranked highest by the panelists in formulation 2 with a score 2.8p. The formulation control formulation has the highest texture rating of the 2.87. The formulation 2 had the highest test rating of 2.67. Based on analysis fragment, the texture and control parameter each show that there are different in the panelist acceptance, while in the aroma and taste parameter there is no difference. In table 3, the organoleptic assessment result of the 30 untrained panelists for boba product with complementary milk drink, specifically formula control and formula 1, which are the most favorite formulation with high average value for each parameter. Same in discussion, the panelist organoleptic, the finding for the boba formulation with pumpkin seed flour substitution, but based on 4 organoleptic parameters, namely color, aroma, texture, and taste, reveal that all formulations were acceptable for a boba without the addition of milk. The average score of the organoleptic test was 2.5 until 2.8, putting in the somewhat light category. Meanwhile, the panelists enjoy boba with the additional milk of a supplementary drink, namely milk. The average score of boba with milk is 2.9 until 3.4 or in category somewhat like. Conclusion and further research. The finding of organoleptic tests reveal that there was no significant difference in panelist acceptance of a parameter based on the judgment of trainee panelists on boba product. The result on organoleptic test on boba product without additional milk drink conducted by untrained panelists reveal a significant variation in color and texture parameters. Meanwhile, there was no significant change in panelist acceptance of the aroma and taste parameter, the result of untrained panelist evaluation of the boba with milk as combination drink. Not to be significant variation in the color and texture parameter were then present, and there is no significant difference in the aroma and taste parameter. Thank you.
Okay, thank you so much. So we only have 1 minute discussion and I couldn't find any questions. So maybe I would have question for you. So it's I think it's the creative idea to put healthy fiber into a popular boba drink. So thank you for doing this study, Mauriska. So do you want, I noticed in your results that the food 3, F3 had a highest pumpkin seed component, but it seems to have a high score of organoleptic test from the trained panelists, but it's in opposite by the untrained panelists. Would you talk about this? So it means the more highest component of pumpkin seed on the F3 have different score from the trained on the untrained panelists. Do you want to talk about this?
I'm sorry.
I can't hear you.
Hello?
Yes,
I can listen. Hello?
Oh, you have technical audio problem. Hello? Yes. Can you hear me?
Hello?
Hello?
Yeah.
Can you hear me?
Yeah.
Yeah, so I'm sorry. I think we ran out of time, but my question is, could you explain more why the trained panelists and untrained panelists have different score into feed 3? Like they have pumpkin seed, high pumpkin seed, but different score. Hello? Hello? I'm sorry, I think we have a technical problem with the audio from Ms. Mauriska. Should we continue to the next sessions, which is the token appreciation?
Hello?
Hello? Hello? I'm sorry, I will write on the chat.
Sorry, my internet is unstable.
Okay, that should be fine. That should be fine. No worry. No worries. I think you have a great presentation. The time is over for you. Thank you so much for coming, for presenting your research, Ms. Mauriska. Thank you. Okay, so I think we are coming to the last part of the session for giving the token of appreciation to all the presenters. I would like to invite Ms. Sheila to deliver the certificate or presentation to the first 3 presenters, please.
Yes, Dr. Hani. So let us Let us give the certificate of our presenters. Okay, so certificate of presentation for Mrs. Alia Persina, Faculty of Medicine, FKUnisba, is it correct? And certificate of presentation to Dr. Mia Kosmiati. Congratulations. Let's have virtual clap. And certificate of presentation for Mrs. Sylvia Mustika-Sari. Congratulations, Dr. Sylvia Mustika-Sari. Okay, virtual clap for the three presenters. Okay, so Dr. Henny.
Yes, thank you. The certificate is next for the Dr. Arif Budi Yulianti. Yes. Thank you for coming and presenting, Dr. Arif Budi. And then the next presenter is to Dr. Anna Surjan Veterini from Erlangga University. Congratulations and thank you for presenting your study. And then the last but not the least is to Ms. Mauriska Sabrina Septia from Universitas Negeri Malang. Thank you for presenting the study here. Yes, I think we are coming to the very last session. We are ending the session of last oral presentations. Dr. Sheila and I think we should return the event to the committee. Is there anything want to talk, Dr. Sheila?
I'd like to. Is it invitation? I'm sorry. There's a call. So maybe I'm going to invite Dr. Henny for testimonial.
Oh yeah, sure.
Okay. So may I call on the first presenter to say something about her experience in our program today. Yes. So may I call on Miss Aliana Persina for your testimonial in our presentation today. Or conference, I'm sorry, international conference. Miss Aliana. Yes.
Yeah. Okay. Hello.
Hello, Dr. Alia.
No, Dr. Alia, I think is not around.
Okay,
so I think we can pass this opportunity.
Yeah.
But once again, thank you so much, Dr. Henny, and also Ma'am Sela Vera for conducting a very great sharing session for the online presentation session. And both of you are successfully bring the discussion alive. Okay, once again, thank you so much,
Thank
Dr.
you.
Henny and Ma'am Sela. Okay. Okay. So I think we should back to Mr. Yusuf to get the e-group photos. Yeah.
Yeah, okay. Thank you so much once again, Dr. Henny Mufliha, Ph.D. and also Associate Lecturer, Mashila Fiera for the greeting sharing in this session. So for the next, congratulations to all presenters who already presenting their papers. All of you already done a really good job. And finally, thank you to all audiences and participants who already participated and actively involved in giving the questions, comments, feedback, and also even suggestion to the presenters. Your active participation and contribution in the scientific forum in the BICMHS 2021 is highly appreciated. And to memorize and cherish the moment, let's have a group photo together. Please turn on your Zoom video or camera in your device, please. I will start to count and take the e-group photo. Okay. Are you ready? Okay, for the first page, smile In 3, 2, 1. Okay. Yeah. For the second page, in 3, 2, 1. Yeah, next. For the third page, in 3, 2, 1. Okay, for the next page, in 3, 2, 1. Okay, for the last page, in 3, 2, 1. Okay, thank you for all the smiles and wonderful energy for today's e-group photo, and back to Miss Santi.
Okay, thank you so much. And I hope my sound is still audible and clear, because this is in my place is heavy rain. Okay, so before we start the next sessions, I would like to read some information again regarding the post-conference process. So this is to be remind to all the author or to the presenter who already submitted the conference paper and already published in the conference paper proceeding. So later on, don't forget you still have the chance to be published in the indexed international journals if you send your full paper, your extended full paper, which is already revised, at least 80% from your submitted conference paper to avoid the self-plagiarism. So you still have a chance to be published in the indexed international journals. And then later on, the ISSN, the DOI numbers for your article also will be assigned after the conference finish. So your article in the paper proceeding can be cited also and indexed in Google Scholars and soon to be submitted to the DOAG indexer. And don't forget to upload or send your full paper at least 14 days after this conference finish to your ScholarBrain account, or simply just email to the info@bicmhs.com. Okay, and this is, yeah. Okay, so moving on to the next agenda is the sharing sessions. So in this opportunity, I would like to give the opportunity to the representative of this session. So I would like to invite Dr. Sylvia Mustikasari. Dr. Sylvia Mustikasari, if you can hear me. Okay, already being the co-host. So you can unmute and you can share the experience of the two-day conference. Please welcome Dr. Sylvia.
Yes, thank you for this opportunity. But first, maybe I will say congratulations for Fakultas Kedokteran Unisba. This is a very great international conference. So my experience is really as great as this conference. So this is actually my first experience to be presented my medical education research in a huge medical science. So as we know that medical education is actually the kind of new science for a medical. So thank you so much for this opportunity.
Okay, thank you so much. And we're also glad to have you here in the BICMHS 2021. Thank you, Dr. Sylvia. Okay, so once again, to all the participants of the BICMHS 2021, do not forget to fill the feedback forms. Once again, do not forget to fill the feedback forms, and then also the committee also will send or type it in the Zoom chat box. Okay, so
yeah. Thank you, Ms. Santi. And also please remember that at 4 p.m. we will have closing ceremony. At the closing ceremony, there will be an announcement for the best presentation category and also for the best paper category and acknowledgement for session chairs. So please ensure that all the participants stay until the end of today's conference session. And now we will have a short break until 4 p.m. for the last plenary session. For the last plenary speaker session. Yeah.
Yeah, Mr. Yusuf, so we will continue because the plenary speaker session will be start at right now.
Okay.
Okay.
So
Okay,
what time,
so
Miss Santi?
right now.
Oh, okay.
Currently, right now.
Okay, so
Okay,
okay,
so
yeah.
okay, so allow me to bridging up and also give some informations and also clue for the technical committee. So
Recording
permissions-
stopped.
Yeah. So for this plenary speaker session, we will not record these sessions. So please kindly pay attention to give your full attention to our next plenary speaker sessions. Okay. And for the plenary speaker session, I think,
okay.
yeah, already here. Okay. You can continue. Okay. Please.
Okay. So as usual, before the session begin, I would like to read the moderator. The moderator profile, please. Yeah. Okay. So Mr. Rizky Suganda Dr. MKes Ph.D. from Universitas Islam Bandung, Indonesia. His PhD in prevention and treatment of lifestyle diseases from the Department of Nutrition, Exercise, and Sports, University of Copenhagen, Denmark, graduated 2020, and also Magister in Basic Medical Sciences, Medical Nutrition Concentration, Faculty of Medicine, Universitas Padjadjaran, Bandung, Indonesia, graduated from 2010, and also Medical Education Padjadjaran University or UNPAD, Universitas Padjadjaran, Bandung, Indonesia, graduated from 2006. And for the working experience, at the present, currently, he is lecture staff of the Faculty of Medicine at Universitas Islam Bandung, Indonesia, and also the medical doctor of PPSDM, Geominerba or Geology, Minerals and Coals, and also part-time doctor at Prodia Laboratory Buah Batu Branch Bandung, and also the general practitioners at Unisba Clinic Telemedicine. And this organizations, organizer of IDI Bandung Divisions of Science and Technology, and also organizer of BICMHS, Indonesian Medical Nutrition Doctors Association, West Java Branch and Center Scientific Division, and also he's a member of ASI, Association of Indonesian Sexologists, and also the member of AHI, Indonesian Hajj Health Association, and Perdoki, Indonesian Hajj Doctor Association. And now, to the moderator, Dr. Rizky Suganda, PhD, the time is yours to lead the plenary speaker session. Thank you.
Okay, thank you to the host for giving me the authority to guide this plenary session activities this afternoon. To the Honorable Chairperson of the conference, Miyaku Smiati, Dr. Medical Education Magister, PhD, to the Honorable Co-Conference Chair, Dr. Hendrati Dwi Mulyaningsih, and the organizing committee, to the speakers, Eva Wulff, and for all of the attendees, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good afternoon, best wishes for all of us. I'm very glad to see you here and welcome all of you to this plenary speaker session. This afternoon, the plenary session topic that we will discuss is about osteogenic training in musculoskeletal health promotion, why and how. And the speaker is Associate Professor Eva Wulff, PhD, MSc. Hi, Eva.
Hi, okay.
Morning,
Thank you very
Gordon.
much for your kind presentation of my presentation.
Ja, hvordan går
I'm
det?
very, sorry?
Hvordan går det?
Det går fint, tak.
Glad.
Very good.
Jeg hører det. Jeg også godt.
Unfortunately, I don't speak any Indonesian, but you speak a little Danish, so that's good.
Okay, I'm sorry for that. I asked her about how are you, how about Eva, and she said she's fine. Okay, before the speaker presenting her talk, I will tell you about her. Eva Wulff is Associate Professor at University of Copenhagen, Department of Nutrition, Exercise and Sports. Moreover, she is head of the bachelor's program in exercise and sports, as well as the master's program in human physiology. Eva received her PhD degree in 2005, entitled Exercise Training as a stimulus in osteogenic adaptation. From the perspective of the female athlete triad, what can be learned? In 2010, she was awarded as Teacher of the Year. Yeah. At Department of Exercise and Sport Science, the Faculty of Science, University of Copenhagen. In addition to her academic career, Eva has been a board member of the Danish elite sport organization, Team Denmark, and she has been an elite athlete in artistic gymnastics, obtaining 3 Danish championships. Wow, amazing, awesome. Before the speaker deliver her talk, I will convey some of the rules of this session as follows. Okay, we will give the speaker deliver his talk. As long as the speaker deliver her talk, there is no interruption. And if you have any question, you can type your question in chat box. Don't forget to mention your name, your company or your institution, then your question. After the speaker has delivered her talk, a discussion session will be open. These are the rules we will use. Now, we invite our first speaker, We will come Eva Wulff Helge. Please, the screen is yours.
Thank you very much, Iggy, for your kind presentation. I've been looking forward to present for you what is my main scientific interest, namely how we can improve the strength of our bones through exercise, and how exercise therefore must have a role in musculoskeletal health promotion. Thank you very much for your kind invitation. And I will start by sharing my screen. And if you can't hear me or you can't see anything of what I'm showing, please don't hesitate to say it to me. So I will share my screen. So, as Eki told just before, I'm associate professor at the University of Copenhagen, which is one of the oldest universities in the world. But this is our main building in the middle of Copenhagen, and our own buildings are not quite as old as what you can see from here. What I would talk about is how can we choose to be physically active? How can we choose exercise or sports that improve our bone health and our bone strength? And what I have shown here on the first slide is how different exercise or sports can be if you are older, if you are quite young, if you are elite athlete, if you are these guys here, they had prostate cancer, but even though they can play recreational football, you can make gymnastics in several ways, like here from a study we had, or here just from a recreational perspective. But whoever we want to improve bone, if it is young people, if it is old people, what we have to acknowledge is that the training or the stimulus that their skeleton is receiving has to be larger than if they were just living their habitual normal life. So osteoporosis is of course a key issue when you are talking about the skeleton. Osteoporosis is characterized by reduced bone mass, impairment of bone microarchitecture, which results in low bone strength and increased risk of bone fractures. So that is actually why we are so interested in trying to diminish the risk of getting osteoporosis. And as you can see from the picture over here, we know that the fractures is mostly seen in the vertebrae, in the hip, or in the wrist. And now this picture is showing a woman, and that is because normally osteoporosis is seen as a women illness. But actually, it is getting more and more frequently seen in male as well. So maybe this is just an old-fashioned picture of a woman, but you could even, with well argued, had the same situation for male persons. How important is this illness? And it is actually estimated to affect 200 million women worldwide. And you can see how many fractures that is associated to. And that is, of course, not only a problem for the individuals that suffered from osteoporosis, but it's also a very large disadvantage for the society, because it is very what's it called? It costs a lot of money to treat these patients. So therefore, we need to do something. Here you can see some pictures about when we see these different kind of fractures, which is normally associated with osteoporosis. And what you see on the y-axis here is the incidence. So 1,000, for example, per 100,000 person years. And you can see the prevalence or the incidence for women and the incidence for men. And what you actually see is, if we make a line here from 1,000, you can see for the hip fractures, which are the fractures which normally is seen only in a higher age range. For women, they meet this incidence around 75 years of age. But if you go to male, they meet this incidence when they are around 80 years of age. And this is, in that way, you can see that it is not so that men doesn't have these fractures, but they are getting these fractures in a later age. And you can see that for the other fractures, they are increasing more in women in a lower age groups. But in male, it is only the spine fractures, which also is increasing in the older ages. So we have for sure a problem here with the wrist and the spine fractures, which is also in the middle-aged women. So bone strength, what is deciding how strong bone is? You could imagine from these two photographies that this is a normal bone without osteoporosis, and you can see here a bone in the spine which have a very inconsistent pattern of trabeculae, and you can see there are places in the bone where there are actually no trabeculae because they have been absorbed in a place where the stress on the bone was too strong compared to what the bone could resist. You can also see that you have this compression of the spine, and that means that you are also getting a reduced height due to osteoporosis. So, but that is what we can see just by looking at the picture. But what is it really that determine the bone strength and the fragility of the bone? We have 3 grades of determinants. We have the macro scale, which is the blue one, especially in the top here. Then we have the nanoscale, And then we have the microarchitecture, which is in between the two others. So you can see the whole bone geometry. For example, bone size, the cortical thickness, and the cortical, that is what we have at the rim of the bone. So if the cortical thickness is high, then the bone is stronger. If the bone is large, then the bone is also stronger, regardless how it is in the microarchitecture. So a stronger bone will always be, a larger bone will always be stronger, even if the microarchitecture is the same than in a smaller bone. So if we go to microarchitecture, then the determinant is the trabecular connectivity, which you see is compromised over here. It's also the trabecular shape. If the trabecula are plate-like, the bone is stronger than if it is more like rods. And of course, also the cortical porosity is a determinant of the strength. So the more pores there are in the cortex, the weaker is the cortex, and not so supportive of the bone. And then at the nanoscale, we have osteocyte network integrity, and osteocytes are a kind of bone cells which is actually working in the transmission of the force that acts on bone and to the modeling of bone to getting stronger bones. So it could also be the hydration of bone, the amount of non-collagenous proteins, and the type and degree of the crosslinks which is in the collagen. So you can see there are many determinants from the macro to the nanoscale. So how can we measure this? Normally we use the DEXA scanning because it has a low radiation doses, so we can actually use it also when we want to measure bone mineral density or bone mass in children, whereas CT scanning has a much higher radiation dose. Then we have two other kinds of CT scans, but they are not applicable for the whole body, for the whole skeleton. It is only useful for minor regions of the skeleton, for example, an arm or a leg, and even if you are a very large man, for example, you can't use the micro CT because the dimensions are too small. So what can we do apart from that? We have these other kind of nanoscale measuring of the bone mass and density, but apart from that, we also can evaluate how the bone turnover is in the bone. So is it in a state where it is diminishing the bone mass, or is it in a state where there are more bone building than there are bone resorption? And in that situation, we would use bone turnover markers, and I will come back to that. So here you can see on two microscopic pictures again, where you see a young normal bone, where you see the different trabeculae, and here you see an osteoporotic bone. You can also see what I told about before, the resorption pits, where there are no trabeculae, which is a very weak place in the bone. And you can also see that here you see more plate-like trabeculae, like up here, compared to here, where there's a little bit of plate-like trabeculae up here, but most of the trabeculae are thinner, and some places they have just lost their connectivity with the place where they had a connection beforehand. So it is, when you're talking about osteogenic training, you have to be aware what are the normal how are the bone mass normally developing throughout life. So if you go from you are born and until around 70 years of age, you see that if you look at this full light line here, that is the normal development of peak of bone mass. So it increases up to around 20, maybe 25 years of age. And then you reach the peak bone mass here. From there on, the bone mass is stable until around the 50 years of age, and then it is decreasing after that. But you can also see that there are a line which is called the fracture threshold, an arbitrary line in this picture, but where you can see that if it is possible for the individual to increase bone mass to a higher level of peak bone mass, then along the developing years, you can see that it will take longer time before this line crosses the fracture threshold. So one strategy in health promotion would be to make children and young people physically active so that they can reach a higher peak bone mass, so that they actually have more bone mass in the bank, so to say. So when they are grown-up adults, they can bear to lose bone mass without reaching the fracture threshold. So what is going on is, to put some words on it, on what is going on in the bone. Here we have a bone modeling where we have more bone building than bone resorption. And then we have the bone remodeling, which is the main way of bone turnover in the adult life. So what we would like to study is that is it also important? We know that this is we know that it is possible to increase peak bone mass if you are living physically active years in the life in the younger years. But is it also possible to increase bone mass when you are in the adult years? That is actually a very important question that we are trying to study. So when we are measuring DEXA BMD, we have just short to sum up, because maybe it is not everybody of you that are known with DEXA and T-score and C-score, but it is important to say that when we measure bone mass, we have a C-score, which is a C-score of bone mass or bone mineral density, where it is compared with age and gender matched populations. So you can say that if you ever see a C-score, that assesses what is expected for this individual. So for example, if you are doing a DEXA scan on a 80-year-old woman, you will expect from the slide beforehand that her bone mineral density is lower than when she was 20 years of age, as for example also bone mass. So it is quite understandable that, and we know that during the years, our physical capacity is getting lesser. But this is not the C-score we use when we want to study if a person have osteoporosis. That is the T-score, because with the T-score we compare the measured BMD, bone mineral density, with the bone mineral density for young adults. So in that way, we compare with a desirable situation, the desirable understood as when we had our highest bone mineral density and our highest and our lowest risk of fracture. You can also see it from here that if the t-score is less than minus t.5, a standard deviation from the young value, then the diagnosis is osteoporosis. So I have tried to just given you a view of how it looks on the print. So here you have a 60-year-old woman, and you can see that how her C-score is as you would expect, because it is right at the average for her age, and the C-score is 0.1. So that means 0.1 standard deviation over the expected. But that is not what we want to know when we want to diagnose osteoporosis. That is the comparison to the young normal mean. So you can see when we compare to the young normal mean here, then the t-score is the distance from what we measured to the young age. And here you can see that the t-score is minus 1.1. So from osteoporosis view, this person doesn't have osteoporosis, but she has osteopenia, which is categorized as between minus 2.5 and minus 1.0. So you can see that she is at a minor risk of fracture, but it is expected that she is that when she is 60 years of age. So what is going on? Why are we getting weaker bones and with less bone mineral density with age and in osteoporosis? This is actually due to a change in bone turnover, because bone turnover is accelerated in osteoporosis, and the net bone balance in each remodeling unit is negative. So if you have a net bone balance in each bone remodeling unit, and you have an acceleration of the remodeling, then of course you will have a problem over years getting a lower and lower bone mineral density. So it is shown here as here you have the normal situation. You have a remodeling cycle of around 4 months in total. You have first 3 weeks where you have a resorption by the osteoclast, a resorption of bone. Then after resorption, you would have osteoblast, which is the bone forming cells, and they make an osteoid fill of the cavity that has just been made by the osteoclast. And at the very end, we will have a mineralization of the remodeling place. And here you can see that you have a totally balance. Here was the here you can see the amount of bone mass beforehand, and here you can see that the cavity that the osteoclast made in the bone, they are completely filled by mineralized bone. So here we have a remodeling cycle, which is just in balance and is just used for renewal of the bone mass due to necessity over the years in everyday life, like every other cells in the body. But in osteoporosis, you have, first of all, you have a diminished build up of bone mass in every bone remodeling unit. So you can see, if you compare here to this, you can see that it is not a completely coverage of what has been absorbed by the osteoclast. And at the same time, you can see that when more we have a higher bone turnover, we have an acceleration of the problem with negative net balance. So in osteoporosis, we have this doubling of the problem. This is just to show you how we can measure the bone turnover, which means that we can actually measure what is going on acutely after we have exercised or been physically active. And in that way, we don't have to wait 4 months, which is a remodeling cycle, before we can see if the training we made have an effect on the bones. So you can see if I have to wait for 4 months until I can evaluate by a DEXA scan if the training has been optimal, that is a bit of a long time for the patient and also for the scientist. So what we can do instead is to measure what is actually going on in the bone turnover. And we have some resorption markers and we have some formation markers. And the recommended resorption markers are primarily CTX. And we have formation markers where the recommended one are P1 and P, and the matrix proteins osteocalcin. So if we look at risk of bone fracture, you could also say, does a little difference have any significance in clinical view at all? And here you can see that if you have a change in bone mass of 1 standard deviation, that means a lower bone mass minus 1 standard deviation, you can see that the fracture risk in the spine is increased by almost double, in the hip times 2.4, and in the forearm 2.7. So even a relatively minor improvement or reduction in bone mass have an impact on the risk of fracture. You can see the same in this right panel, where you can see that if you have a T-score which is not too much diminished from 0, which was peak bone mass time, then you can see then it hasn't that much effect to change the T-score from, you could say, from minus 0.5 to -1. You can see this is very flat part of the curve. So over here, there's not so much change in the fracture risk. But if you are in, if you have osteoporosis, and you can change the T-score from, for example, -3 to -4, then you have a huge increase in fracture risk if you decrease it. But if you go in the, if you go in the opposite direction from -4 to -3, you will have a similar decrease in the fracture risk. So that is what we would like to be the case. So this is just to show the evidence of impact on peak bone mass, because as I said before, we can have 2 strategies to improve peak bone mass via the lifestyle factors when we are children or young, or we can have the strategy to improve bone mass and bone mineral density in the adult life. So this is for children and young people. What is the evidence to increase peak bone mass? And the 2 factors with the highest grade of evidence is calcium, increase the calcium intake through dairy products and what's it called? Vegetables, sorry. That is evidence A, but also the effect on bone mass and density has an effect on A when we look at physical activity and exercise. So there we have the 2 main evidences. When we will try to change bone turnover in a more positive direction to make it in a bone building way. It is again calcium, which is have a large impact. Also magnesium, potassium, vitamin D, and vitamin K, sex hormones, and what is my main issue is the mechanical loading. So I will talk a bit about that now. No, sorry. So mechanical loading is an osteogenic stimulus, and that is because the bone adaptation to loading shall ensure that the mass and the geometry, as I have talked about, and the material properties match the applied load. And here you can see some elite sport people, which actually have very large bone mass and very large bone mineral densities. So especially ball games, for example, badminton, or for example, gymnastics, which have a very diverse impact on the bone and very high forces. When we look at these athletes, we can see that they have really large bone mineral density. For example, this study, where you can see here, we compared elite gymnasts with controls, age-matched controls. So if the gymnasts had a higher bone mineral density, then the columns here would be in the positive directions compared to 0, which are the control. So you can see here that in all regions of the body, especially the artistic gymnast had a much larger bone mineral density. And if we go to the female racket players, where it was studied if there were difference in the playing arm compared to the non-dominant not playing arm, these studies showed that if you started before menarche, then there was around 20% difference from left to right hand in the humerus. And around menarche, 15% difference in the humerus, and in the post-menarche, if the participant started after menarche, then there was around 9% difference between right and left. So therefore, the hypothesis could be, when it is like this in elite athletes, can we transfer this to a health promotion perspective to get everybody to have a higher bone mass and bone mineral density and have a diminished risk of osteoporosis and fractures? So I think I'll step over this. So we have 5 general key training principles that is important to know when you want to use physical activity as a health promotion activity. And that is that whenever you want to be better in a certain training modus, that could be muscle strength, that could be flexibility, that could be, as here, bone mineral density, the training that you are doing must actually have an impact of course, specifically on the area you want to train or improve. So that is very important, and it also counts for bones, that I must know which part of the bone I would like to
Recording in
improve.
progress.
Sorry?
Recording stopped.
Yeah. Sorry. The load that we use must be higher than we are normally used to, because we have to make a training adaptation, and we only adapt if the stimulus are larger than we are used to. It's also, of course, like a pill. It usually only works when we take the pill. And the same with training. If we stop training, then there will go some time until we will go back to the value we had before we made the anabolic training and here the osteogenic training. And then if you have a very, very high bone mineral density in this case, but it could also be muscle strength, then it gets more difficult to make any increase. And of course, the volume, intensity, and the frequency of the training. So osteogenic training, how does it actually work? The mechanical stimulus that stimulate the bone building is bone deformation. Of course, not bone deformation that stays forever, but only an elastic bone deformation. That means there's a bone deformation, and when the mechanical stimulus is diminished, then the deformation stops and we go back to a normal situation. And that is what we call strain. So we know that this deformation is important in the osteogenic training, in the osteogenic stimulus. And what is more important than that, or even as important, is that the strain magnitude, that means the more the bone is deformed, the higher the strain is, the better the stimulus. The other thing is that the strain rate, that means the power that is applied to the bone. If it is applied very so slowly, you can say, if I pressure there, if I do it slowly, it is not as good as if I do it fast. So the size of the deformation, the rate, and the variation in the strain, different angles on the bone, for example. So it's not always like this. It could also be like that. It could be from here. It could be a torsion. The more variation on the bone, the higher potential for osteogenic improvement. Moreover, rest periods is important. And you could ask yourself, these forces that act on the bone, where do they actually come from? And they come from two sources, from ground reaction forces when we move. That means that we have to move with high accelerations and receiving large ground reaction forces from the ground. And moreover, we had to use the possibility to move in different directions. That is what we call odd impact. And that is what we, for example, have in recreational soccer, in badminton, in tennis, and so on, where you move in different directions all the time. Could also say orienteering in the woods, where you have to move between the trees. That is also a kind of odd impact, and gymnastics. And that is one source of strain. The other source is the muscle forces, which pulls onto the bone. And here you see how different the peak ground reactive forces can be in different kind of exercises. And in the lowest part here, you have the drop jump, where you see that the drop jump has 5.5 body weight in peak ground reaction forces, compared to, for example, walking, where it is 1.2. So we want to get to use the different kind of exercises, which is in the lower part here, if possible. We made a study of the acute response of biochemical bone turnover markers. Eki published that. And to see, can we see if we measure the ground reaction forces in different high impact exercises, different jumps in postmenopausal women, can we acutely see that there is a change in the bone turnover markers? And here on this panel, you can see that we have three kinds of jumps: counter movement jump, drop jump, and diagonal drop jump. And you can see here the ground reaction forces in three directions. This is the vertical direction. This is the sideways direction, or the mediolateral. And here you have the ground reaction forces in the anterior posterior direction. And what you can see is that the vertical ground reaction forces are the largest one in all the three jumps, and there are no differences between them. But if we go to the anteroposterior ground reaction force, you can see that the drop jump has the largest compared to the two other kind of jumps. And you can see here that if we go to the mediolateral ground reaction force, then the diagonal drop jump had a higher ground reaction force in that direction compared to the two other jumps. And then we wanted to see if we combine these ground reaction forces, Fx, Fy, and the Fc, and make a correlation to the change in P1 and P from pre-jumping to after jumping. Is there a correlation between them? And what we actually found was that there was a R-square value of 0.05, which means that actually 50% of the response in P1 and P is caused by the combined ground reaction force. So that shows us that it is important that we increase these ground reaction forces or optimize them when we want to make osteogenic training. So the other thing I mentioned was the strain magnitude, which was important when we want to make osteogenic training. Just want to see how much time I have left.
7 minutes.
How much again?
7 minutes left.
7 minutes, thank you. Here, strain magnitude. This study is with elderly men and women, and it lasted 6 months. They made some resistance exercises, but one group made them with low intensity. That means low weight, but more repetitions, 13 repetitions with 50% of 1 RM. You don't have to need what this is, but it just means that the load is lower up here than here when it is 80%. So they had fewer repetitions, but a larger weight. And what they found was that when we look into femoral neck, then only the group that made the had the high intensity training improved their bone mineral density in the femoral neck. But nothing happened with the two other groups, nothing statistically different. You can see there's a diminishment here in the control group, which could be due also to the normal reduction in bone mineral density in that age group. And then they also tried to study this with the bone turnover markers. And we have here the case that they compare the three groups before the training. There was no difference. But after the training, there was a significantly different amount of osteocalcin. When the legs, the low intensity group, was compared to the control, and when the high intensity group was compared to the low intensity and the control. And the same picture is seen is bone alkalic phosphatase, which is another bone formation marker. So that shows us, or could show us, that the difference in the response to the training is due to the change in bone turnover with the training that had been performed. Strain rate, as I told you, was another important factor, and here you can see another study where they had 2 years of supervised training, 2 times weekly, and you can read that by yourself. But they had 2 groups. One made what they called strength training. That means they had 4 seconds of concentric training and 4 seconds of eccentric training. So that means the same speed up and down. But the power training, in that they should make the concentric phase as fast as they could, but still had 4 seconds to get back to the baseline. And what they found here was that if they looked at the BMD in the lower spine, there was a significant difference between the groups, so that and here again, you have to be aware that it was people who were in the age where we normally diminish our bone mineral density. So here you can see that there is a diminishment in the slow or the strength training group. There is no diminishment in the power training group, and there is a statistically significant difference between them after these 2 years of training. In the total hip and the neck, there's also a significantly reduction in bone mass in the slow training group, but not in the power training group. But there was no difference between the 2 groups if you look at total hip or in the neck, but still it is promising regarding the importance of the strain rate.
Eva, maybe 3 minutes left.
Yeah, but Egi, are you aware that you used some of my time?
Yes.
Yeah,
I found that.
Very good. So if you have varied and novel strain, that is what we call odd impact. And you can actually, from these pictures, see that, okay, here you have some joggers, and you can actually intuitively get the impression that here, that is not very varied, ground reaction forces from the ground. They are just continuing very repetitively to do this. But if you look at, for example, soccer, you can actually get the intuitively impression that here we have forces which are, during the game, are acting in different directions on the body. And that is what we would like to go for. So I think I will skip this. No, this is here we have tried to study lifelong recreational football from a cross-sectional study to see does it matter to continue your training from you are young to you are old. And here you can see that we have untrained elderly group, and we have a football trained elderly group who has trained from they were young. And you can see that there's a significantly different bone mineral density in the whole body between the 2 groups. So it matters to continue your training into old age. But you can also see that if you look over here where you have the bone mineral density in total legs, that even if we compare the football trained elderly to the young untrained people, then there is a significantly different in total legs BMD, where the elderly people being higher than the young untrained people. So this study where we compared football or resistance training, what you can see here is that they trained for 1 year, and you can see that they were 68 years of age. And you can see here that if we are looking at the BMD in right femoral neck, there is a significantly in the football group, there's a significantly increase after 12 months compared to 4 months and from baseline. The same in the left leg. And if you go, but there were no difference in the resistance training group and in the controls over the 0, 4, and 12 months. If we go to the bone turnover markers, we see the same, that in the football group there is an increase in the bone turnover, in the bone formation marker P1 and P after 4 months and after 12 months, also in osteocalcin, but with no change in the resistance and the control group. So unfortunately, I can't show you this because I don't have the time, but it is actually a video showing what the training looks like when we are training elder men, because of course it's not the same kind of football training than if it was elite football players, but it is differentiated to the actual population. So, can I have 5 minutes?
Okay. Yes, Eva.
Thank you. So, football, and you could compare it to handball or to tennis, badminton, different kind of ball games. I would like to go to gymnastics, which also have a varied kind of training. And we made a study, over 19 weeks, 11 weeks of detraining, and the training was supervised. It was high impact, as we aimed upon odd impact. There was some resistance exercises, and what we asked the participants to do was to, whatever you do, you have to do your maximal effort. So we didn't measure, are you doing it? But we just made them self-responsible for the training in that way. When you are to this supervised training, you have to do all you can and as fast you can. And then we worked with few repetitions in an interval training manner. And we had 2 groups, a postmenopausal group and a premenopausal group, and an intervention group and a control group. And here you see some of the different kind of training exercises we made, using the arms also, jumping on a air track, using arms again, making different kind of stomping and jumps forth and back and in different direction. And we used the stairs. And what we actually found was that after these 19 weeks of training, when we compared the postmenopausal group, the intervention group, which is here, and the control groups, which is here, we found that in total proximal femur, left, right and left, in right trochanter, in proximal femoral shaft, right and left, and in the lower spine, in all these regions, we saw a significant and even highly significant in some regions difference from pre to post, but also between the groups. That means between the intervention group and the control group. And you can see here that figure of it, that we had these large increase in the training group, in the postmenopausal group, and we had a reduction during the 19 weeks in the control group, which means that we found these between group differences. So I would say that apart from measuring the bone turnover, the bone mineral density, we measured also the bone turnover markers, which was in agreement with the BMD measurement, showing that it was the bone turnover we had changed by changing the formation without any change in the resorption. So we had a really anabolic impact of the training by changing the formation. So if we should give some guidelines, and this is my two last slides, you can say for the participant, what you have to say is that, okay, you must train hard. It is really hard training, but you must do all you can yourself. You must have the feeling that okay, I should really use my muscles. I should exercise with a variation. I have to move fast, but also to take breaks. I also have to stop my speed fast if I can. And jump is really good because that also improves the bone, the ground reaction forces from the ground. And then, of course, be active as a child. That can't always be done when it has passed, but it's also important to continue throughout life, of course. And most of all, find something that you enjoy, because we know everybody that if we don't enjoy what we are doing, then it's too easy to get good excuses for ourselves. Okay, I have no time today, but if I find it enjoyable, then I might do it more often. And if we have looking at the patient, some of the principles are the same, but what you also must do is train your postural balance. Train your balance when you move around. Torsion with resistance is prohibited, and no forward bending of the spine, because that is really important. If you have weak bones due to osteoporosis, the skeleton or the lower back will not be strong enough to withstand these forces. So this is really important. No torsional resistance, no forward bending of the spine. And it's more safe to go from resistance training to weight bearing. So firstly, you have to train your postural balance, and you have to train your muscle strength so that you don't fall over, of course, and have a body control, core training. And move from slow to fast so that you can coordinate and have the control before you go to the very fast movements. So this was the end for me. I hope that I didn't take too much of your time. Thank you.
Okay. Thank you, Eva, for a very nice presentation. Well, ladies and gentlemen, now we are come to the discussion session. To all attendees, please, if you have any questions, you can type your questions on the chat box. Don't forget to mention your name and your institution, then your question. Okay. Thanks for it. Okay. There is one question here. The first question coming from Dr. Mia. Thanks for your great talk, Prof. Eva Wulff. I have two questions. Maybe this likely not related to your topic, but it's still important. How effective giving supplement calcium to improve the condition of osteoporosis at elderly person? Maybe this is not Eva, but do you have in mind regarding this? Eva?
Yeah, I think regarding calcium, as you also say, Mia, that is not exactly my research area. But what I know about it is that it depends really much how is your calcium status, because if you are having satisfying amounts of calcium in your body system, then you shouldn't try to improve calcium, because that have some other risk regarding cardiovascular disease, for example, or other diseases. And that is just, if you try to improve more than is necessary, then you just use your money for something unnecessary. So if you are too low in calcium, then there's evidence, as I also showed on one slide, there's evidence that it is important to have a satisfied level of calcium in accordance with the recommendations. So it really depends how important it is, and in that way it could be like also comparable to the osteogenic training, that okay, if you already have a very strong bone with large bone mass and large bone mineral densities, okay, then you don't need, maybe you like it, but then you don't need to improve your bone mass and do a lot of training just for that reason. So you have to know what are the status for the bones, what are the status for calcium, for vitamin D, before you can make the right health promotional initiative in combination. Was that?
Yeah.
Was that good? But you had another question,
yes.
which I didn't ask. How frequent and how at risk osteogenic
training, the possibility of musculoskeletal injury?
Yeah. Well, was that related to the last slides, or
maybe is there a maximum times of training that could be possibility to musculoskeletal injury? Okay, you don't have to 4 times a week, or maybe something like that.
Yeah, I think that so the gymnastic study that I showed you some results from, there we had, or the participants, but also us who supervised it, had 3 times, they trained 3 times weekly for half an hour effective training each week. And then there was some, maybe a quarter of an hour where they met and talked together, and then we started, but only half an hour of effective training. And you could see the improvements they got during these, let us say 20 weeks, it was 19, but 20 weeks, was around 2 to 4 percentage of increase in bone mineral density. And that was from the beginning. They were untrained, and they were the postmenopausal group was, of course, postmenopausal, so it was not young fit people. We had some of the participants, only a couple of them, but they were 70 years of age and hadn't been training before. So I would say that the most important is that, of course, you have a DEXA scan beforehand, if it is patients. And if you really have a huge, try to make a huge impact in the anabolic of the anabolic training, then it is nice to know and at least ask the participants if they are diagnosed with osteoporosis. And if they are diagnosed, then of course you have to take more care than if they are just osteopenic or having a normal bone mineral density. But it's good to know, or it's necessary to know, if they are diagnosed with osteoporosis. And even if they are diagnosed with osteoporosis, if they are young, then it is not as risky to make a huge impact, because they still have if you take the first microscopic photographs, then you could see that when you are getting this very loose and unorderly trabeculae, then the risk of breaking the bones are larger than if it is more regular. And in the younger years, even if you have osteoporosis, then the microarchitecture is more orderly arranged. So in the younger, you can say up to 40, for example, then it is more orderly arranged, the trabeculae, so it is not that risky to make this impact on the bones. But of course, if you are 80, then you have, if you both have a diagnosis of osteoporosis, and during the normal aging, some of the trabeculae have got smaller and the connectivity has got less, then the risk is larger. So if it is old people, it is nice to know their bone mineral density, but it is absolutely necessary to know if they are diagnosed with osteoporosis. And if you are in the old population, then as I had the last slide, or yeah, almost, stated that, okay, you have to try to improve normal body control before you go on to have large impacts. And that is, of course, also why if you have a low muscle strength, then you are more prone to fall over, and then you are more in risk of having a fracture. So in that way, you can't separate the bone mass strength, the bone health, and the muscle strength. You have to be aware of both, because of course you can't say, okay, you have to jump like a kangaroo and be very energetic all the time if you don't have the muscle strength that can stop you from falling if you make some miscoordination. So you have, of course, to be aware of both, but you can see that, but you know that the muscle strength, if you improve that doing resistance training, that will also improve your bone strength, due to the muscles when they are attached to the bones, when they contract, they will pull in the bone. So it's not only the ground reaction forces from high impact exercise, but it is also the resistance training. And that was why I, in the last slide, also recommended that start with resistance training, so that you get fitter regarding muscle strength. The resistance training will also improve your bone mass and your bone mineral density. And then when you are ready for that, you can progress to more intense high impact and odd impact exercise. And you can do it several places. I mean, some people, they think, okay, I have never played anything like football or something. Can I do it? But you can really manipulate the way you are playing football for people who are unaccustomed to it. And if you don't like ball games, find something that you like. Maybe you like the nature. Maybe you can jump up and down from, I don't know what it's called in English, but things in the nature, wood, like this, where you not higher than that. You can go up to that, jump down. So there are a lot of circumstances in the nature, in the parks, in your home, which you can use to improve these ground reaction forces without necessarily start by going to recreational football or gymnastics. So find something that is possible in life. Find something that you like do. If you hate nature, then it would be stupid to say, okay, I will start by doing exercise in the nature. You have to find out something else, and maybe find a buddy you can do it together with, so that when you have a day where you think, oh, I'm too lazy today, then your body will say, ah, come on. Now we have to do it today. That was our agreement. So bodies are good.
Yes. Thank you, Eva. So, Dr. Mia, it will the difference between the trained and
Thank
untrained
you, Prof.
people,
Eva.
and
Sorry?
it has
Thank
to know
you
about.
for your answer, Prof. Okay.
Thank you for the answer, Eva. She said thank you for the answer.
You're
And
welcome.
it will be nice to know if you have examined by the DEXA scan to know the bone mineral density or the lean mass. Okay. Okay. There are another two questions. Maybe you would like to choose between from Mirasari or Dr. Susanti Damika, because I think the time is running.
Was it a question to me?
Yes, Eva.
What did you say?
Would you choose the question from Mirasari or Susanti Dharmika? Because we are running out of time, so I think.
So should I ask? Should I try to answer this question from Susanti?
Yes. Okay.
Yeah.
From Susanti Dharmika, for osteoporosis patient, which exercise should be prioritized? Is it for the arms, legs, or trunk? And two, which one is more functional? Is it strength training or power training? And is there any correlation between sarcopenia and osteoporosis? Yes,
yeah.
Maybe that
yeah.
be the last.
Try to answer that. I think what should be prioritized, I would say that for most for most people, and therefore also osteoporosis patients, what we need to do in everyday life, it is to walk around. So we have to move, and therefore I would say that okay, it's good to be to improve bone mass in the arms, but it is more important to improve the legs and the trunk. Because the legs, because you move around on your legs, the trunk, because that is it is often prone to in the trunk you get some control of your coordination so that you can diminish the risk of fall. So together the trunk and the legs will make you will diminish the risk of falling, and that is really important. And I would say that the power training is important, and it seems so when you say power training. Okay, can you do that as an osteoporotic patient? But it is very much about the principles that, okay, power training, it means that you try to move fast. And that just means not move distance fast, but also on the spot where you are, try to move fast. When you jump, try to really press down in the ground. Try to do these fast movements. But of course, first when you are ready for it. So it's really important that this about controlling your body in movement, because if you are not able to do that, then you shouldn't, of course, do these very fast and forceful movements, because if the movements you are doing doesn't succeed as you intended, then you are really at risk of falling if you don't have the kind of body control. So and body control is good for everything apart from that. But therefore I think that I would start with legs and trunk, because that is very important. And if and later on I will couple the arms onto that. But again, as from this gymnastic study we made only maybe 6 to 10 repetitions of every exercise. So then maybe we came back to that afterwards, but we only had a short and very active and forceful performance in every kind of exercise, then we move to a new kind of exercise, and then to another one, and then to another one, and maybe we then went back to the first one. So in that sense, I would say that if you want to make the training diverse, it will be possible to include both trunk, legs, and arms, because it doesn't afford so much time. It affords intensity, but not so much time. And is there any correlation between sarcopenia and osteoporosis? And certainly there is that. Yeah. Maybe not for the specific, not necessarily for the specific patient, but of course, if you look on a larger population, then there are certainly a correlation between sarcopenia and osteoporosis.
Okay, Eva. Thank you very much.
You're welcome.
Eva, very inspiring. The time, I'm sorry, but the time is running out, and hopefully this presentation will give benefit for us. And ladies and gentlemen, finally,
I had one question, Nikki.
Okay, Eva.
Yeah, it was just these two videos, which I didn't have time to share with you.
Yeah.
Could I in some way send it to you or somebody else? If anybody would like to see what does it actually look like? Have you any possibility of sharing with the participants? Is there any things sent out or what? You know, I just send it to you, Iki, and then you can find out if it is possible to share it.
Yeah.
Right?
Yeah. Okay. Okay. Thank you. Okay. Okay. Thanks, Eva. Ladies and gentlemen, finally approaching to the end. We would like to say thanks again for Eva Wulff, Helge this afternoon, and all for the audience for active participation. The conclusion of presentation this afternoon is there are many strategies to increase the peak bone mass, the high evidence according to Weaver 2016. From macronutrients, there are calcium, that the high evidence, very high, grade A evidence, and then vitamin D. And a physical activity and exercise effect on bone mass and density, also grade A evidence. Okay, and mechanical loading as an osteogenic stimulus, the central objective is site-specific adaptation to increase bone strength and prevent overuse and bone fractures. Which kind of exercise? That the exercise that affect on bone mass, that is the exercise that applied the 5 general key training principles: the specificity, overload, reversibility, initial values, and diminishing returns. And it also considering the training dose, the training volume, the intensity of the training, and the frequency of training. And then the strain rate should be high to stimulate bone formation, so as fast as you can, and the magnitude should be high as well to stimulate bone formation, and also the strain distribution applied to the bone, so from various direction. The varied and novel strain, the odd impact such as football, the bone mass and the bone turnover marker are higher after 15 weeks of soccer training than moderate intensity or swimming in premenopausal women, in middle-aged elderly pre-diabetics, and in elderly men. So hopefully this session will give benefit for us. Thank you for your attention, and thank you very much for Eva for coming here today. Eva, thank you very much.
You're very welcome. Yeah. I hope that you have had a good conference.
Yes, thank you. See you later, and I see you as well to the attendees. Now I will give the session to the host again.
Well,
Dr. Ahmad Yusuf.
thank you. Well, thank you so much, Dr. Rizky Suganda, PhD, as the moderator and Associate Professor Eva Wulff Health, Ph.D. MSc. Thank you very much, Professor.
Welcome.
We learned a lot of we learned a lot from the topic in the discussion. Thank you. And back to Ms. Santi.
Okay. And as the recognition and also the appreciations from the BICMHS committee, we would like to give a token of appreciations to our plenary speaker and also the moderator. So please welcome Dr. Alia
Recording in
Tursina.
progress.
Please welcome Dr. Alia Tursina, SPN MHS, the Head of Study Program of Medical Education, Medical Faculty of Unisba, to give a token of appreciations to Associate Professor Eva Wulff Health from University of Copenhagen, Denmark, as the plenary speaker. Please welcome Dr. Alia.
Thank you, Dr. Santi. Thank you for the very interesting and very clearly presentation, Prof. Eva. We will give you an e-certificate as token of our appreciation for you. I hope next time we can collaborate and meet in Bandung.
That would be great.
Please, big applause for Prof. Eva.
Yeah.
You're welcome.
Okay, thank you so much once again, Associate Professor Eva Wulff Health. Glad to have you here in our forum. Okay, next, please welcome again Dr. Alia Tursina as the Head of Study Program of Medical Education, the Medical Faculty Unisba, to give a token of appreciations to our moderators today, Dr. Rizky Suganda, PhD. Please welcome again Dr. Alia Tursina.
Okay, Dr. Rizky, thank you for being a moderator and guiding this event very well. Please, big applause to Dr. Rizky.
Welcome.
Yes. Thanks again for both of you.
Okay, thank you, Dr. Alia Tursina and also Dr. Rizky Suganda, PhD. And once again, ladies and gentlemen, welcome back. And also this is almost the end of our session of the second day of the BICMHS. So thank you for all the excitement of the today for BICMH. Starting yesterday until today, we have finished the second day of the Bandung International Conference on Medical and Health Sciences, or BICMHS 2021. As we have already informed at the opening ceremony this morning, by the end of today's conference, we will announce the best presentation, the best paper award, and also the recognition for the session chair.
Okay, well, ladies and gentlemen, this time we will announce the best presentation and best paper awards. And for the first category is for the best presentation awards. The session chair had selected the best presentation based on several criteria and in the assessment form. And thank you to all presenters that gave his or her best efforts in delivering the research through the presentation. And there are two best presentations for today's conference, since they achieved the same score. So we congratulate to both of them and they deserve the awards. On this occasion, we would like to give a special token of appreciation and recognition to the best presentation. Kindly please welcome Ibu Mia Kusmiati, Dr. MPD Kedokteran PhD as the conference chair of BICMHS 2021 to give away e-certificates as the token of appreciation and recognition to the best presentation.
Okay, thank you very much, Mr. Yusuf and Mrs. Santi. Ladies and gentlemen, as mentioned the ceremony before, talk that we have two best presenters today and one best paper and best session chair for today's conference. Based on the assessing from our chair session and moderator of online presentation, the winner best presenter today are, please, Miss. Okay. Dr. Meta Maulida Damayanti for the winner best presenter today. And the others, Ms. Santi or Mr. Yusuf?
Okay, moving on to Miss Santi. Okay.
Okay, the best presenter, the second or maybe it's the same spot is Sylvia Mustikasari from Fakulty Medicine, Universitas Jenderal Ahmad Yani. Congratulations to you all, Dr. Neta and Dr. Sylvia Mustika. And the best session chair.
Sorry, Dr. Mia.
Okay.
Okay, I will continue first.
Okay.
Okay. Okay, thank you once again, Dr. Mia, and also congratulations to the best presenter. So moving on to our second award category is for the best paper. So the conference scientific review committee already select the best paper during the submission and also the review process. And the criteria for determining the best paper are format or template alignment, good scientific writing structure, academic English language, the novelty of the content, the research gap filling, the academic, practical, and also societal contributions, the clear limitation, and also the good mapping of future research. In the BICMHS 2021, we have one best paper. And on this occasion, we would like to give a special token of appreciation and also the recognition. So once again, please welcome Dr. Mia Kusmiati, Ph.D. to congratulate the Best Paper Award. Still mute, Bu?
Thank you very much, Ms. Santi and Mr. Yusuf, for the best paper for this conference. We will decide and we will choose to, please, okay, the neutrophil lymphocyte ratio and C-reactive protein in non-dialysis chronic kidney disease. And the first author or authors for this article is Profesor Nuzirwan Acham, Ph.D. KHOM, Budiman S. Susilawati, K. Pidiate. Congratulations to Prof. Achang and the team. Nggak ada ya? Are you still been here, Prof. Achang? Congratulations. Ada ya? Okay.
Okay. Okay. Thank you once again, Dr. Mia. And also congratulations to the best paper. Okay, we still have another category, the last category. Moving on to our third award category is for the session chairs. And on behalf of the organizing committee, we would like to appreciate and also thank all the session chair today, because with his and her contribution during the presentation session, the scientific forum, feedback discussion, and also the knowledge sharing environment has been built to encourage participation from all the participants. So once again, Dr. Mia Kusmiati, Ph.D. as the conference chair of the BICMHS to give away the certificates and also the token of appreciations to all of our session chairs.
Okay, thank you again, Ms. Santi and Mr. Yusuf. And this is maybe the last but not least for the winner, best session chair for today's conference, yesterday and today. And the winner for best session chair is
for all the session chairs. Yeah.
Please, Ms. Santi or Mr. Yusuf.
Yeah. Okay. Next slide, please. This is to all the session chairs, Bu. To all.
Okay, to Prof. Herigarna, dokter spesialis anak konsultan Ph.D. from Faculty of Medicine, Universitas Islam Bandung, Indonesia, in recognition and appreciation as the best session chair. Congratulations, Prof. Herigarna.
We still have, Bu.
Okay. And the other Dr. Kasine Kateleka, the researcher, the Halal Science Center, Chulalongkorn University, Bangkok, Thailand, in recognition and appreciation as session chair today. Congratulations to Dr. Kasine Kateleka.
Two more.
And then for the third session chair is the Dr. Heni Muflihah, MKes, Ph.D. from Faculty of Medicine Universitas Islam Bandung, Indonesia, in recognition and appreciation as session chair. Okay, lagi. Yeah. And the last, we also thank you to Associate Lecturer Masila Tevera from Arellano University, Philippines, in recognition and appreciation as session chair. Congratulations to you all to contributing for our conference.
Okay. Thank you so much, Dr. Mia Kusmiati, for giving, and also congratulates to all the awardee of the second day or the last day of the Bandung International Conference on Medical and Health Sciences.
Well, for the next, I would like to invite Professor Dr. Nanan Sekarwana, Dr. SPH-KEYNOTE, as the Dean of Medical Faculty, Universitas Islam Bandung, Indonesia, to deliver the closing speech of the second day of BICMHS 2021. And please welcome Professor Dr. Nanan Sekarwana. Time is yours.
Thank you, Mr. Yusuf and Mrs. Santi. Bismillahirrahmanirrahim. The Honorable First Vice Dean, Dr. Santun, the Second Vice Dean, Dr. Lisa, the Third Vice Dean, Dr. Jurmansa, Professor and member of the Academic Senate of the Faculty of Medicine, structural and functional official within the Faculty of Medicine, Head of Preclinical and Clinical Department of Faculty of Medicine, lecturer, Head of Alumni Association, student, and all BICMHS participants. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillah, alhamdulillahillazi arsala rasuluhu bil huda wa dinil haq liyudhira lahu ala dini kullihi walau karihal musyrikun. Asyhadu an la ilaha illallah wahdahu la syarikalah wa asyhadu anna Muhammadan abduhu wa rasuluh. Allahumma shalli wa sallim wa barik ala habibina Muhammadin wa ala alihi wa ashabihi ajmain wa man tabi'ahu bi ihsanin ila yaumidin. Rabbi shrahli sadri wa yassir li amri wahlul 'uqdatam min lisani yafqahu qawli, amma ba'du. Alhamdulillahirabbil alamin. Praise and gratitude we pray to Allah Subhanahu wa Ta'ala, because for His grace we are still given the blessing of health until the end of the Bandung International Conference on Medical and Health Science, which has been going on for 2 days. Salawat and greetings do not forget to extend to our Lord, the Prophet Muhammad sallallahu alaihi wasallam, to his family, friends, tabi'in, tabi'ut tabi'ahum, to all of us, and to all his people until the end of time. Ladies and gentlemen, in the past 2 days, we have kind of lot of new knowledge, both through plenary session and online presentation research. We have heard about the role of the academic health system in improving health services, utilizing technology advance to improve the quality of learning, health problem in the view of Islam and bioethic, and so on. Of course, this opens our horizon for all of us about the importance of conducting quality research to keep trying to find new breakthroughs that can be applied in better health services. Opportunities to get research grant and community service are very wide open. Sources of fund can be obtained from faculty, universities, government, and other source from within and outside the country. If we can conduct collaborative research involving several researchers from various countries, it will certainly be better and more open source of funding are obtained to large amounts. We hope that the participant and presenter can continue to establish communication, which in the future can be followed up by signing a collaboration. To all speakers, Professor Dr. Omar Hasan Kasule from King Fahd University, Saudi Arabia. From Tatsuya Iso, Medical Doctor Ph.D. from Gunma University of Health and Welfare, Japan. Associate Professor Dr. Badrul Hisham bin Yahaya, Ph.D. from University Science Malaysia, Malaysia. And Associate Professor Eva Wulff, Ph.D. MSc from University of Copenhagen, Denmark. Mrs. Aliza Ramli from Republic Polytechnic, Singapore. And on behalf of the Medical Faculty of UNISBA, thank you profusely for sharing your knowledge at this event. I also thank to all the moderators who managed the plenary and parallel presentations well. We also don't forget to express our gratitude to the Research Synergy Foundation and UPT Scientific Publication UNISBA who supported this event. Finally, of course, I would like to thank the entire committee team who have worked hard to organize this event. May Allah subhanahu wa ta'ala give you a worthy reward and be good deed that will be the way to his heaven in the future. Amin. On behalf of the Medical Faculty of UNISBA, we also apologize if in the implementation of this conference activities, there were things that didn't go as expected. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much, Professor Dr. Nanan Sekarwana, for the closing speech. And back to Ms. Santi.
Thank you. So finally, we are at the end of the Bandung International Conference on Medical and Health Sciences, BICMHS, Day 2. So ladies and gentlemen, distinguished participants and attendees, we hope it has been a beneficial and also fruitful day for all of you. And on behalf of the host or the organizing committee, the Medical Faculty of Universitas Islam Bandung or FKUnisba, Research Synergy Foundation, and UPT Publikasi Ilmiah Unisba, also supported by the Scholar Vein Research Synergy Institute, Reviewer Press, F1000 Research, and also the Taylor and Francis Group, in conjunction with the 17th anniversary or the Milad of the Medical Faculty of Universitas Islam Bandung.
And once again, thank you all the distinguished participants for taking the time to join us in Bandung International Conference and Medical and Health Sciences 2021, since the first day until today as the last day. And it has been our pleasure to host this virtual conference. See you again in the upcoming BICMHS 2022 next year, inshallah. Please take a good rest, stay healthy and safe. Thank you very much, and wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Is there any group photo? Is there any?
Yes.